University of Minnesota is showing that if we do the right thing, then we get 2D cross validation, 2D Q square. If we do the wrong thing, we get naive or wrong Q square. We have written some paper on these ideas. Now here I am showing if we do wrong thing, what happens with the air water partition coefficient with the ridge regression. When you did the true, Q square is 0.82. When you do the wrong way, ordinary least square, it becomes 934. That shows if you do the wrong way, it shows a pretty picture, but that's not really pretty. Here is one example. We took the APS data over 1,000 chemicals. That is a model we got using simple mathematical graph theoretical descriptors. And then we also did some hierarchical approach with our collaborator, Dr. Balasubramanian, who is a quantum chemist, that can we use the topological geometrical quantum numerical descriptor in a hierarchical way. So we did these two levels of calculation. And if we look at the different predictor one at a time, you can see that R square. But then if we take the hierarchical approach, we see if we take the topological approaches, R square 0.81, and then they go on increasing. Finally, at a very high level, we get some improvement. But the point is, this costs a lot of time, computer resource. So the idea is that I have seen, we have done that in many other properties. We found that these simple topological indices can develop good model for many different physicochemical, pharmacological, and toxicological properties. More recently, with our collaborators, we did study on blood-brain barrier. We took our 198 poly descriptor, model accuracy was 75%. We took the Romanian descriptor 579 Schrodinger plus Topocluse, model accuracy was similar. So basically, when you combine them, there is no difference. So the idea is that theta is not better. If we just add more descriptor, that doesn't make it better. Now, mutagenicity QSR also we did. I am not going to show that. We published this paper before. Now, molecular similarity. When QSR doesn't work, means you cannot predict a property. So we try to develop analog. That is what is molecular similarity. Now, similarity idea is similar structure usually have similar properties, but the point is, how do you define similarity? Similarity has a problem. One is subjectivity. You know, two people given two different entities, they can tell similar, dissimilar, and also similarity is non-transitive property. Means A is similar to B is similar to C, but then A and Z could be quite dissimilar. Here is an example. If we start with white, and then if we see that two words, they differ in one letter, we say white. We say similar. We come to white, while, well, shell, ultimately we say stork, stock, stack, slack, and black. So this is from Lewis Carroll. He showed that from white you can go to black. So basically, as we go further and further, white becomes similar to black. But we know in language that's not true, but in chemistry, how do we know? So another example, if we start with benzene and go on adding this kind of tail, this benzene and this one could be quite dissimilar. So we did some study with EPS database about 4,000 chemicals, and we found out that if we have a multi-dimensional space, here are the two-dimensional PC1, PC2 plot. And then if there's a new chemical that comes, if the chemical is here somewhere who has quite a few neighbors, then we can easily tell from the neighbor property what kind of property it would have. But if the chemical is far away, like hot ball, both dur mein hai, very far away, then it's very difficult to tell what is going on with the chemical. That chemical probably needs to be tested. Now we go to biodescriptor. After the Human Genome Project, the face of science has changed forever. Now descriptor from biological system, because often people knew that just chemodescriptor is not enough. We need to supplement chemical data with biological data. So what is biodescriptor? When I was a graduate student, I read this point. If your chromosomes are XYY and you are a naughty guy, your crimes the judge won't even try, because you have a legal reason why, he will raise his hands and gently sigh, I guess, but this you get a buy. That means if you are XY or XYY, one variable difference, that will be easy to say. But now we have this kind of thing. Proteomics data, one of my collaborator, Winsman, he got probably more than tens of millions of dollars grant from US Air Force NIH, and he gave us the data. So now if there are 2,000 proteins, 3,000 proteins, a few chemicals, how do we predict them? What do we do? So we have to be able to develop compact descriptor from this proteomics data. In our group, we developed different kind of approach: graph theoretical approach, inference theoretic approach, statistical approach. I don't have the time today to discuss those. But then, more recently, we applied to multi-world nanotoxicoproteomics or multi-world carbon nanotube PIO2. Dr. Brachko later in the day will talk about this in more detail. Also, we developed in collaboration with Dr. Nandi, Dr. Ashish Nandi, we developed some alignment-free approach. Means, there are dust methods, they align the sequences. But then you can also do, like in Kumbha, there is alignment-based, but there are alignment-free QSR methods. Similarly, we developed alignment-free sequence descriptor, and then we applied them in different areas. And then we recently we applied them to characterization of these various variants of COVID-19. Now, as you know, that COVID-19 started in 2019, 2021 was all mask, nothing else. But 21 in this year, we have now vaccines going on. But then we have now emerging Omicron. We don't know how much these vaccines will work out. So here is the work we did using a bunch of sequence-free, alignment-free sequence descriptors of 32 March sequence, 252 SARS sequence, 252 SARS, and then COVID sequence. We did self-organizing map. It was done by in collaboration with Dr. Brachko. In fact, Dr. Brachko is the one did the sum analysis. As you can see, the three different groups of viruses, they are well separated. So the idea is that if a new variant comes, new virus emerges in the world somewhere, we can look at the sequence and we can see where does it belong, But like in our country, they say when the boy is doing something wrong, the grandmother says, "Arey, ye ladka kahan se aata hai? Kisko saath milta hai? Who is his friend?" Similarly, here it is a virological ladka, We can ask that what is going on? Which neighborhood does this virus belong? From there, we can do a kind of surveillance. That work is going on. And now this is coming out in the journal Current Computer-Aided Drug Design. So in our research group, we have done this hierarchical QSR, first topological, then geometrical, then solvation, then relativistic QSR, and then biodescriptor. But if we can do at this level using topological modeling, then that is better. If we cannot do that success, then we have to go all the way using the biodescriptor, which are really, really costly. So this is what I said I will tell you at the beginning. First, optimal represent the molecular structure using graph theoretical, quantum chemical, whatever, calculation of descriptor, quantitative QSR development, quantitative molecular similarity. And then these models, if they are good, then they can be used as a decision support system for screening of chemicals and biologicals like viruses. So that is my talk. This is my house. I am talking by the side of this window. Sometimes things happen like this. A couple of days ago, it was 30 below Fahrenheit. Now it is better. But then when this happens, we have to clean the snow. But then once we clean it, we have got some time. We do some QSR also in mathematical model. So with that, I will end my talk. I will say, Ashesh Dhanyabad in Bengali. Bahut bahut dhanyabad. Thank you very much. And Charai Beti, we will continue our journey. Thank you very much.
Okay. Thank you very much, Dr. Basak sir. Dr. Basak sir has elaborated upon the history of physical property and graph theory. He described on how difficult to search a drug. He also talked about problems on data availability and elaborated upon the chemical structure evaluation strategies. He described QSAR and high QSAR modeling and blood-brain barrier entry model using computer descriptors. He discussed about molecular similarity concept with some example. He also touched upon different approaches for biodescriptors and applications of AFSDs of DNA and RNA sequences. So thank you very much, Dr. Basak sir, for your very good lecture that I am sure all participants will be benefited. Now I request participants to ask queries if you have any, please.
Is there any query from participant? Check also in online. One question from Professor A.K. Saxena, sir.
Yeah.
Please, sir, please come.
First of all, I think good night, good evening at your place,
Yeah.
Subhash.
Yeah.
It's
Sure.
it has been a very interesting talk and reminded me of all the things which you had been doing all along the time. My just question or you can say clarification is that are you be able to take a different kind of enantiomers into your these calculation of these parameters?
Yeah, we developed a paper on chirality characterization based on the CIP rule. We developed that. We published a paper on JK. But I did not discuss that here. But we already published a paper on that.
Okay. Okay.
Characterization of
thank you very much.
chirality using graph theoretical approach, starting with CIP.
Okay.
If you want, I can send you the paper.
Yeah, that would be nice.
I will send you the paper. Yeah. Already published.
Hello.
Yeah.
Sir, very nice talk, very scientific information for the young scholars. I have one small question to you.
Yeah.
Under this pandemic situation, actually there is no specific antiviral medicines to combat the COVID-19. Only drug repurposing are there. Small molecule specific anti-chemotherapeutics. But in different laboratories, different compounds are being tested at different experimental conditions. For example, against protease inhibitors of SARS-CoV-2.
Mm-hmm. Yeah.
Hello?
What is the question then?
Yeah. What happened? Echo is there. Now, my question is, for example, if I consider 80 compounds, excuse me, sir, just 1 minute. What happened? Hello?
Yeah.
Yeah, at different experimental conditions, different molecules are being tested. So these are SARS-CoV-2 protease inhibitors. So shall we take these molecules in QSA modeling by calculating this topological indices?
Point is we have to be pragmatic, because first point is you are taking 80 compounds, but then how many of those have good quality test data? First point is you have to have good quality data. Second, sufficient number of good quality data so that you can develop a model and then forecast from that. So point is under good condition, under good environment, laboratory condition, there should be good quality data. And then I suppose if there is good quality data and the descriptor is good, we should be able to have good model. So if the data is not good, we have to generate good data. If the model is not obtained by the descriptor, then you have to find some new descriptor. That means I could not describe the relevant part of the molecule that is responsible for the protease inhibition.
Okay, sir. Thank you very much, sir.
Am I clear?
Is there any other question from offline participants who are present here in the seminar hall?
Hello, there is a question on chat.
Okay.
If Chairman Sir can read it.
Yeah, just
Yeah.
please wait.
What is the question?
Hello, one question is
Yeah.
Professor VP Combo sir.
Mhm. Yeah.
VP Combo sir. VP Combo sir, please. You please ask directly.
Yes, sir. The question is that did you synthesize any compound and test in vivo?
You know what happened? We are theoreticians, but in the 1990s, our program Poly was implemented in Upjohn Company, which is now part of Pfizer. They had a 5,000-6,000 favorite chemical data set. Whatever they want to discover drugs, they used to do the high throughput screening. Using our data set, they clustered them and then selected the chemical, and they discovered quite a few lead compound. One of them was HIV reverse transcriptase inhibitor. If you send me an email, I can send you the letter and the paper where they published that.
Okay, thank you.
Okay.
Yeah, you're welcome. Good question. Good question.
Okay. Thank you very much, sir, for nice responses from both the sides. We'll begin with then next session.
Excuse me, sir.
Thank you very much.
Yeah.
Chairman sir, chairperson sir, excuse
Yes,
me.
sir.
Yeah, now I would request Honorable Chairman of the Institute, Professor Dr. A.K. Saxena, to offer a small memento to the Honorable Speaker of this session. Please, sir, please come.
So, Professor Bazak, for your very outstanding this lecture. This is a token, and I think it will be sent to you by mail. So please accept it. And thank you so
very
much.
Good, Supriya. Thank you very much.
Thank you.
I appreciate that. Thank you very much.
Shall we start with next session?
Please, please, sir.
Okay. Respected dignitaries, Professor P.V. Bharatam sir, and all dear participants, welcome you all once again for the second session of this second day of DDDR 2021. For this session, we have today with us Professor P.V. Bharatam sir. He is currently working as professor in Department of Medical Chemistry in Naipur, Mohali. His field of specialization includes medicinal chemistry, theoretical organic chemistry, organic synthesis, molecular modeling, and pharmacoinformatics. Professor Bharatam completed his PhD in 1990 from University of Hyderabad, India, in the field of applied theoretical chemistry. He has a total of 7 years of research experience, which includes 4 years in USA and Germany and 27 years of teaching experience. Apart from his current position, he also held various positions such as Associate Dean Academics during 2015-16 and Dean of Naipur, Sasnagar during 2016-18. He is recipient of various awards such as OPPI's Scientist Award in Medicinal Chemistry in 2009, Ranbaxy Research Award in Pharmaceutical Sciences in 2008, Chemical Research Society of India Medal in 2008, Fellowship of Royal Society of Chemistry London in 2007, IBM Faculty Award in 2007, and Fellowship of Alexander von Humboldt Stefan, born in 2002. Till now, he has supervised 36 PhD students, 173 MS Pharma students, 16 MSc Chemistry students, and 12 fellows. He has received 15 government-funded research projects, 6 institutional grants, and 2 industrial grants. Professor Bhartam has 258 original scientific articles, non-Indian journals, 19 original scientific articles in Indian journals, 23 reviews, peer-reviewed, and book chapters, and 15 science education articles to his name. So I take this opportunity to welcome Professor P.V. Bhartam sir and request him to enlighten us on 3D thinking in drug discovery. I request respected sir to follow time schedule so that all other ensuing sessions will be smoothly conducted as per schedule. I request all participants to ask your queries, if any, once sir ends his deliberation. Professor Bhartam sir, you may proceed with your presentation. Thank you.
Okay, can you hear me and can you see the presentation?
Yes, sir.
Okay, I would like to invite all of you to start thinking in 3 dimensions, because drug discovery scientists cannot remain in only 2 dimensions. Most often when you look at a pharmacology research article, they mention the name of the drug, but they don't even show the 2D structure or 3D structure. Go to pharmaceutics, it is the same situation. There are several pharmacy fields where they don't think in 3D, they only think in 1D. They only worry about the name of the molecule. The rest of the thing, they leave it. They don't think in 3D about the drug. But in computer-aided drug design and drug discovery, without thinking in 3D, we cannot work. This is not the message, my message. Message is given in this journal article. This is a perspective article which was published in 2018. A very well-known computer-aided scientist, Murko, he published this, but he was not talking about only computer-aided. He was talking about a medicinal chemist. The title of his research article is "What Makes a Great Medicinal Chemist?" He listed 10 points, 10 or 12 points. Out of that, he said that 3D thinking is one of the important characteristic features of a great medicinal chemist. If you want to succeed, we have to think in 3D. We have seen those senior professors who are attending this, they all know that those who think in 1D and 2D, they will succeed, but they are not great. They cannot become great. Only those people who think in 3D, in addition to doing many other things, they are becoming great. So thinking in 3D is very essential. So especially in drug design, when you go to the computer-aided drug design, we need to go to this certainly in 3D, because the drugs are 3D objects, they are not 2D objects. For our convenience, we have used 2D on paper and pen, and we write, we use 2D, but they are indeed 3D. 1D are the chemical molecular formula, or the chemical structural formula, or Inchi key, or the Smiles notation. They are not sufficient enough. This information has to be converted into 3D, and then we can proceed. Because once you have the molecule in 3D in front of you, you can play with it. You can zoom it. You can represent it in many, many ways. You can show the surface property. You can look at the internal characteristics using the quantum chemistry. So many, many ways, 3D is very, very essential. So for example, look at this molecule. We all know this molecule. Probably most of you know this is a simple compound called isoniazide. It was a magic bullet. It has done many important things for the TB patients. And earlier, before inventing this drug, TB patients were kept outside the city. But after inventing this drug, TB patients are living inside the society as normal human citizens. They don't have to be thrown out of the city. So that kind of magic effect this molecule had. But if I look at it in 2D, it is looking beautiful, pyridine ring and amide bond and very nice. But if I start thinking in 3D, I come to know much more details that there is a pyramidalization at the two nitrogen centers, and then there is conformational flexibility due to the sigma bonds and many other details we will be able to grasp if I look at it in 3D. So to do so, what to do? How to do that? If I want to become somewhat relevant in medicinal chemistry, if I want to become relevant in drug discovery, I need to think in 3D. But for that, what are the facilities available? Do I have facilities? Yes. The very most important facility is our brain. We are if we can tune our brain towards this, everything we look at it, we reimagine it in the form of 3D, then we can do very well. Because the 3D objects, the drugs are 3D, the macromolecules are 3D, the interactions are 3D, the dynamics is 3D, and everything and anything that is associated with drug discovery is 3 dimensions. For that, pharmacoinformatics is offering many tools. This is a topic, though a young topic, only in maybe starting in 2000, in 1993, the molecular modeling originating from crystallography and computer-aided efforts as applied to drug discovery became very popular since 1993. In 2003, pharmacoinformatics came into picture, and then both these fields are contributing significantly, especially in this time of pandemic. Computer-aided drug design is helping in many ways. You can work while sitting at your home using your laptop. You can still do computer-aided drug design, and you can remotely use your facilities in the laboratory. All these things are happening using this particular field. So I'm sure the contribution from this CADD field in future is going to extensively increase. Our Corona Bhai Sahab is going to help us in increasing the CADD efforts in drug discovery. Anyway, I'm going to tell you how to think in 3D. For that, we simply have to look at coordinates. Every atom in a molecule, every atom in the drug molecule are associated with X, Y, Z coordinates. How to find these coordinates? There are simple methods like energy minimization and first build the 3D structure in the computer screen and then perform energy minimization. Then you will come to the appropriate chemically relevant and the pharmaceutically appropriate and relevant 3D structure of every drug molecule you can easily find. And these are the coordinates of the isoniazide molecule. You can see that there are so many carbon atoms, nitrogen atom, then there are hydrogens, and then carbon, oxygen, all of them, each of them has got coordinates. This is one way of thinking, but it is very tedious. We don't want to look at coordinates and remember the coordinates of every drug, and these will change as a function of the rotation. So we cannot remember this. But then there is another facility called internal coordinates. This is about the bond length, bond angle, and torsional angle. It is difficult to remember this also. But then these 3D figures are possible. There are several software available today. Earlier, about 20, 30 years ago when I was doing PhD, that time these facilities never existed. Now these facilities became very handy, and they are very easy to use. They are very user-friendly software. Many of them are free also. You can download and use them. For example, PyMol is a free software which can be extensively utilized to understand the structure of macromolecule and the drug complexes. So there are many others. So as a part of it, you can see that molecular modeling can be done if and only if 3D structures are available. Now, estimating the properties of the chemical species can be done easily, and atomic level details of the chemicals can be obtained. Hydrogen bonding, van der Waals contacts, ionic interactions, bonding patterns, all can be visualized and appreciated, and then further utilized for further research using if we have the 3D structure. Let me give an example of metformin and metformin hydrochloride. Look at this 2D structure of metformin that is given on the in the blue color. The metformin is actually metformin is not a drug. Metformin hydrochloride is a drug. Metformin hydrochloride is different from metformin. If we think in 3 dimensions, we come to know, because in 3 dimensions, when I look at metformin is a flat molecule with intramolecular hydrogen bond. After the protonation, the protonated metformin or the metformin hydrochloride is not 2D object. It is certainly, it is not flat because protonation happens at the N1 position. And when protonation happens, it does not happen at N4 position. When protonation happens at N1 position, it is possible to consider as if it is shown in the pink colored structure. The intramolecular hydrogen bond is broken, and because of that reason, the molecule, it becomes flexible. CN rotation can happen, N inversion can happen, and as a result, you see what they are all happening in metformin hydrochloride. You can see, appreciate the difference between simple metformin and metformin hydrochloride. The differences are tremendous. The differences are as if a young boy looking for a job, before getting the job offer and after getting the job offer, he becomes quite dynamical. The moment he gets the job offer, he will start dancing and jumping, and he will party and all those things. But before getting the job offer, he is dull, dumb, and he is depressed. So that much quite difference is there between the protonated and non-protonated metformin. So protonated metformin is losing, there is no conjugation, loss of intramolecular hydrogen bond happens, loss of resonance stability happens, they reduced molecular rigidity. These are all happening. However, there is lots of gain. What are the gain? Presence of two lone pairs at the electrons at the N4 center, a high electron density at N4, proton affinity of the complex is very low, the N inversion barrier is low, CN rotational barrier is low, the molecule becomes highly dynamical. As a result, the molecule also shows polymorphism. Polymorphism, there is form A and form B of that metformin molecule, and you can see that thermodynamic form and the metastable form, the difference is only in the form of small conformational change. This energy difference between the two conformers is only of the order of 0.01 kilocalories per mole. However, in terms of solid state, they are different. When I am trying to prepare a tablet, whether I should use polymorph A or polymorph B, whether the drug action is being shown, how the drug is swimming through the organic cation transporter, and how it is entering into the cell, and after entering into the cell, where how it is interacting with the cytochromes, or how it is interacting with the AMPK. All these details can be understood only if I know the 3D structure. So just by thinking in 3D, we were able to realize that there is a tremendous difference between simple neutral metformin and metformin hydrochloride. Exactly like that, we can think of any drug molecule. Now, pharmacoinformatics is a topic which helps for us to think easily. You simply download either on your mobile or on your laptop, you download some software and you start building the 3D structures very easily. Today, it is so easy to do 3D thinking. We should shun away from thinking in 1D and 2D. Okay, 2D is useful, no doubt, but 2D versus 3D, the amount of information we gain is extensive information gain. All of them can be utilized in rational drug design, computer-aided drug design, molecular modeling, structure-based drug design. All these listed here are used as synonyms, and all the scientists working in these fields are indeed utilizing the 3D thinking. But the youngsters need to adopt it, cultivate it, because we are told 2D is good enough while writing on textbooks, but it is not true. Now, the technologies that are involved, QSAR, docking, virtual screening, you know all of them. Artificial intelligence is also one of them. It also requires information in 3D. Now, I am going to give an example how we utilize this information in our day-to-day research. First thing we do is observe. Observe the 3D structure of the enzyme carefully. Which are all the cavities? What is the size of each cavity? What are all the amino acids present in each cavity? Which are flexible, which are rigid, which are reactive, which are not reactive? We observe the 3D structure of the macromolecule. This is the first thing we do. After that, we do structure-based drug design, pharmacophore mapping, QSAR, dynamics, and quantum chemistry. And then we design. Once we design, we go to the organic lab. From the organic lab, we go to the in vitro and in vivo labs. This is our job. uh, and then when we think in 3D, we also get an opportunity to think on the surface. Every drug has got a surface property. Just like every human has got a smile on his face. If based on the smile, you can guess what is the mood of the person. Similarly, based on the surface of the drug molecule, you will be able to know whether it is effective or not effective, whether it is going to be useful or not useful. This is all the information. Look at this again, metformin molecule. Metformin, neutral metformin is represented with the symbol BG1. Look at the surface of BG1. A beautiful distribution of electrostatic potential in blue, red, and blue alternatively, and that is complementary to the bigonide surface of the is complementary to the micromolecular surface as shown in D in this figure. But on the other hand, look at the tautomer of the metformin. The metformin can have a tautomer which is generally wrongly, many books are wrongly representing bigonides with the wrong tautomeric state. That wrong tautomeric state is written here with BG5. Look at the surface property of BG5. It is not complementary to the macromolecule. The BG1 surface property is quite different, and BG5 surface property is completely different. These differences are telling us we should never consider BG5. Also, energy is telling BG5 is not stable at all. If BG5 structure is available 1 in a million, whereas the rest of all the molecules are available in BG1 state, because energy difference between BG1 and BG5 is about 9 kilocalories per mole. That is what we can get from the quantum chemical analysis. So what we got the information that the surface BG1 is suitable, and structurally this particular tautomer is more favorable, and that is what is showing the drug action, and BG5 tautomer can never show drug action, even if it is uh, available in one particular temperature. Even if BG5 is available, it is not going to show any drug action, because I know the 3D structure of the drug, and I also know the 3D structure of the surface of the drug molecule. Now look at BG3 is also not very suitable. Now on the other hand, protonated BG1, that is BGP1. look at the structure E, BGP1, the surface of the BGP1 is not much different from that of BG1. That means protonated bigonide is not tremendously undergoing any surface changes. They are minute changes. Tautomerism is bringing lots of changes, whereas surface, whereas the protonation, though it is breaking the conjugation, though it is increasing the dynamism, the surface property is not really changing. So that is still continues to be, and actually BGP1 is the only suitable structure. And look at BGA2. This is by chance, if you remove a proton from the neutral bigonide, then the molecule becomes negatively charged. Even in the negatively charged condition, the surface property is maintained. Under positively charged condition, the surface is maintained as the same as BG1, but the tautomers are not maintaining the similar structural features. As a result, we have got to we came to know the importance of structural features and surface features and their importance with reference to tautomerism. That has become clear. So having said that, we can look at the pharmacophoric features of the anti-malarial bigovannides. That is cyclogovanil progranil gets converted to cyclogovanil. But if I rewrite the structure of progranil in its right tautomeric state, I will come to know that the pharmacophoric features between progranil and cyclogovanil are very, very comparable. And thus, the hydrophobicity versus the hydrophilicity centers of progranil and cyclogovanil are not quite disturbed during the cyclization. This particular factor becomes clear after performing 3D thinking and tautomeric rewrite, re-representation, as well as after understanding the pharmacophoric features, surface properties. So this is another advantage. This much work we have done in 2005, and we have further utilized this information in 2014. So there are many other details. I don't want to give the details right now, but there are molecular orbital details. There is a pi orbital, there is a sigma type of orbital, p type of molecular orbital. All of these are very, very comparable after performing 3D structural analysis and after understanding the internal details of each and every molecule. Using this information, we wanted to design new molecules. That is where the guanyl thiourea derivatives have been found to be having the new the character that is required. Required pharmacophoric features are available in guanyl thioureas, and we decided to go ahead and synthesize many of these molecules. Synthesis is not very difficult. Start with a primary alcohol, treat with HBr, and then you will get a bromine derivative, and you condense with guanyl thiourea, and then you will get the series of compounds very quickly. Within 2 months, you can synthesize 20 compounds. This is how, and then we perform molecular docking. When we do docking at the appropriate site, it becomes clear that the PFDHFR is the target, and the newly designed compounds are appropriately fitting inside the cavity. You look at this 3D structure, it is very clearly explaining the entire chemistry. What is the chemistry happening inside the cavity of PFDHFR? It is nothing but the transfer of hydride ion. What is that? Hydride ion is available on NADPH. NADPH is getting converted to NADP plus. At that particular moment, H minus is getting transferred. What is PFDHFR is doing? It is bringing the dihydrofolate and the NADPH to close proximity. That means approximation is a mechanism that is happening inside the cavity of PFDHFR. That clarity comes once you look at it in 3D, because the hydride ion, which is ready to get transferred, is directly exposed to the positively charged center in dihydrofolate. That is a natural phenomenon that is happening, but I want to inhibit the same. If I want to inhibit the same, I have to identify what my newly designed inhibitor is doing. My newly designed inhibitor has got 2 lone pairs extra at a particular nitrogen center. That means there is excess electron density at the nitrogen center. This electron density is directly pointing towards the H minus of the NADPH. So what is happening? The NADPH is ready to supply H minus, and the 2 lone pairs at nitrogen are telling we are already having too much electron density. Already 2 lone pairs are at nitrogen, which is not rare, which is a rare event. In the design GTU derivatives, there is a nitrogen center which has got excess electron density at that point, and that nitrogen is exposed to the H minus. That means H minus is ready to be transferred, but this nitrogen is telling, no, don't send H minus, because H minus is having extra electron density. I am also having extra electron density. I do not accept the transfer of H minus. That means inhibition is initiated. That means the electrons are electron transfer in the name of H minus is prevented by the excess electron density on the inhibitor. So molecular docking, when I think in 3 dimensions, it also gives me an opportunity to think into the molecule, look inside the molecule, and where are the electrons and how the electrons are participating in the inhibitory process. So likewise, we need to pay attention, and then we will get clarity, atomic level clarity, as well as electronic level clarity we are going to get. After getting that clarity, we synthesized many compounds and we docked many of them and saw that whether the designed compounds are efficacious towards quadruple mutant DHFR, wild type DHFR, double mutant DHFR, and another variety of double mutant DHFR, all of them versus the human DHFR. What are the differences? We could understand because we thought in 3 dimensions. Okay, now these are superimposition of the molecules, and then where are the differences, where are the common factors. You can see that on the right hand side there are common factors, on the left hand side there are several differences. This information is also very useful. And then we went to molecular dynamics. Dynamics is telling clearly that the designed compounds are showing very small interaction with only 22 kilocalories per mole stability with human DHFR, whereas with the wild type PFDHFR, quadruple mutant PFDHFR, double mutant PHDHFR, as well as another double mutant PHDHFR, you see minus 22 versus minus 34. Minus 22 says that it is relatively less binding. And with the PADHFR, the binding affinity is very strong. That means the drug will be entering into human body, but it will directly go to parasite. It will
Sir,
not
kind
disturb the
request
human
to conclude the
PHFR.
session.
Okay. And likewise, we know which amino acid is responsible. And then after getting all these details, we went to in vitro. In vitro experiment is only to measure whether NADPH is utilized or not utilized. That is again confirming that NADPH is not utilized in the case of 16 compounds out of the total 23 compounds that were tested. Now, when we went to in vivo, in the in vivo testing, we utilized almost 100 animals. All of them were treated with Plasmodium berghei, and then they were treated with the designed compounds, and observations were made for the day we subjected the inhibitor, and then from that day, 4th day and 7th day, observations were made. No, all the days observations were made up to 7th day, and there was some improvement. 14th day observations, it clearly told that out of the 16 compounds, 6 compounds are efficacious. Observations were made up to 60 days. then it became clear that out of the 6 compounds, in the case of 5 compounds, recurrence has been observed. So they are not really useful, but out of those total 23, one compound can be declared as curative. This was the this was the final. That means that in that case of that compound, animals lived happily ever after. That means this compound cured the malaria from the animals, and that is an excellent news. If 3D thinking is helping in the design of one compound, useful compound, and out of synthesized 23 compounds, it is a tremendous savings of experimental organic chemists time and money, and biologists time and money. So thus we have been able to identify one curative molecule. And with that message, I would like to stop my lecture. Yeah, I would like to tell that 3D thinking helped us in identifying a curative compound at a faster speed. That's my message of this lecture. Thank you very much. Okay, can I stop
Okay,
here?
sir. Thank you very much, sir, for your very informative session. Unfortunately, because of the time restrictions, we could not spare more time for you to talk in much detail about the deliberation. Although it was very good, I hope you will share your presentation to the organizers. Okay, sir has highlighted on thinking in 3D drug discovery. He deliberated on how to think on 3D. He discussed on methods for energy minimization using isonucleotide example. He talked on complexity of coordinates. He also talked about advantages of molecular modeling and actually what we miss if we don't use 3D structures. And he explained that by giving example of metformin. He discussed a lot on importance of pharmacoinformatics in drug discovery. He elaborated upon various technologies that also includes artificial intelligence associated with the 3D activity. He also talked on use of molecular electrostatic potential analysis for BGI that shows a concentration of electron density at N4. He also deliberated on designing of new molecules and the approach to synthesize the same. In the line, he also talked about molecular docking and in vivo activity studies. So thank you very much, sir, for your nice presentation. I would like to invite questions from participants, if any. Yes, there is one question that we received in the chat. Is a 2D QSAR not at all useful for the identification of new molecule? This question from Smriti Sharma.
That is a interesting question, but the answer is no. Your question, 2D QSAR is also useful, 3D QSAR is also useful. Both classical QSAR, 2D QSAR, 1D QSAR, these are all excellent technologies. They are all, when there are so many technologies available, which technology you would like to use? You use the 2D QSAR, you design new molecules, and then you go ahead, and when you go towards further research, reconfirm and reaffirm your result with the help of 3D thinking and 3D QSAR. My emphasis is on 3D thinking, and my emphasis is not on 3D QSAR. So as a result, every technology that was introduced is useful. The way we want to use is the art. We need to develop the art of utilizing the technology, and then we will certainly get very good results in this effort. Okay, we can go to next lecture. Next question. As the drugs which have come out of molecular modeling are captopril as a C2 inhibitor and protease inhibitors for his inhibition as helped optimizing leads. But I would like to add that there are almost every drug that was introduced after the year 2000. Every new drug molecule that was introduced utilized the computer-aided methods at one stage or the other. It may be at the stage of design, it may be at the stage of drug delivery, it may be at the stage of drug metabolism, or whatever may be the stage. There are every drug that was newly introduced utilized the computer-aided drug design technologies, and certainly 3D thinking also. So it is not a captopril is a good example, but there are many examples. Almost all the anti-HIV drugs are designed using computer-aided methods.
No, they have been optimized using computer-aided designs, but not generated any new leads.
Well, the computer-aided methodology is a technology. We can use just like NMR. We cannot ask the question, how many drugs came out of NMR? Similarly, we cannot ask the question, how many drugs came out of CADD?
No, because CAD was because you have a 3D modeling of the molecule as also of the receptor. So once all are matching, so why it should not be for drug lead generations?
I showed the example. I showed our example, which we have been able to find.
Not yet reached anywhere. If 100 molecules enter the clinical trials, only one becomes a drug. So your molecule has not yet reached clinical trials or even in the toxicity studies. That way, hundreds of molecules have come through the CAD, but what have ultimately become drugs? That is the main question. The example of metmorphin you gave, metmorphin hydrochloride was introduced way back in 1995. And that was the chemist's ingenuity in generating a molecule and then making it water soluble. That is why it was made as hydrochloride, not used as metmorphin.
No, but all the drugs introduced after 2000 have utilized computer-aided drug design. They all came out of computer-aided drug design. All the drugs came out of computer-aided drug design only.
No, that is a general statement. What I tell is lead generation, computer-aided drug design help you to optimize the activity in that particular series, but not generated the leads which are expected. Because if you look at all the drugs which have come from 1981 to 2020, only 25% have come from by synthesis, 50% have come through the plant natural sources, optimizing them, are using their pharmacophores and developing new drugs, and 20% have come are biologicals. So if computer-aided drug design was so efficient for generating new leads and developing new drugs, why only 25% has come in the last 40 years?
That is a wrong perspective. I don't agree with
that.
Not
wrong perspective.
I completely object for that particular perspective. I believe that every drug utilized computer-aided drug design. Without that, the last 20 years, no drug would have come to market.
I think.
No, I gave you a classical example.
I request participant as well as resource person not to argue. Don't take it to personal level. Please move on to next question. Is there any question? Else, I will begin with the third session. Okay, organizers, can I begin with the next session?
Time is very important.
There is no question from our audiences.
Okay, so we'll move on to next session.
Okay, sir. Just a minute, sir. Sir, just a minute. Now, I would like to invite the Professor Dr. Anil Kumar Saxena, sir, to present the memento to the Prashad Bhartam, sir.
Bhartan, you have given an excellent talk, and I think the discussions will go on. Maybe my talk afterwards may clarify some things, and I think we can always discuss the open question. And I'm very thankful to you for your lecture, and it was really very kind of you that you had accepted the invitation by this symposium. And this is a colleague of honor for your lecture. Please accept it, and it will be sent to you by mail. Thank you.
Okay, thank you, sir. Nice talking to you after a gap of 1 year or so.
Okay, let's begin with the third session. Respected
Yes, sir.
dignitaries.
Yes, sir, we can continue, sir.
Professor Dr. Anil Kumar Saxena, sir, and participants, welcome you all for this third session of second day of DDDR 2021. Today for this session, we have with us Professor Saxena, sir. Dr. Anil Kumar Saxena is an emeritus scientist, ex-chief scientist and head of the medicinal and process chemistry division CDRI Lucknow, and is actively involved in the domain of medicinal chemistry and computer-aided drug design and drug development research. During his 50 years of research experience, he has published more than 215 research papers, 24 review articles in books and monographs, and has 70 patents to his credit. He has delivered more than 190 invited lectures and chaired more than 50 sessions, and has made almost 57 visits abroad. He has supervised more than 200 postgraduates for their research projects and 45 students for their PhD programs. He is the recipient of several awards. Some of major awards includes Alexander von Humboldt Fellowship, Insayang Scientist Medal, Chemist Chemicals UDCT Diamond Jubilee Distinguished Fellowship, Ranbaxy Research Award in Pharmaceutical Sciences, and honorary medal for outstanding contributions to medicinal chemistry and international scientific collaborations, Scientific Partnership Foundation, Moscow, Russia in 2004, and the Professor P.K. Bose Memorial Award, Indian Chemical Society in 2009. He is a fellow of Royal Society of Chemistry, UK, and is also a series editor for books, book series, Topics in Medicinal Chemistry, published by Springer Verlag. He is the editorial member of several prominent journals like Medicinal Chemistry Research, SAR and QSAR in Environmental Research, Online International Journal, ARKIVOC, and Patent Evaluator in Current Drugs, UK. He is also a member of several committees, including American Chemical Society. So with this brief introduction, I take this opportunity to welcome Honorable Dr. A.K. Saxena, sir, and I request him to enlighten us on the classical to modern drug design perspective. I request all participants to ask your queries, if any, once Professor Saxena, sir, ends his deliberation. Dr. Saxena, sir, you may please proceed with your presentation. Thank you.
Thank you very much for the nice introduction. And I am also very thankful to the organizers, particularly this institute, JIPA, who has, this echo is coming, so I can speak. Offline mode. So I'm very thankful to you, and there's a lot of students and the faculty which is present, who are present here, for organizing this symposium, particularly to commemorate my 70th birthday. And it was for 1 day. Previously, it was told, but then it has extended for 2 days because of the interest of my friends from all over the world, I should say. I think just before my lectures, I think there was some discussion going on. Maybe I might add something or maybe clarify, maybe some clarifications. This whole lecture is almost taking as classical to modern drug discovery, drug design and development part. So the thing is that I am covering a very wide, almost about 40, 50 years and in a very fast way. And it's like this that I am taking you on an airplane and showing you the things. Those who are interested, they will find the references. They can go through them in details. So I will not go very much into detail, but basic concept certainly I will clarify. You know, I think it has been shown by slides. By my friends also, Dr. Subhash, by Nikhil. It's very difficult, you know, you can't reach this kind of chemicals which are possible, very possible. Even if some scientists are working for 1 second per second, and so many chemists they do, they will take so many years to complete the whole chemical space. So if somebody wants to explore it, I think what are the approaches? That's why I come, and that's why the whole right from the beginning, the science evolution of drug discovery is right from the beginning. You know, it's basically a process which goes all around random screening, rational design, random. But you know, there's a difference. The difference is normally it is not circular, but it is a kind of a helical form. Each time we go deeper into it. So you should see previously random screening, then come receptor design, and this then comes combinatorial chemistry, STS, informatics. But the cycle will go on. The random screening will go on. The thing is how the random screening goes just by synthesis of the chemicals, or maybe just the screening of the millions of molecules and then getting it. So screening will always be there, no doubt, whether it is through real screening or through computational screening, which I will never put it. So I think this just shows how much difficult it is and how much time it used to take and how much little reduction we have done for the getting the leads as well as this thing. Here I would like to focus that the medicinal chemistry or the chemistry takes a very important role in the whole of this process. What has been in the past, pre-genomics and post-genomics, the lead generation from random plant origin, random screening, biochemical screening or this, they used to come, and then the optimization takes by the study of analogs, synthesis of analogs, comparison of their activities, then comes the targets used to come. But here in the pre-genomics, people have started taking off molecular targets because the targets are getting known through this proteomics and proteomics. Then comes again lead generation, part very important of the composers, asking how the leads have not come on leads through computated. I think it will be clear in my talk how they come and how they are used. But the lead optimization parts has been always useful. This used to be SAR, simply structure activity. What is structure activity relationship? And then improved by QSARs and then CAT, comparison between standard. Further part of the drug development process is of course very important, where toxicity studies, where also these things help. Then phase 1, phase 2, phase 3 clinical trials till the approval of the marketing. So the whole part of that design actually focuses in this part basically. So the molecule is selected for the candidate drug for development. Now I will tell you what has been the classical part, because I had been the one, I think joined in '69 and was reading at that time. That's the one important thing for a classical one is a pattern recognition. Some structural approach or hybridization. I think this has been the approach which was at that time, particularly my mentor, Dr. Nityanand, they had a very good pattern recognition. A chemist has to have a good pattern recognition. And what is a pattern recognition? You know, our human mind, the computer which is inside in our brain, which is far, far supercomputer. I have attended conferences in India, abroad, have met them and discussed what a computer takes several times when two persons are standing before you, and then he comes again, you recognize this. Even in the house, when you sitting, you know which is the rebel, it is a maid servant or is a press wala or it is a vegetable one. If you have usually seen that, you know your father, young students who are here, your father has come, who is talking the code? What is, this is pattern recognition. Only thing is, when the development came, this pattern recognition has been done even through computers also, on top of that. If the three statues of Ganesha is there, you can find out which one is example to other. But the person who was there, I'm forgetting his name at this time, he was sitting with me in France, and he says, Dr. Saxena, which I'm showing the slide, you have recognized this within some seconds. But you know, I have to take 5 days for computer time to do it. How that comes is how the person designs the molecule, which also can, and like Dr. Nishan used to do. You know, you identify from the pattern reduction of these parts, which are the molecules. So chemist sees a structure, the structure directly goes into his mind. He doesn't think it is a 1, 2, 3, 4 substituted, not like that. He simply sees a structure, goes into it. And then he grabs these pharmacophores, assimilate, and then sometimes he conformational restriction, all of it, and then finally he designs the molecules. Now it used to be qualitative. When we say structure activity relationship, qualitatively, quali means you say that if this substitution is there, this will increase the activity, all this will decrease the activity. But the only thing is, with this substructure, you can quantitatively see that. Suppose you have seen 100 structures. Out of them, you found that this substructure is presenting 50 compounds. The activity frequency, now you give a mathematical bond into it, 50 upon 100, so it becomes 0.5. So just like this, you know which substructure is having high substructure activity frequency. Then you can calculate all those substructures into it. You can know what is the mean substructure activity frequency or not. So it is simply a kind which is classical known. Sometimes even a person can do far better than the computer one. But basically, this is logic, this is rational, which is being rationalized through these techniques. Now, this is a kind of a medicinal chemical hybridization approach, which is being coined for. And what you find, if you want to put two activities together, they can also be done. You see the example provided in vascular. This is broken bond, and you see the solid structures, which are important to make this molecule, which has more properties. And when I'm talking, I'm a person who is a practical chemist, who has been an experimentalist, not just theoretician. So I use this kind of approach, and also I will show the examples. Then comes the computer-aided design, which is direct design. Of course, direct design is a design, and you know the structure of the target. So which you can get X-ray and arm and all this. Otherwise, then you don't know the structure of the target where the molecule has to interact. It is just like this, that you have not seen any data. So you can't say pinpointedly, but somebody has been to Manila, that is the molecules. They are giving information. So you can visualize about Manila through the information which you are getting from the persons who have visited that place. Similarly, so I mean that this is a kind of indirect design. And where the SR, unique SR, SRs work. Now, the structure function relationship, the molecular structure is related to biological activity, which is related to the. You can represent this structure in 0 to 3 time. Then you have to have a parameter. You have to listen to Dr. Subhash Basa. So I have been there in his lab also, and I think I know him like in 70s. He was also working there for these topological parameters or graphical parameters. They simply want how the molecular structure can be formulated. And those were at one time there used to be very similar, less number of variables. It was very difficult even to choose them, physiochemical parameters. But now that in one point you can get 2,000 parameters, 4,000, you have to select out of them to get the model. And then you can use these models for predicting the activity of the analysis. Now, if I go to physico-classical QSRs, then it is that the basic structure of the substrate enzyme reacts with the enzyme or structure to make it complex, which a lot of things take place inside which you don't know. Finally, you have to find the response. But we know that this is important for this to this. Now that can be formulated in terms of biological response into three major things. How a molecule interacts. Either it goes into the body, apart from pharmacodynamic part, but when it goes and binds with pharmacodynamically, the electronic and static effect, and I hope it is important, they can be put in terms of free energies, and then this has been a physicochemical effect. At the same time, in 1964 or so, that was Corbin Hans, and I had a very good chance to meet him, to discuss with him. The other one is Stevenson, whom I have not met, but these fellows, they said, stay with the contribution of a structure. Add these contributions together with some kind of relation constant. You can get the combined approach which I introduced in this I have published this work, this video also. And then you can combine both, and these parameters you can take anything about this. So, in a nutshell, the thing is, the basic method, you have to identify some structure, biotic conformation, pharmacophore, and then ligand receptor docking or QSR, mix the binder, and this is the whole process which as a semi approach is one say in progress. And you know what is the continuous nowadays people are using a kind of one streaming paradigm. This is also streaming. But the difference is here you are not making sending the molecule to biological system. But you are screening, and the screening is you have a chemical drugs commercially now databases. You have the models. You can put millions of molecules into this, and then they will stay now. But nowadays, many times it was found that the compound is effective in vitro but not effective in vivo. So we put these aggregate parameters into it. It's a primary filters. Then you can got molecule, you can have a similarity search basis, you can get this filter. Once you come to this level, you can have them use 3D pharmacophore or design and so on and so forth. Molecules to get some activity prediction, QSR models. Then finally, if you know that the structure of the target is known, you can docking to validate. In this way, you can reduce the number of compounds. The synthesis for synthesizing. Without synthesis, I think things cannot go. But I think what you can do is, instead of just thinking beforehand, you can previously people think this maybe I think this should be it. But then you can have this hypothesis checked with these models. Now I come to the practical one. When I joined CDRI, at that time, earlier than this, almost in the 1960s, they were working on making the molecules rigid. Ethylene diamine is a very important cell structure, and which is in a lot of hormones, neurotransmitters, and so forth. If you make it rigid, you become piperazine. You see a lot of piperazines. Piperazine you put activity with the allied piperazines. And this has very important part, phenyl, and that's why phenyl piperazines or allied piperazine is important, because if you take phenyl piperazines and dopamine, even so many neurotransmitters are there, adrenaline, this is important. In this way, when I joined CPR, one group was working, which was on alliloxypropanolamines and all this. We had lots of activities and paralytic and we could be resolved through simple SR. No QSR. We want antihypertensive and neuroleptic as a dynamic. Not only these molecules, they were of course high activity too. This was this has been completed for further 74. This was to this approach. I'm not going to detail. This was simply the SAR, structure activity relationship. You are knowing what substitutions making the molecule as a CNS active and what are the substitutions making it hypotensive. I told you about medicinal chemical hybridization. In medicinal chemical hybridization, you see, this is a semaglutide molecule, which was also synthesized by us. And yesterday, I think my message, Dr. Shriman, which is now in the market, but it's for different use. So this was a substructure which I identified with arrived depressions for hypertension. So this, and
We had also one working, it was working, and this was the molecule, right? This is a molecule. So why not make this I think beforehand? And I think Dr. Kamboj is here. I'm so happy. Then because the question is at now, I think when I used to give lectures in CDR in those times in the 70s, everybody was like very junior. You were asking, can you? So this was the structure. I think Dr. Dhawan at that time, I put in the envelope. I said, this is the structure not made here. I will make it. I think it should show activity. I think this I presented in a project meeting of CDS. And this molecule, 86204, that showed activity compared to centroquine in some respect was better than that. Now, this was the compound 86204, it was 1986. The number was this, and this is the global, this is the whole animal activity. Normally, one can't take guarantee what will happen in the animal. One can take guarantee something, or not guarantee, I should say, one can say, yeah, of course, it should be that much of the vitro model. But since I have used this approach, which takes into account that or two, this is the activity between of this molecule and this is your dose wise. And not only this, even the mechanism of action were say they are both centrally active compounds. And when Dr. Shival was at that time testing, he was amazed. He says compound were and not only this on the receptor binding, these were actually on a receptor at 5-ST2, which is important for anti-hypertensive action, which was 31 for this, and nanomolar of 21, 30 nanomolars. Now another one. Now this was totally classical SAR pattern recognition, and I am not the person who can comment that this is my design molecules. I was a part to it. It was my mentor, Dr. Lieutenant, who spoke to me. And then this is a, when I joined CDRI on November 17th, it was perhaps 22nd or 23rd of November. He told me not to do this whole one thing, but since this was the more important economic compound CDS 189, 183, which was NOS compound, which was also showing hypotensive as well as tranquilizing activity. So we planned this compound, and he told if you can make this new prototype, this heterocyclic was not known in the literature by that time. It's known by us. And I think yesterday it is massive PS code. Pyrosinophilidoid, octahydropyrosinophilidoid compounds. So this molecule was started. And I tell you to the students and to others, and if you are very true, he told me, Anil, it is the plan for 1969. So he wanted I should make it by end of 1969. That means I had only 1 month and 1 week, which was a new prototype at all. Since I was very young, so I got challenged, and I was for it day and night. The NMR and IR of this compound, IR was taken on 31st of December 1969, around 11:30 in the night. So next day when I came, I told him it is great, and I think it seems to be correct. Oh, he said he had virtually forgotten one. He never meant that I should take it so seriously that don't enjoy the new year at all and work and hard. And he forgave me. And finally, this one is the drug, which was approved for marketing from CDRI in 1986 by the Prime Minister at that time, Rajiv Gandhi. And this is, but later on, we have a lot of patent for it. Now, later on, this molecule, I showed how this is active through TSR. And I could find out what is the important indicator variable, substructure. Now, again, to prove it, what I did, this was a substructure, this was important. So I wanted to have a limitation. I told you substructure activity frequency. If in that particular molecule, that substructure repeats and the complete, they actually more active. I thought if this is a substructure, in this molecule, it repeats 3 times. They are more than scientific. I think this was the 1.286, like 373. And if you compare the activity of the two, I will not go into detail, this compound was better than scientific. It vanilla steps. And it was very much comparable to respiratory. And so far the part will come, why the drug has not come to the market, I think it can be hour-long discussions which can be put to it. But and I will be very happy if somebody discusses at any time. But the thing which I'm going to tell you, how does the air helps? No, this molecule was fully developed, but we have lots of molecules synthesized around. This is always done by medical science. There were two activities. One was antihistaminic neuroleptic, and they found that if you replace this part by NSCO, they become appropriate antihistamines. And I think these are different other things. So what I wanted, as because publication cannot take place normally if you are reporting in a good journal the whole animal data. You need to have target. At that time, in our institute in CDRI, Dr. Patnaik was testing antihistamines in the previous thesis also. And I was using this thing for my work for QSR's development. These are the issues I will not go into detail. The thing is that the hydrophobic was important, and that some structures were important, and it was possible to dissociate these activities. I will not go into detail. There were three molecules, ethambutol, and one was more, this estamindole, which was named like this, and they showed very much good activity than mepiramine, which was a standard drug at that time for that. But one important thing which I want, and I have been working, in QSCRs, many times those compounds are not fitting. You put them out saying they are outliers. Now, these are the compounds which are not fitting into my prediction, and they have been outliers. And the two most important compounds, this was the actual activity, and this was the predicted activity. There's a far more difference. And the important point for a drug design, for a person, any person in any branch of science, is your inquisitiveness. Why they are not? What's the reason? So, right from that time in 1980s, till 2013, I was searching the answer for these outliers. And what I developed from there at that time itself, that was that when I was doing this work, I found a paper by Colvin Hunch on antihistamine diphenhydramine, which is also in Avil, it used to be diphenhydramine and phenethylamine in all molecules. And this was a similarity. How did I know this? Can any chemist normally see this structure versus the structure that one had shown before have a similarity? Nothing. But if you think that this is structure which is in the third line and that is structure which is in dotted line in nitrogen from aromatic, they can this means it has a form of thinking. So when I was discussing with CDRI at that time, even chemistry nobody normally could do it. And my very senior colleague, Dr. Jain, who unfortunately lost cancer very early, he told me, I think, why don't you take a result molecules? That means you you drop it here or here. We made these two kind of molecules synthesized. And finally, we found this molecule was comparable. Rest molecules were not actually that much. So it means diphenhydramine, when it is going into the body, it is taking this kind of a conformation and it's fitting there for the receptor. And this is a comparable activity which was diphenhydramine, this one. And then one of the isomers which is shown, and this one, while the other isomers are with a planar version, we find less. So what I thought was, this is the assumption which was made by you. The ligand receptor interactions may be viewed as the association of its component substructures with the complementary subside about the receptor, and the prototype producing certain response may not necessarily have all the requisite substructure elements. Still, those common substructures in different prototypes which associate for same subside could cause similar. I think better for the students. What is this? Suppose this is a room. We all are different, but we all have a substructure area. If I put a heater here, which is initially less heat and more heat and more heat, any one of you, when you will come, you will experience a same rate of change. You will say,
lesson. But its real terms may not be told. So this is the substructure. It will experience the same. So I wanted to prove it experimentally, particularly for history, this substructure. So what we did, as you see in our receptor picture, you see, this is the size, A side, B side, this side. So if you have something to interact here, they should be active and distance. So I put why is NS slow and slower? It does not make a difference in the distance. So why not put this in this? Similarly, CS2, CS2Y, you can have why we have this thing, you need aromatic part, you can have this one only. Then why you have this, you can have this, or you can have this one. So we synthesize these molecules and compare the rate of change of any stepping activity. Then you see the slope, because since I'm keeping error elements cost, I don't need another 0.3, 0.4, the dependence of the substitution parameter are almost similar in all these molecules. Of course, the main activity as a whole was different. Why? As I told you, a person who is the one who is making a very, I think his sensors are better. You can say, yeah, this must change, the heat in this side. But basically, the thing is that the changes are there. So this one it could be given a formulation equation, and this was by 2D QS. I think somebody asked a question, 2D cannot solve, 3D can solve. So this was simply 2D. And I will tell you, I think this was a discussion which I had with Corbin Hunch at one time in US. He asked me, what is the thing that people are showing so much 3D is coming, because Hunch was the person who originated. I don't think there is much difference between 3D and 2D. I said, yeah, I have an example, but of course 3D adds to it, particularly when it comes and then Schumer's and others. So then the computer came. This was in 87 or so, and I was the first one to bring computer-aided drug design, this Microvex computer with free of cost software to CDRI, and we used these. And what happened was, when I was at a conference in 1984, they said a lot of these big guys who were Marshall, etc., who was the generators of this form of Consia, he said, you have an ideal problem. There was one person, Dwiko, who was developing Hassel approach. And I would like to clarify also what is the three-dimensional. Now, this is the structure. If you want in 3 dimension or from 3D to predict it, how do you? You have a room. Suppose this is the reason that I'm standing. You can make this in 3 dimensions, put the thick structure down, make its coordinates in terms of a tree, and which are the points which you want to superimpose, and you can identify the activity. Suppose there is a mesh. You can have different mesh, different wires. Maybe the distance may be different. You are touching each point which is occupied. Suppose there is a 10 activity, you can say 10 points, 1, 1, 1, 1 equal. Now you put next structure, and suppose you occupy only 5, you can say it should have 5 plus or minus, and not going to you can make a puzzle into that, and the superimposition which you consider from. These are the element calculus which you use in this hazard to develop. Yeah, I think because of Pershora and Baldwin, we developed the hazard for this, and later on, for this substructure identified, because in the hazard, this was a greater part. So it means this out of this molecule, this is really the substructure, not this part. And because orientation we compare out of it. Now came the computer-aided software started coming, and we had a text 3D, and I think it was very thankful Dr. Composes here. He was the one who, at that time, we got some grant for CDI. We purchased the software, the first one from Python, and this was the text 3D one. And I used this thing, this my same molecules, and this was the structure.
which predicted in terms of equation the same thing. What I would like to show that what I've shown there, the computer was also said that there is a 3 sites BS1, BS2, BS3, which are important, and less are sub-sites which modulate the activity. Increase or decrease which was my equation. And we could predict. This is one molecule shown BS1, BS2. And I will show you the same thing what we have taken from their the sites, they were important. The 3 sites were important. And then later on I found, what should I do for this one? It was in 1999. Some paper was published on NTD studies. These were the molecules. By that time, I think that we had gone much ahead in computer. I wanted to test my model. And how can you test? Either you design, so these are the published data. I put these model compounds into my model and predicted the activity what I had given them, because we had the IC50 data. Only thing is we had an option that they can be correlated, so we put it. Only one was outlier, and if you remove, it was a very high population. But that process was not bad. It means that this is the thing advantage. This is the advantage. It means when it can predict, this is the prediction. This is not my molecule, right? It was inside the molecule. These are the published. So this is the way how these models help. Now let me come. This was one software, other software which is now with Discovery Studio Catalyst. And I will not do it here. Again, we did the same thing for the same series. And you know we got the same 2 hydrophobic sites, one anionic site and positivity. So this is again the same model which we had derived even very, very early in 1980s. Again the same thing. Hydrophobic, hydrophobic, polarizable. Now, still, the answer to my outliers was not clear. And by that time, I think modeling came, and I immediately put the student to the homology modeling for this. We homology modeled it, but the homology model also could not solve. I will not go into detail later on the real and histamine H1 receptor, for which in 1970s, late 70s, we didn't know even the amino acid sequencing properly. Whole crystal structure was there, and we could, of course, this is the homology model structures, and this is the difference between the homology model and the reality. This was the distance between the task membrane, and this was elongated. So this was the real thing which later on solved this problem of outlier. I will not go there to very much how they are finding, but I will show you that this was proximity to the crystal structure. Because this has a structure. Aromatics, rings, bifusical group, and this is the which finds, which we thought even at that time that yes, they are the sites which can do that. Of course, again from pharmacophore we can do that, but I'm not going to show you. These 2 outliers, what they were doing this thing at the active site when they go ahead. Fast. These are the ones which fits. But what has happened that since at the para substitution, these molecules have, because this turns into this side. CO-200 is the outlier. If we put into this, yes, it's just not hold, so it bends and this binds is an extra binding site, which is increasing the activity and what was predicted, because predicted activity was taken into this. So this was the explanation in last, perhaps it took me about 30 years, approximately 25 years to find out the answer why these 2 molecules or 4 molecules were more active and what they were doing. Now this was one example I have elaborated. I will show you how in the recent, not recent, I say, we were working on Alzheimer's. We were designing ACH inhibitors because it was very important concentration. You want ACH inhibitors for dementia molecules. And these are the molecules which were the clinical trials, clinical already used. And so what it did, this is now comes the designing lead structure. Those who think that the computer aided cannot give new leads, can optimize, certainly optimize. But you can generate new leads by putting this classical concept of pattern recognition at some structure. We found out that this was a modeling set with a known compounds. We could find out the pharmacophore and then we put in document scoring these molecules. So the virtual library was there. We identified this, and again, very, very important. Those who have to work for synthesis as well as design, they have to think what synthetic capabilities there. I cannot synthesize any molecule. So one has to choose. So we need a focused design on these molecules. And you will see that these two molecules which we designed on this basis have shown very good activity. These are the numbers. We had all created all this, and this was very good. And a lot of this thing. This is published in Journal of Medicinal Chemistry also. This was a new leads far better than revised technique. These are, and compared with scope, not only that, this is the enzyme data, but also in the passive avoided test, which was taken and compared to this compounds were very good. We had further more compounds. But then this is, I will not show, we are going into detail very much for these because we had done all the time talking, etc. studies to substantiate our results. And these are the things, lot of ACAT, ICT observed in vivo process there in vitro data. And similarly, and somewhere in 2011 or so, 2010, I was interested from the institute about tuberculosis, which I have never worked as a coordinator. So I thought that I will be able to control the chemist and the other ones. If I don't do anything, simply just say by my words, this is correct or not correct, I must initiate my work. And I started working on this. And this was a nitazoxanide, which was a kind of a repurposing or drug showing activity. And from this, I took lead. And again, using the pattern recognition and these studies, we got these molecules, which we put. And I think, again, very shortly, I was in Indo-US thing in tuberculosis. I went for the first time from CDI. And I think when I presented these molecules, they jumped over it. And immediately, I had to find the patents for these molecules because they were very active. Then yesterday, Dr. BSC was remembering. He told me, Anil, you are working, you have gone to I need something for LiftY. And he told me, I won't go in detail because it takes LiftY is responsible for collection of story and text. So that it is very good for latent TB. And I didn't synthesize rather. I had the molecules when we already, and then we got it. And the molecule which I gave to him was the one which was first one on LiftY in the whole world. Doing very good at it. And again, I think we have the same thing shown. Then also another, because beta-quinine was going to be introduced, so we worked for beta-quinine, and isomer situation. If you have isomers, that's a different problem for chemists. I want simple molecules which can be synthesized very fastly, and we designed the molecules that other molecules were active. And later on, I was asking the question from Subhash, how can you take enantiomers into it? We have published recently a paper 2011. How this was the one initially, this we did the data. And these are the molecules which we synthesize very simply. And these show very high activity. And I think in vivo as well. And these are some of the data for these. And these are the compounds important, very well fit into this ATPase synthase model. And show these activities. This is the one which I am showing how you can take into account the parameters, but how you can resolve the problem because the activity is particularly for resin molecules. I have taken so they were moved. I think those who are interested, you can see this paper in 2020 they are published. I think it's a remarkable paper, particularly when how in 2D, 3D or other QSR you can use the racemic data, but for taking consideration, because always when you do computer aided on computer design, you always get one enantiomers. You cannot get mixture because the structure is of one. So it can be 8 compounds, 8 mixture of 8 enantiomers, or 2 enantiomers, 4, and depending on the asymmetric center. So this is how I have told method, how you can make that. And then of course you have to justify the physicochemical significance of these parameters. Little bit of repurposing because some of the COVID work, repurposing, very well. The students yesterday, I think Professor Vladimir talked about it. If you know the structure, the molecules are already in the used as drugs. If you want to find out a new drug, it's very, very difficult, as I told you. But you can utilize these drugs for their activity through computer-aided design in the sense that you can test them, and you will find that the repurposing gives you it was a viral activity for cancer. So these are the key ones which have come with the activity which was their new use. And I think the same kind of thing in CDR and Tipper also, we did something for this corona. At that time, Shishin Nandi asked me, I told you how to do, you can do only proximity protease. And he did that. And the question was how to prove. And the paper returned with the comment, you prove it either by synthesis, but then to test it. But we were lucky that we started research on 2020 at the same time this paper appeared, and we related the activity of these molecules, which are the real activity observed in this published paper, with one which is very well correlated. So again showed that the model which we are showing is showing. So in conclusion, it's evident that the chemistry plays a key role in the process of drug design. The classical approaches involved the identification of substructures through human pattern recognition of the active molecules, the incorporation of the structures and the structure-activity relationship in the so-designed molecules comprising a series of structurally modulated prototypes. The gradual development took place from qualitative SAR to quantitative SAR. It was followed by computer-aided design involving both linear and structure-based design, led to overcome the issues having potential to overcome the limitations of classical SAR in terms of taking molecules of same conjugates. Because in those times, we have to take the molecules of the same prototype, same conjugate space. You can't go diverse molecules, but you can go through this now, which are now used in virtual space. The application of these approaches has been exemplified in our own work. The identification of novel molecules as protein anti-hypertensive, zero-lactic, anti-histamines, anti-Alzheimer's, and reposition of existing drugs in the case of SARS-CoV-2. I think with these, I would like to thank you very much. I would like to thank the audience here offline. I think I'm very happy to see so many students here, and also online who must be watching this lecture. And certainly I've gone a little faster, but all those who are interested, I'm always open all the time, 24 hours, I could say. You can call me, you can discuss, and I should be very happy to have the answers for this. But of course, here if you want, I can also reply, answer the questions. Thank you very much for all of you. Thank you very much for your attention.
Okay, thank you very much, sir, for your valuable and informative presentation. Although it gone beyond scheduled time, it was really a great opportunity and pleasure to listen to you, sir. Dr. Saxena, sir, discussed about a lead prototype and its design. He deliberated on direct and indirect design in CAD. He discussed about virtual screening, especially a ligand-based approach. Under classical 2D QSAR, he elaborated about Hunt's analysis, pre-Wilson analysis, and combined approach. He also described in silico approaches in drug research. He talked a lot about virtual screening as a multi-layer hybrid prediction protocol. He explained about aryl piperazine, an important pharmacophore in screening. He talked about design strategy using structure activity analysis and CAM for antihypertensive agents and many other drugs. Sir, thank you very much for answering some of the queries raised in earlier session in most simple way. So thank you very much for your nice presentation. I request participants to ask any queries? If you have, I will check in the chat. Okay. There is no. I think there is no question in the chat box. If anybody wish to ask question, can directly ask. Am I audible?
You are audible.
Yeah. So if anyone is having any question, please ask to Saxena sir.
Very good self-explanatory talk.
Okay, I think time is short. I think you can.
I think Saxena sir is not able to hear the question. Can you make provision for that?
Yes.
Organizers.
Well, I said very good self-explanatory talk. No question.
Thank you very much.
We are not audible to sir.
Yes, sir. Please, sir. Sir, can talk to you, sir.
Yeah. Participants, please don't miss opportunity to talk. Dr. Saxena sir is a great personality in the field of medicinal chemistry. So please do ask queries if you have.
If there is any question from the audiences, you can ask the question from Dr. Saxena sir. From the offline participants sitting the hall, you can also ask the question.
Sir, there is one question from Kumbo sir again.
So I think you go to next speaker. Next speaker is there?
Yes.
Yeah, I think the time of the next speaker has to be taken.
There is one question from Kumbo sir. For TB and other bacteria should look for colony forming inhibitory activity.
Sir, please recite again.
Can you just
Okay, what to do? Shall I move on for next presentation?
please repeat the question?
Just a minute. Question is, for TB and other bacteria should look for colony forming inhibitory activity. This question is from Kumbo sir.
Okay. Yes, I think one can look into them, but I think this will depend. Actually, that leaf pile will be useful because it stops the food of this, and so they cannot multiply. So if they cannot multiply, of course, the colonies will not be produced. So this is maybe a new target, which may be very useful, not only for in the ones, but also for latent TB because they are inside, so they will not survive. So this can be used for that as well.
Okay, thank you, sir. We'll move on to next presentation now, the last one of this session.
Okay, sir.
So respected again dignitaries, Professor Dr. Indira Ghosh ma'am and participants, welcome you all for the fourth session of second day of this DDDR 2021. For this session, we have with us Professor Dr. Indira Ghosh. Professor Dr. Indira Ghosh is working in JNU since 2008 as Dean and Professor to steer the School of Computational and Integrative Sciences, which deals with computational approach to biology, chemistry, and economics. She has nourished the school as Center of Excellence under Department of Biotechnology, Government of India in computational biology, and spearheaded to initiate a new stream supported by UGC called Complex Systems, harvesting few faculties from physics and econophysics. Yes.
No, there was a call from this number. No query from me. Yeah.
Okay. She has been one amongst the earlier scientists to realize the importance of bioinformatics and initiated MSc courses in Pune University, when she joined as professor in Institute of Bioinformatics and Biochemistry in Pune in 2003, leaving her corporate job from AstraZeneca in Bangalore. After completion of MSc in Physics from Calcutta University, she received in 1982 her PhD at IISc Bangalore under the guidance of Professor V.S.R. Rao in molecular biophysics, a group led by G.N. Ramchandran. She joined University of Houston, USA as Fulbright Scholar during 1983 to 1986 with Professor A.J. Andrew Macomb and contributed to novel method of computational biology. She published one of the first docking algorithm in early 80s as part of her thesis and developed difference of a pre-energy calculation method in molecules during her postdoc. Her major contributions are to develop and contribute in the field of bio and chemoinformatics using systems biology approach to find pathways and target enzymes and developing novel tools for molecular simulations and pharmacophore designs using known protein structure. She has guided 12 PhD, 5 MTech and 20 MSc students, mentored about 7 research associates since last 15 years to direct them towards the evolving field of computational biology. During 2003 to 2017, she has completed 12 projects supported by IBM, DBT, UIT SSF, and Dell ITY. Some of them have potential to collaborate with industry and has published 65 papers as communicating author. So I take this opportunity to welcome ma'am and request her to enlighten us on designing of novel chemicals using molecular multi-target approach on COVID-19 target. I request all participants to ask queries if any once she ends her deliberation. Ma'am, you may please proceed with your presentation. Thank you.
Thank you, Professor Hajare, for giving me this introduction. Do you hear me clearly? And can
Yes,
you see my slide?
No
My
problem.
slide. Okay, thank you. I hope the JIPAR people also are able to see this and hear me. Let me really thank Professor Saxena exclusively, because in 1990, long way back, 91, we organized in AstraZeneca a conference, and I got to know 3 people, Professor Kulkarni, Professor Gupta, SP Gupta, and from CDRI, Dr. Anil Kumar Saxena. That was my first meeting with him. And he actually the person who almost guided me and taught me how to think like a medicinal chemist. My background is physics, biophysics, which really have least overlap with chemistry and other subjects. But he was a wonderful mentor. Many of his papers, today I'm so happy that he displayed some of my papers. And Professor Saxena, if you were not there, I don't think I would have been giving these lectures and called in the meeting of chemists. Only because of him. So you can see him, even such a great mentor for his own colleagues, means, we are of the same age almost. So let me now take you to the presentation. This is a very exclusive presentation, and it will not talk about my many other works which has been cited in different places. The students, if they're interested, they can go to my domain is there, indirag.org, which can explain them whatever other works I do. This work was targeted only few months we could have the work done. It's against to find some chemicals which will be specifically against COVID-19. So there is not much time left. However, I will display if we do not have chemicals, actually we do not have any drugs, but we have many structures, how to go about it. Unfortunately for us, in March, the first structure of one of the protease came out. Of course, the spike protein came out much earlier. And later on, another protease came out. So we used those series of structures which were available for us to generate a pharmacophore. The previous speakers also, yesterday Poraikov told us, Vladimir, and many speakers told us importance of pharmacophore. So I will not talk much about pharmacophore. I will actually go in detail how we developed. And this is a program which we have made the program. We developed the program also for it, and it's available upon request, and you can load it and you can look for many targets. So this designing of pharmacophore is absolutely different from chemist approach of chemistry driven approach. This is structure driven approach. And the guy who did this work within 4 months almost is Pavan, whose picture is shown on the left side. So let me go to, and we have published this in one of the journals in the Finger Science book publication chapter. Generic protocol for chemicals to generate pharmacophore, which some of these type of pictures you have seen in Professor Saxena's slides are mostly in this. So you unalign set of ligands, then you do the good alignment, and depending on good alignment, you get some features. And using these features, you can screen compound library. However, there's publication, but we started working when this paper came out. This is a review, but we started working little earlier than that, and we developed programs and available and published also in 2012. What we did is, instead of following the pathway of ligand-based and pharmacophore and then going for screening, we actually took receptor-based and designed the pharmacophore. Now, this whole technology based, I'll show you, and we have published papers on it, so one can go through details of it. Now I'll show you the target. What is the why and what is the COVID-19? Now, structure-based designing was our goal, but we wanted to find only pharmacophore, because just structure-based, we need to put chemical informations. And as you know very well, I am not a medicinal chemist, neither I am a chemist. My handicap is that I cannot really put a fragment and find how they can be. I can find the interaction, or I can generate pharmacophore out of the most important interaction. And we developed that software between 2009 and 2012, and we used these for so many other presentations earlier. But here the most important are two proteases, and both are cysteine proteases. One is chymotrypsin-like, which is 3C-like protease. Sometimes it's called 3C something, and then sometimes it's called M-Pro or main protease. Another is PL-Pro. So when we started, this M-Pro or ML-Pro was more targeted. And the reason, of course, is very common. That is because it cleaves the whole peptide coming out of the RNA transcript. Now, there is no structural homologs found between either of these two proteins. Hence, may be important. We can really do the work on this. So we chose both of them proteins. And as it belongs to the same cysteine protease, this is very good for multi-target chemical scaffold. However, we started with each separately. So 3CL-Pro or M-Pro is one set. Now, what is our principally what we're doing? M-Pro and PL-Pro. And we developed the pharmacophore knowing their crystal structures. Not one, many structures of each one of them are available. And our program takes care of all the flexibility of these many structures. And we do of course, we find a common binding factors, and later, so that means we'll develop pharmacophore, and then we find each separate pharmacophore we will screen. Also, we will find, is there common between the two, and we find that too. Let me give a little brief about the structures, because most of you probably know it. However, there are three domains in the CL-Pro or M-Pro, which is the major protease. And against this only the Pfizer has raised a compound and found. Now, when we analyze the drugs and when we analyze active site business, so we found this is the place where the active site domain would be. And there were typically like protease designing we are doing for along with the proteases, but earlier we did with acid proteases. Now we have P1 to P4, so all these positions. MPro, very interesting that it has almost 11 cleavage sites, almost more than even on the largest protein possible, that is 790 kilodalton, big replicates. So it actually cleaves many sites. And of course, it starts with the leucine, glutamate, and just between the glutamate and serine, it just chops. And it's quite specific. So this inhibition of this particular protein will block the viral replication. And that's why Pfizer started working on it and made the compound. Now, earlier also some other compound were shown to have inhibited. When we come to PL-Pro, not very much explored, however, structure was known. And this protein is not really acting on specifically on viral proteins. It actually has a surrogate property, and that's an interesting property. It is deubiquitination, which it does on the interferon. And this IFN1, which is important because interferon interferes with the host immune system. Hence, this PL-Pro target would be the best to stop this cytokines efflux. So what we did is, so we decided that we should do both because both are cysteine protease, so they might have some common activity. Well, they have cysteines, however, their orientation or their superimposition does not look that they have similar type active site. How we looked at the other way around. We looked at if we design some compound, there might be some compounds which satisfy the space as well as at the active site amino acids, which it can satisfy. So pharmacophore approach in these cases is best. So now we describe you how that method is. So we do this called ClickFarm program, and we wanted also the flexibility of both of them. So there are 5 structures of CL-Pro or M-Pro, 3 structures of PL-Pro were known those days. So we took them, we did run through our ClickFarm process, which we use 3 major probes or features to identify, and this is the ClickFarm paper, which you can cite here, you can look at it. So these are 3 structures, and each structure, if we take each structure, we do probing by blue, which is amide, carboxyl, and red, which is hydroxyl. So after all those 3, so you see, we have something like 1, 2, 3, 4, 5 structures for M-Pro and 3 structures for PL-Pro. And none of them ligand bound, So we took the native structures. Then we run our softwares, which is absolutely ab initio pharmacophore finding, and we found out many pharmacophores. So we start with the largest possible. So you can see how many are the MIA points, and out of those common, we choose the best binding sites, best energetically, and we found out this is that. Now, anchoring is necessary for this pharmacophore to be somewhere. So we use cysteine as our anchoring. Now we go to the how we use these 7 motifs. Okay, using 7 pharmacophores, 7 side feature pharmacophore. So here it shows how they map in the active site. And this is which amino acids are actually providing this interaction. So if we place a compound at this position of 7 of them, we can satisfy a large molecule. So this is a starting point. So we can have this pharmacophore and search in the database and get some compounds. This is our the one we'll show you. Also, interestingly, cysteine 145 does have a satisfactory hydrogen bonding, that is the carbonyl. Over here, you can see the yellow color. So these are the validation slides. These 2 structures were out after that. And what we did is, you can see these 2 structures, when we put those pharmacophore, which is shown here, 1, 2, 3, 4, 5, 6, and 1 more is hiding in it inside. So 7 of them, so 1 HBA is hiding somewhere, but that is farther away and not satisfied by any of this. So you can see In the case of first, that is peptide known structure came out, which has 1, 2, only 3 of them satisfactory or 4 of them. And the compound which is later on Pfizer made the drug on from 00832531, it has only these 2, 3. So our pharmacophore provides extra features. So modeling can be done to satisfy some of them. Okay, so we are planning to do, means we are already on the work of designing different compounds from this 835231, which can be extended. However, Pfizer has already done some work which we didn't satisfy yet the amide here. It does satisfy one of this blue one, but not the amide. Okay. This is the utility of such pharmacophore and such. This is number 1. Number 2 was another mapping. So this is the mapping. These are the compounds already been known to inhibit. However, not so good. The one best was this PF083. It only satisfied 3. I showed you 3-dimensional structure also known. However, the other variants, except the sequinavir and teliabine, these 2, of course, has more satisfactory HBA and HBG, or hydrogen bond donor and acceptor satisfaction. Now, it shows us that 7-way pharmacophore would have been better to search for more molecules, which I'll show you later. What is the story of PRO? Because we wanted PL Pro also. So here is the designing of PL Pro. We did get a very good 7-feature pharmacophore. Let me explain here. We can start with the maximum 7-feature and then we go down. We have even 4-feature, 5 features, 6 features. And they, of course, will be less in interaction energies because they lose 1 or 2 features. Now, we did map in the 7, and this is the only compound we could find. This is the peptide inhibitor. And we found that they also satisfy quite a number of points. And certainly, this is the cysteine, and which you can see here. Okay, so now if we remember the previous and compare this one, we have common more or less 4 features, maybe superimposable, maybe common. So next comes that, can we make a common? And we did find superimposing the pharmacophore, not the structures, but the pharmacophore, we see there are common 4 motifs and features. And these 4 features actually were also put it in the active site and found that actually we can do, that means that we can do the multi-target. That means we can take the M-Pro, we can take also the PL Pro, and we can find by 4 features some of the hits which can be inhibiting both. Now, how it fits to PL Pro? It does fit very well to PL Pro also, but it is less number of interactions here. And next comes, so we decided that we should have a 2-prong attack. One, that is CL Pro, which is M Pro, which is the protease, which will cleave the COVID proteins. However, PL Pro will inhibit our interferon types. And that 2 of them attack can help most likely the people who would be doing research on. So I hope to show you now using this only PL Pro, only M Pro, and common of them, what is the screening results, and that's shown in this. So these are the molecules on the left-hand side, which contains, all of them are high or good hit, more than 4 side features, at least more than 4 features. Now, let me also tell you one thing. When we do the fitting, we do not do only chemical structures. Actually, we generate conformers within 20 kilocalorie of each of these conformers. And we have taken a ACS antiviral inhibitor data set, and generate 3-dimensionally and pick up if they hit the lowest one. So they may hit in the higher energy level, but lowest one we pick up and show you 2-dimensional structure. All the work done in 3-dimensional conformers, generated conformers. Now, the C is the one which is PL Pro. And when we use only 4, I showed you the 4 pharmacophore, which is selective. Between both of them, it works. We got these 4 compounds are the best compounds on the lowest energy available. And very interestingly, we see also the compounds of these two compounds are very similar to the Pfizer, which has just published a paper on the publication recent. And actually, we could not download even the paper, but we saw the molecule structures, and it did have a very similarity between these two structures. Actually, they have good similarity between all of them, but maximum similarity is on this course. So I would just like to come to our conclusion slide, which is we actually took the protein structure without the any of these bound structures and many structures. So flexibility was our most important. So both all these 4 or 5 structures had different flexibility. Our method or our developed method actually takes care of all these flexibility. And we even did MD simulation, not with this protein, but others, and took the structures, and we can actually map a pharmacophore, which will be, which we call dynaphore, which can be dynamic. Here also we did 5 structures of one and 3 structures of others, and we did a very good pharmacophore designing. And this does not limit us with the flexibility of proteins, okay, because the protein structures are different here, there. Then we got the structure target specific it is. Also, not only we did with the 7 feature pharmacophore searching, we did also common 4 feature. Here we can actually show you how multi-targeted pharmacophore can be used for screening or doing hits. Very interesting to quote yesterday's lecture from Vladimir Buraikov is, he said, actually MPRO has least amount of mutation, which gives more chances for that protein to inhibitor, and probably mutation may not inhibit, may not stop the inhibitions. Then we did the screening hits. This is also done with the repurposed drugs, as you know, but most of these repurposed drugs are against the peptide proteases. Hence, now recent information which is cited here, you can see this compound, and as I said, that it has a very similar core to our structure, and similarity is more than 80%, which gives us the hope that maybe some more structures, little higher energy probably we can explore, and we may find inhibitor. So now testing by Pfizer has been done on every level, but testing for other issue, that is PL-Pro against PL-Pro or interferon, we have to do that. And there are people who are working on interferon, and the best part of the story is this: within 4 to 6 months, we completed the work, and it wouldn't have been possible if collaboration is not international. So here with COVID-19, what we noticed that it doesn't matter what field of science you do, but people are ready to collaborate. So we are trying to collaborate with some experimental groups to find the PL-Pro inhibitors. So let me stop here. But thanks to the JIPAR members, I had a wonderful time in 2020, just before the hit of the coronavirus. And I planned to visit ZIPAR this time, and I made all the arrangements. Then the train started canceling, then red alert started coming everywhere, so I could not go. And physically could not be present at Professor Saxena's birthday, which I really miss a lot. So hopefully we'll make it up later on. Thanks to most of the fundings, etc. No funding, absolutely I had no funding. However, my student did, or my co-worker who is presently working in NII, and he's just joined another institute presently, he used the facility from NII. So I would like to thank NII for that facility he used. Thank you very much. Jio Hajaro Sal, Professor Saxena and Mridulaji. I can never forget their smiling face and their help all the way. Thank you, Dr. Hajare. It was nice to help us in organizing our talks. Lots of things even I learned, and I'm sure the students have learned a lot. thank you. That stops my talk. I hope I have covered the time was lost.
Okay, thank you. Thank you
Any
very much,
question?
madam, for your brief and focused deliberation on multi-target approach for COVID-19. Dr. Ghosh
Any
ma'am
question?
deliberated on a design of pharmacophore and pharmacophore modeling. She talked about multi-target bullet to eliminate COVID-19, under which she explained multi-target hypothesis. She also discussed about different domains to block the replication. She elaborated upon modeling of pharmacophore and mapping its features over known inhibitors. So I hope participants are benefited out of her deliberation. On the behalf of organizers, I would like to thank Dr. Indira Ghosh ma'am for her nice presentation. Thank you very much.
Okay, I would like to read out one question has been asked, or not question, suggestion has come from Professor Kamboj. Dr. Kamboj has suggested, wonderful. Actually, Pfizer drug has modified form now, and they are at clinical trial as the paper published, as well as some of their, their suggestion comes that they have written something. However, nobody has tried their compound and even our compound on the interferon inhibitor. And that's what we are trying to target with the groups who are working in India itself, so that we can be sure. So it will be two warhead, the same compound. It's not synthesized same compound, but it will be same compound will inhibit that and will inhibit this. But testing, actual in vivo testing has to be done in the clinical trial. The reason is, the testing can be separately done in vitro on the culture for interferon inhibitor as well as protease. However, it will be effectively work only in the clinical trial. It will be much better than only MPRO inhibitor. I'm not very sure, but I'm sure Professor Kamboj and Professor Saxena can lead me or guide me how we go about doing the clinical trial.
There are in vivo models also.
Okay.
There are in vivo models also at the NIV Pune.
Okay,
They check
sir.
in vivo models, monkey in vivo models also.
Oh, okay. Oh, that's very good.
NIV
Pune. So I'll try to contact them and see that if they can do the NIV Pune, the monkey model, because it is necessary to test in the interferon as well as that. So standard MPRO inhibitor would cause some specific inhibition. However, the compound which is common to them, they will be even better. So that level will be almost better. Yeah. So I hope the designing part, I am very confident. I hope that this will help us. Thanks, Professor Kamboj, very much.
Thank you very much.
Any other comments? So any other questions, please, also.
Okay, ma'am, there is no question at all.
Oh, actually, Sanjay Batra has suggested to contact Thasti. That will be wonderful. If you know somebody, Professor Batra, if you could know or help me, we will be very happy. The in vitro or in mouse model, because the compounds are available online. So I don't need to make compounds. They are already there. So only they have to do those testing. Okay.
Sanjay Batra is in CDRI. Talk to
CDRI?
Professor Saxena.
Okay. Oh, great. So Professor Saxena ji, we can do that as well. I hope so. Okay. Any other questions, sir? No, I did.
I think there is no question.
Yes. Looks like.
So
people are hungry.
Thank you everyone for attending all 4 nice sessions. I'm once again thankful to all the resource persons and the delegates. And now I'll move on to organizers. Please take charge.
Share nahi hai. Advanced basic. No.
Sir,
How I
now,
share.
now I would like to call Dr. Shishi Nandi sir and Dr. Kapil sir to present the memento to the Dr. Anil Kumar Saxena sir.
Can we clap?
Now I would like to invite Dr. Anil Kumar Saxena sir to present the memento to the our honorable chairperson, Dr. Ashok Hazare sir. Oh, okay. Just a minute. I would like to call the our honorable chairman sir to present the memento to first Professor Indra Ghosh.
Indra
ma'am.
ma'am.
Oh my. Thank you, thank you, thank you. Okay. Did not get. Thank you, sir. Model and can't see. Thank you very much, but I can't see. Ah, great. Really don't know what happens to this. Thank you very much. CPN. Yes.
Now I would like to invite Professor Dr. Anil Kumar Saxena sir to present you memento to our honorable chairperson, Dr.
Yes.
Ashok Hazare sir.
Yes.
I think you have taken all the praise for chairing this session so nicely.
Thank you. Thank you very much.
Thank
So much, very nice comments. Such a short time for all lectures. So thank you so much for spending time for coming and listening to topic. And
This is Saxena sir's moment.
The other one.
Yeah,
Ah.
happens.
Yes, that happens. It's so difficult.
We are not much competent with the technology.
Yes. No, it is not. We are not confident, but the technology doesn't work like our brain. Okay.
Yeah. That
We are
limitation
far ahead.
is bound to be
We
there.
are far ahead. We are far ahead. I don't think it has reached that much. But good that we can at least do, we can do this. It's a
Yes.
great help.
Yeah. In pandemic, really, it worked a lot. It helped a lot.
It has put us on different front, more challenges it gave.
Yeah.
And slowly we are combating. Yes, now it looks gorgeous and nice.
Thank you very much.
Wonderful,
Yes, thank you.
wonderful.
Thank you very much.
To the invited
Thank
speaker of this scientific session. This scientific session is over, and thank you also, Chairperson sir.
Thank you. Okay, have a nice day, one and all.
Thank you, sir, and see you and meet you soon, sir. Wish you very happy COVID-free New Year 2022 in advance, sir. Thank you very much.
Yes, sir. Same to you. Thank you.
Now tea break. I request all the participants to have tea break shortly within 5 to 10 minutes and come back to the seminar hall. But we cannot discontinue the session because we are late. So now I would request Professor Nidula ma'am to take care of the next scientific session. Participants.
Now, I would like to introduce chairperson of this session, Professor Mridula Saxena ma'am. Professor Mridula Saxena, head of Department of Chemistry, Amity University, Lucknow, India, is dynamically involved in teaching and drug discovery research. She worked for her PhD degree in Central Drug Research Institute, Lucknow, and received her degree from Meerut University in 1986. She had worked on different positions like research associate of ICMR, CSIR, and research pool scientist in 1999. Thereafter, she got herself associated with both teaching and research and got DST sponsored project in 2004, as well as faculty position in Amity University, Lucknow. Although her 37 years of teaching and research experience, she has more than 45 papers and new book articles in national and international journals, more than 35 conference papers, and has 4 patents in her credit. She has visited many countries like United Kingdom, Germany, Hong Kong, China, Turkey, South Korea, Greece, and Russia for representing her work. Now I would like to hand over the session to the chairperson. Thank you very much, ma'am.
Hello.
And later switched to the molecular biology. He was one of the pioneers in the new field of graphical representation of biomolecular sequence and the study of sequence characterization, data analysis, and sequence of and molecular functioning, etc. His group also developed mathematical frameworks for quantitative assessment of graphical plots, fixes of DNA, RNA, protein sequences, for quantitative symbol representations. He has edited books and journals and published widely in national and international journals and consistently on peptide vaccine design. Now I request Dr. Gandhi to give celebration.
Am I audible, Shyans? Hello.
Yes. Yeah.
Shyans, am I audible?
Yeah, you're audible.
Shyans, am I audible?
Yeah, you are audible. Am I audible?
Okay. Now you can share your screen.
Oh, okay, sure. Fine. Thank you.
You can share your screen.
Yeah. Is my screen visible right now?
Yes, yes, you are audible.
Yeah. And is my screen visible? Is my screen visible right now? Hello? Hello, is my screen visible? Can anyone confirm, please?
Screen is visible to us, Shyans. You can present.
Yeah, sure. Thank you. Thank you so much.
Yes.
Thank you so much. I would request Professor Dr. Ashish Nandi to begin with the presentation if he's available. Like,
okay, can you hear me?
Yeah, sure.
Yes, sir. Yes, sir. Okay. So my thanks to Dr. Shishir Nandi and the organizers of this for inviting us to give our inform about our work. Next slide. Next slide. Yeah. So this work is basically about design of antiviral vaccines, peptide vaccines, and how it takes care of mutation. How it takes care of mutated sequences. The principal tool that we use is graphical representation and numerical characterization of sequences for this job. It is one of the things that I had I was originators, and it is now a subject that is very widely used. What we will do is present to you first how we use the tool to generate peptide targets for peptide vaccines. A review of this is going to be published in CCHTS Volume 25, Issue 3 soon. So you might look at that for more details. And the important thing is that we use this tool for viruses, and we have used them for many viruses, including a lot of mutations thereof. So since I have some problem, I'm handing over the responsibility to Shubhamoy Biswas to give a talk about the design of the vaccines, and then Shreyans Chatterjee to talk about how the mutated sequences are taken care of. And I would also like to mention here that in this, using this tool, we have had sequence, we have assigned descriptors to each sequence, and the index for that is GR for RNA sequences and PR and QR for protein sequences. Using these three types of sequence descriptors, we have been able to compare sequences in an analog-free way, whereas usually it has been one uses BLAST or some things like that for comparison. So this is the first time that AFSDs have been used in these comparisons. So can I ask Shubhamoy to continue?
Yes, sir, sure. Next slide. Next slide. To begin the discussion, first we will talk about how graphical and numerical characterization of DNA and RNA sequences are done. So after getting the sequence, we read them base by base. So accordingly, we get 4 different options for nucleotide bases, as we know, A, G, C, and T or U, depending on whether it's DNA or RNA. And in a 2D Cartesian coordinate system, we represent these 4 nucleotide bases along 4 different cardinal directions. And we respectively move along by 1 unit along the respective directions based on the kind of base that we get while reading the sequences. And accordingly, we plot the points. Now, after plotting the points, we see that when we find the first order moment along the x direction, we get a value mu x, which is given as this formula. And similarly, we get the first order moment mu y along the y direction. And using these two values, that is mu x and mu y, we get a quantity which is known as the graph radius, or GR, which is given as the square root of the sum of squares of mu x and mu y. And what we do is we represent this DNA or RNA sequence using this single value, which is the GR. Next slide. So as you can see on the screen here, this is the picture that we got for the GR plot for the nucleotide sequence of the surface glycoprotein of SARS-CoV-2. And from these plotted points, we find the mu x and mu y, and accordingly, we find how the GR value is obtained. Now, this entire methodology is for DNA and RNA sequences. Similarly, for protein sequences, we consider a hypothetical 20D system where we generate similar kinds of moments and sequence descriptors like PR and QR. Next slide. So this slide represents the entire protocol for the vaccine design. First, we consider the surface protein of the virus and we consider its nucleotide sequences. Now, the data set that we get for these nucleotide sequences is very noisy because they undergo a lot of spurious mutations and as well as some mutations which are quite widely seen in the population. So accordingly, we need to filter out the most significant sequences, and then by filtering out the significant nucleotide sequences, we find their protein sequence equivalent to obtain the finally filtered data set. Now, after obtaining these filtered protein sequences, we characterize them using characterization methods and we find how their mutational pattern looks like. And after applying computational algorithms, we try to understand that in which part of that particular protein sequence the region is surface exposed or is conserved against mutation. And then after finding such regions, we again consider different such analysis like epitope potential analysis, or checking whether they cause any sort of autoimmune threats, or even visualizing their 3D structural details. And after all of this, the regions which satisfy all of these criteria will finally appear in the final shortlist for the peptide vaccine candidates. Next slide. So first is the filtering of the data. So after retrieving all the nucleotide sequences for the surface protein from databases like NCPI or GISAID, we find the GR values for all of them. And accordingly, we plot a histogram for them. Now we consider the center points for the histogram bars, and using those center points and their coordinates, we fit a Gaussian curve like the formula given below. And after getting the Gaussian curve, we fit a full width half maximum criteria to get a bandwidth. Now the nucleotide sequences which will have their GR within this bandwidth, they will be accepted, and the rest of them will be rejected, because the ones which appear inside the bandwidth, they have much greater amount of frequency. Now the nucleotide sequences which are appearing within the bandwidth, there are also many duplicate ones, because if two nucleotide sequences are completely identical to one another, their GR value will also be identical. So we use this criteria to remove the duplicate ones, and finally we have obtained a crisp set of nucleotide sequences that have discrete GR, and at the same time they occur within the bandwidth. And accordingly, we find their protein sequence equivalent to form the finally filtered data set. Next slide. So here we have considered the case for hemagglutinin protein of H1N1, which is a surface exposed protein, and we have considered the sequences from 1910 to 2018. And accordingly, we have fitted a Gaussian curve like this shown below. And you can see that if we find the bandwidth according to the full width half maximum criteria, we can find out the significant nucleotide sequences within that range. And accordingly, we can do the respective analysis. Now, the major problem that we faced while finding this Gaussian curve for spike protein for SARS-CoV-2 is that there was not much data present with respect to time. That means here you can see for hemagglutinin, the data was available from 1910 to 2018, whereas SARS-CoV-2 has occurred only for 2 years. So if we could have got more amount of data, we could have found the Gaussian curve with better accuracy. For that reason, we have considered only the histogram, and we have tried to estimate the bandwidth according to the following figure. Next slide. So here you can see this is the histogram, and we have accordingly considered the bandwidth, and the bandwidth was around 367 to 369. So in this range, the GR values varied. Next slide. Now, when we have already got the filtered set for the protein sequences, now we represent the protein sequences in a hypothetical 20D system for their characterization. And accordingly, there are 20 different amino acids. So we represent them along 20 different directions, and we move along one unit in a similar way like before. And accordingly, we get 20 different first order moments as mu1, mu2 up to mu20. And from these values, we find the protein radius or PR value for that particular protein sequence as according to this formula. So we'll be representing this entire protein sequence by this formula PR. Next slide. Now, with respect to the protein sequences that we obtained, there are two primary characteristics that we will be considering. First is the protein variability, and the other one is surface accessibility. So here in protein variability, first we consider a protein sequence, and we consider all possible 12-link peptides. Here you can consider any other number, but for considering the fact that the peptide link must be around 8 to 20, we have considered an average value of 12, and we have done that. So we consider all possible 12-link peptides from the sequence, and we find their respective PR. Now, for a particular 12-link peptide, the way their PR value varies across the entire data set, that gives its protein variability. So if the protein variability value for a particular 12-link peptide is high, so we can say that it has undergone a greater amount of change across the entire data set. So we can say that it undergoes greater extent of mutation. Similarly, from the surface accessibility value, regarding the surface accessibility value, we use the Sable protein prediction server, and accordingly we get the surface exposure level for each of these 12-link peptides, and we denote it as ASA. So for 12-link peptides, we have two parameters right now. One is the PV value or the protein variability, and another is the ASA value, that is surface accessibility. Next slide. Now, if PV represents higher mutation, then 1 by PV would mean that the peptide is more, and then if 1 by PV is high, then we can say that the peptide is more and more conserved against mutation. So we want a particular 12-link peptide to be least mutated and highly surface exposed. So we want high ASA, and we also want high 1 by PV. So if we consider a W parameter value, which is given according to this formula, we can say that if ASA and 1 by PV value both are high, then W parameter will be high. And because we want that both these parameters need to be high, so the 12-link peptide must be suitable. So from this, we can conclude that if the W parameter value is high, then the 12-link peptide will be suitable for selection. So using this particular value of W parameter, we rank all the 12-link peptides that we have obtained, and accordingly, we produce a ranking list. And from this ranking list, we consider the top 50 or 100 ranks. And what we do is now we consider this top 50 or 100 12-link peptides, and we try to cluster them according to their location in the protein sequence. So say, for example, there are two peptide lanes that are very close to one another across the sequence. Then we club together them under a single peptide zone. And if they're far away, they are part of different peptide zones. So in this way, we get the peptide zones. Next slide. Now that we have got the peptide zones, now we consider a novel mathematical model known as the 2D polygon representation. So for a particular peptide zone, we already have the overall surface exposure value. We already have the overall value for conservativeness against mutation. And now we consider a third parameter, which is known as the area that it covers on the surface of the protein. So why area is important? Because when we are considering a peptide zone, and when we know that we will be performing further analysis like epitope potential checking or autoimmunity checking, then we want to have a greater amount of flexibility in choosing which part of that particular peptide zone satisfies all these criteria. So for that reason, if we choose a greater amount of area, we will be having greater amount of flexibility. So in this way, we will be having three different parameters, that is surface exposure, conservativeness, and area. And we want all of these three parameters to be high. Now we represent ASA 1 by PV and N value. N means here the area. So we represent these three parameters along the three different arms of this triangle given in the figure. So here you can see there is OA, OB, and OC. These are the three arms, and accordingly they are separated by 120-degree angles apart. And then we represent each of these three parameters along the three different arms. And you can see that if the value of these arms OA, OB, and OC, if they start increasing, then the value of the area of the triangle ABC, that will also start increasing. So we can say if the value of these parameters are all equally high, then we can say that the area of the triangle is also high. So we can use this code to represent the peptide zone, and we can say that if the value of this area is high, then the peptide zone is suitable for selection. So based on this value of area, we rank the peptide zones and we select a particular threshold or cutoff, based on which we select the best peptide zones and reject the worst. Next slide. Now, after getting the filtered peptide zones, we check whether they are capable of producing enough T cell and B cell immune responses, and that we do using the immune epitope database analysis resource. And the ones which give good amount of epitope potential, we then further consider whether they cause any sort of autoimmune threats inside the human host. And we do it with the help of basic local alignment search tool using the E values, and we see whether they cause any sort of autoimmune threats. If they don't, then we consider them further. And we try to visualize them, how they're present on the protein surface, like whether they're covered by any neighboring amino acids or they're completely exposed. And that we do using platforms like CN3D and PyMol. And after doing all this analysis, finally the peptide zones that we get, they satisfy all these criteria, and we list them in the final shortlist. Next slide. So the list that you are seeing over here, this is the final shortlist that we have obtained for the SARS-CoV-2 virus using its surface glycoprotein. Now, one interesting observation that we found was that the mutations that have occurred in the variants of concern and the variants of interest of SARS-CoV-2, all these mutations are not at all present in these peptide regions. That means if we produce a kind of peptide vaccine using these regions, and if we apply them against these variants, then because such mutations are not present in these regions, these vaccines will work effectively against these variants. So this is a very important observation. Next slide. So this is a sample region that we have shown for 527 to 541, which had none of the mutations in the variants of concern. And also the mutations that have occurred in the Omicron variant that is currently surging, none of these mutations have occurred in this region as well as the other two regions. So this is a very important observation regarding whether the peptide vaccines produced from these regions whether they are effective against all these variants or not. So this is a very important observation. Next slide. So with this, I come to the end of the protocol for designing peptide vaccines. Now I leave it to Mr. Shren's strategy to explain how this entire scheme also helps in understanding the mutations occurring in the different variants of SARS-CoV-2 as well as other emerging pathogens. And over to Shren's. Thank you.
Thank you so much, Shubhamoy and Professor Nandi. So I would now like to focus on viral mutations and how our protocol, how our algorithm is quite efficient in fighting against it. So now we are, so in this time, we are like confronted with SARS-CoV-2 virus, and here I've just shown a genomic sequence of SARS-CoV-2. We can see two ORFs, which codes for polyprotein 1 and polyprotein 2 via a -1 frame shift. And after that, there comes the structural proteins, the spike glycoprotein, the envelope protein, the membrane glycoprotein, and the nucleocapsid, among which the spike glycoprotein is one of the most important proteins in a virus that we are targeting, because with the help of spike glycoprotein, any virus, especially the COVID virus, enters our body that is infect us. And the ORFs code for 16 non-structural proteins, which are formed by the enzymatic actions of host proteases, host chymotrypsin-like proteases, and main proteases. Okay, so the spike protein is composed of 3 chains, chain A, chain B, and chain C, and 2 domains, S1 and S2. So the host proteases cleaves the 3 chains, which are shown in the right-hand side of the image, into 2 domains, S1 and S2. The S1 contains the receptor binding domain, which helps in attaching with the human angiotensin converting enzyme 2 or ACE2 receptor. And the S2 domain helps in membrane fusion by interacting with the TMPR SS2 co-receptors. Now mutations. So when a gene is damaged or modified in such a way that now the genetic message that is carried by the gene, or that was used to be carried by the gene, is now altered, we call it a mutation. So there is viral DNA or viral RNA, which is affected by mutagens, which can be physical or chemicals or any other environmental factors. It changed the codons in the DNA or RNA. So now it codes for a different protein, which is a mutated one. Now this mutated protein, if the mutation occurs in the active site, then it can have an altered binding efficiency with the human receptor or any other receptor which it binds to. Now this binding efficiency, it can increase or it can decrease. I'm not telling that it will increase all the time, but here comes a different discussion, which I will come to a bit later. Now, when there is an altered binding efficiency, there will be a variable infectivity. Now, it has been studied that viral mutation rates roughly range between 10 to the power -8 to 10 to the power -4 substitutions per nucleotide per replication. So it's a quite huge number. But the good news is SARS-CoV-2, which falls under the coronaviridae family, it has a large genome, and hence mutations are not quite oftenly seen in viruses of the family coronaviridae than they are seen in other viruses. Now, to analyze the mutations, we use a method known as the QR method. So it is a technique to represent protein sequences in the 2-dimensional Cartesian plane based on their hydrophobicity indices. So what we do, we have 20 amino acids. We divide our 2-dimensional Cartesian planes into segments where the angular difference between 2 amino acids is 18 degrees. Now, how do we plot this thing? So if we consider like the first amino acid in our sequence is alanine, then we start from the origin, which is 0, 0, and we go 1 unit distance at an angle of 18 degrees from the x-axis in towards the direction of alanine. And then if the next one is cysteine, we go another 18 degrees. And if the next one is valine, we go another 18 degrees. In that way, we just change our center of origins, and hence we plot this graph. And the QR, the mathematical descriptor of peptide sequences, is a weighted center of mass of the graph plot. In the right-hand side of the slide, we can see that there's a difference between, there's a QR shown, QR graph shown, which exhibits the difference between the SARS and the SARS-CoV-2 spike glycoproteins. The QR is basically an alignment-free model to understand protein mutations. And we can here visually represent peptide sequences based on the intrinsic property of interaction with water. So we are not considering the primary sequences only, we are also incorporating their hydrophobicity or hydrophilicity indexes, which are very much important for designing peptide vaccines, because the epitopes that we want to target, they should be surface exposed. So the hydrophobicity and hydrophilicity indices are very much important in these studies. Now we tend to find out the mutational hotspots. So why do you want to find out mutational hotspots? Because a very common notion in biology is survival of the fittest. So if a mutation occurs in a particular amino acid in the protein, if the mutation is beneficial for the virus or for any other organism, it tends to grow, like it tends to grow through generations. So we try to understand if the particular mutation is more accepted, that is, try to find out the evolutionary significance. So what we can see is like the frequency of mutation for an evolutionary accepted mutation will be higher than any other mutation. Now we try to understand the positions of amino acid inside the protein sequences where mutations have occurred frequently. And in the left-hand side of the slide, we can find out that we can see a huge spike at the 614th position of the spike protein of SARS-CoV-2. So here comes a D614G mutant, which is actually very much important whilst we want to study the SARS-CoV-2 virus. Here I've shown the variants of concern with the potential mutations that has occurred inside them. And here we can see each and every variants of concern has shown a D614G mutation, which was here shown in the previous slide, which showed a greater spike. But fortunately, no mutations are found in the variants of concern which were present inside our peptide vaccine candidates. So it is a very good news. So our peptide vaccine candidates can actually fight these variants of concern quite efficient. Here I've shown the mutations that are occurred in the Omicron. The largest number of mutations are occurred in the Omicron strain of B11529. Here also we can see a D614G mutation. Specific mutations occurs from the 655th to 681th position, where a mutation is present in the S1, S2 of the spike domain 1, spike protein 2, spike domain 2 furin cleavage site, which scientists are hoping can be a reason for high transmissibility of the Omicron virus. And the deletions are also seen in alpha and iota variants of concern. But a very good news is also like our peptide vaccine is quite efficient, and no mutations which were observed in the Omicron variants were equally observed in our peptide vaccines. So what does this signify? So none of the residues in our potential peptide vaccine candidates were mutated. So our epitopes, which we considered were very much conserved and had less protein variability. And since we optimized our sequences to have lower protein variability but higher amino acid solvent accessibility, the calculation of the W parameter, which was shown by Shubhamon, and the 2D polygon score was highly accurate. So our algorithm was very much efficient in finding peptide vaccine candidates. So this protocol can also be used to find potential peptide vaccine candidates of other emerging pathogens, where like influenza, it can be dengue, it can be like any other virus which is not yet occurred but can occur in the near future. And the optimal balancing between protein variability and amino acid solvent accessibility helps us to obtain peptide regions which are highly conserved and highly accessible by solvents. So since they're highly conserved, antibodies usually produced against them can be very efficient in combat. And since they're very much solvent accessible, so they will be surface exposed, and our antibodies can easily find them and get primed. And so what can we conclude from this? So our method allows a lot of mutational variability in the viral sequences. So mutations will occur and can occur in viral sequences, viral spike proteins or any other protein. But our algorithm will actually find sequences or find sites where mutations have occurred the least. So they are the highly conserved sequences with lower protein variability. And the very silver lining to this is the Omicron variety falls in the same class as other variants, and our prescription of potential peptide vaccine candidates would be able to deal with it like it can deal with the other variants. With this, I would like to conclude with the references that we have used in designing our slides and in our studies. And this is our team, which we can see Professor Dr. Papia Nandi, Dr. Ashish Nandi, Professor Dr. Subhash Bosak, Ms. Konika Bosak, Professor Shukhen Das, myself, Prathagata Shubhamoy, and Dr. Sarojit Nam. So thank you so much, and we are now open for any questions or any comments. Thank you.
Thank you so much, Dr. Nandi, for an excellent talk. And he has covered the different techniques and tools for designing new vaccines and explain how the mutation problem can be tackled for designing of new vaccines, and we can combat these viral diseases, etc. Now the talk is open for discussion. If you have any questions, you can raise. It's repeating.
I have put my question in the chat box. No suggestions.
Now Dr. Kamboj wants to ask some question.
No, I have put already in the chat box. I said very good approach, but I said look at the 40 vaccines which are in clinical trial and another 120 which are under preclinical development. And please see also the efficacy of Zycovid D, and Covovax, which are both protein vaccines and are clinically active. He should look at the structure of these proteins.
Yeah, thank you so much, Professor, for your suggestion. We would surely go through the details of those Zycovid D and Covovax, and also we would like to compare with our models and our design peptides and try to find out how to increase the efficacy and everything. Thank you so much.
Yeah, please look at the 120 vaccines which are under preclinical development and 40 under clinical development. Because
Yeah,
all are available now. All are published.
It's
sure, sir.
Thank you.
Thank It's echoing.
Now you can invite next speaker.
No, okay. Now I invite the next speaker of this session, Dr. Shishir Nandi. Dr. Shishir Nandi completed PhD from Indian Institute of Chemical Biology, Kolkata, as a CSIR Gate Fellow and had been awarded PhD in Pharmacy degree in 2011 by Jadavpur University, India. He did his postdoctoral research as a European Union Marie Curie Fellowship in Laboratory of Chemometrics, National Institute of Chemistry in Ljubljana, Slovenia, Europe. His specialization is pharmaceutical chemistry and research is based in the area of drug design, which includes QSAR, drug design, biological activity prediction of lead compounds, ligand receptor interactions, and virtual screening, combinatorial library design, and lead hopping. He published more than 90 original research articles and review in reputed international journals having high impact factor. He published 5 books, chapters, Springer Nature, and Bentham books. He presented his research work in many international conferences around the world. He is the guest editor of Current Signal, Transduction Therapy and Current Pharmaceutical Design and Editorial Advisory Board of the Cancer Immunology Journal and Journal of Bioanalysis and Biomedicine. What is the Journal of Computational Methods in Molecular Design and many other. He has organized international conferences like CPTPP 2014, CMTPI 2017, CTPMS 2020 as a organizing secretary. Dr. Nandi as a PI completed the project entitled Study of Groundwater Quality of Kashipur Industrial Area granted by Water for Welfare and Uttarakhand Initiative, IIT Roorkee in academic year of 2018 and 19. He is having more than 15 years of research experience and he has been guiding many masters and doctoral students. Now I invite Dr. Nandi for his deliberation.
Thank you very much. Thank you very much, Nidura ma'am, for my brief introduction.
Actually, eco
to have a nice scientific interactions. My talk is based on
Can India get COVID-19 third peak? Will it be more dangerous than the second peak? Does herd immunity develop? These are the questions appear in mind, and it is because, as you know, India is India is famous for high population density. So it is really difficult to vaccinate 138 crore people within very short span of period. There is no specific anti-COVID-19 chemotherapeutic small molecule oral formulations chemotherapeutics till now. The drugs which are given, that is repurposing, existing drugs are repurposing to combat the COVID-19 except vaccination. So how long does it take to develop a specific anti-COVID drug discovery? Already Dr. Saxena discussed. So if we see the drug discovery research, it takes minimum 14 to 16 years to develop 1 to 2 million, and at the expense of $1,000 billion. So it's a long journey. So how is it possible? COVID-19 erupted by the end of 2019, but how is it possible to discover small molecule in a very short period of time? So now the big query about our existence, because COVID-19 lost lives, engulfed the lives, economy, social security. But existing antiviral, antimalarial, antibacterial, antiparasitic, immunomodulatory natural agents, immunoinflammatory agents, they are repurposed to combat the COVID-19. But what are the mechanism of action of these drugs? To explore the mechanism of action of these drugs, we should know what are the different targets, what is the structure of SARS-CoV-2. So if we see, this is the spike protein primary structure, very dangerous. Then envelope protein, it can coat the membrane. That membrane can protect the nuclear protein, and nuclear protein is composed of this genomic RNA. That is very dangerous. This genomic RNA is very dangerous. This genomic RNA responsible for multiplication, and it is responsible to attack the immunity of the host. Now, we know that immunity two types, active immunity and passive immunity. Passive immunity we achieve from the mother. It is long lasting till death. And active immunity, it is when the person is exposed to infection or vaccination, then active immunity develops. It is not the long lasting. So, to stabilize our passive immunity, we must have to take the mother natural resources. So, what are the different resources? We have Mother India gifted us. These are the natural resources: cinnamon, cardamom, kalanji, black pepper, clove, haldi, anise, ginger, garlic, green tea. These are the mother natural resources that can stabilize our passive immunity. And you see, these are the ashwagandha, giloy, tulsi, muleti, kalme, cinnamon, garlic, kalanji, adrak. These are the natural resources to boost the immunity, to stabilize the passive immunity. These are the formulations developed based on the natural Anudalia, Saraswati Bhatti, Mamsamani Bhatti. Next. And now, what is the mechanism for COVID-19 resistance strain, which is very dangerous? This SARS-CoV-2 strain, it infects the animals like cats, bats, and then healthy humans. Then there is a cross-species transmission between animal to healthy human and healthy human to animal. This cross-transmission, it produces highly virulent strain through genetic reassortment. And this highly virulent strain this is responsible to kill the person. This is very, very dangerous. So this SARS-CoV-2, it enters into the human body through nose, through mouth, it reaches into the intestine, and through ACE2 receptor, it enters into the blood. Our phagocytosis system is activated. This SARS-CoV-2 is very dangerous, silently enters into the phagocytous cell, and the person who is immunocompromised, that phagocytous cell burst and extract all the power and become very dangerous. And then this energized, accelerated SARS-CoV-2, it reaches into the intestine, heart, kidney, liver, where there is more expression of ACE2 receptor and engulf the ACE2 receptor. So function of ACE2 receptor lost. So ACE2 receptor is expressed, is present in intestine, liver, heart, kidney. So multi-organ failure. That's why the patient is going to death. The most dangerous thing is spike protein, which is trimeric in structure. And this, you see, this trimeric structure, it occupies the ACE2 receptor of the host, and then it fused and enter into the human body. Anyhow, if we can inhibit this binding between ACE2 receptor and trimeric structure, then inhibition of SARS-CoV-2. If we can destroy this primary structure, then we can inhibit the SARS-CoV-2. But how is it possible? These are the natural agents. They have been clinically tested as SARS-CoV-2 spike protein inhibitor, like curcumin, neem, withania, piperine, berberine, and togapoline. These have been clinically tested. Next. You know, 9 emergency use of vaccine have been approved by the authority. Next. So these are the vaccines: Covishield, Sputnik V, Convadia, PIDMC, Covaxin, Coronavac, BTIVP, Comirnaty, and Spikevax. These are the 9 vaccines which have been approved for the use. And these vaccines, these 4 vaccines, these are adenovirus vector. Means they are formulated by using the adenovirus, who are able to, adenovirus virus is able to produce the spike protein, and our body treats as entry of the SARS-CoV-2 and develop the antibody. In case of inactivated, and this Covaxin, Coronavac, IPV4, these are based on inactivated T virus, and the patient, asymptomatic patient, this virus is isolated, is taken from the asymptomatic patient and deactivated by using heat or chemicals, and this inactivated virus suspension is injected to develop the antibody. And this community developed by Pfizer and Spikevax, these 2 vaccines are based on mRNA vaccines. This is based on the modified RNA. Yes. COVID-19 is critically managed by the repurposing of these drugs, which are approved by the FDA, like remdesivir, favipiravir, ribavirin, galidesivir, nitazoxanide, raloxifene, lopinavir plus ritonavir IV injection, Cathepimab plus Indevimab IV injection, antimalarial like chloroquine, hydroxychloroquine sulfate, antiparasitic like ivermectin, antibacterial like doxycycline, azithromycin. Immunosuppressant as anti-inflammatory agents like dexamethasone, methylprednisolone, statin derivatives like simvastatin, ACE2 receptor antagonists like losartan, vitamin C, ascorbic acid, capsule of zinc and vitamin B complex fortified vitamin C, enhancement of lungs capacity, like antibiotics, to enhance the antibiotic activities, amroxol and promethazine, or DRDO developed this 2G. These molecules, these are the approved drugs by US FDA to repurpose to combat the COVID-19. Molnupiravir by Merck and Beeswax, they have developed this molecule, and this is clinically 50% effective under safety clinical trials. And ritonavir, Paxlovid, this has been developed very recently, about 89% clinical effective in phase 3 clinical trials, already discussed by Professor Vladimir and Professor Bosa and Professor Indira Bose also. Very recent information, so it's a good news for us. So SARS-CoV-2, this genomic RNA of SARS-CoV-2 very dangerous because it is responsible for replication and transcription, and it kills the immunity of the host. Therefore, that this is the main target. If we inhibit the SARS-CoV-2, we can inhibit. Our laboratory, Jipal laboratory, computer-aided drug design laboratory, they have done original research based on structure-based modeling, utilizing the molecular document. So our laboratory has selected these 34 compounds: ampinavir, adagiranavir, darunavir, bosantinavir, intinavir, lopinavir, entinavir, ritonavir, satinavir, tipranavir, TMC 310911, and hepatitis ACV inhibitors, protease inhibitors like asunapravir, osipravir, selupravir, danopravir, Allapravir, Rajupravir, Lipapravir, Nalapravir, Caritapravir, Imanavir, Simulpravir, Soposopir, Sovapravir, Delapravir, Oxalapravir, and these are the miscellaneous like Fluvenapir, Oseltamivir, Remdesivir, Favipiravir, Chloroquine, hydroxychloroquine, Ribavirin, Bencyclovir, nitroxoxide. These 34 compounds we have taken, and we have brought this compound against this SARS-CoV-2 main protease enzyme. Next. These are the structure. Next. So this 5 or 7 day, this is the ID code of, we have safety from DDDR and executive structure of SARS-CoV-2 main protease, and we docked using R2 cell. Next. Next. So you see this our docking studies, they have predicted this molecule already. Dr. Saxena has known remdesivir, chloroquine, favipiravir, ribavirin, bencyclovir, nitroxoxide. Our docking studies, this has been predicted also as a potent molecule, and these compounds have been tested and published in Cell Research. And this is the dog score, our dog score, and this is the PECPP values, experimental value. Now, to validate our studies, docking studies, we have designed an equation by taking a correlation study. Next. Yeah, this is the correlation, and it was seen that the correlation between dog score and activity is correlation square is 0.899. So it's a very good validation study. So our predicted docking model is good, and this is the equation. Next. Now you see these are the 12 molecules: ampinavir, darunavir, sobuspuvir, omiprenavir, oseltamivir, remdesivir, favipiravir, chloroquine, hydroxychloroquine, ribavirin, bencyclovir, nitroxoxide. These are the 12 molecules our docking study is predicted. And out of that, sobuspuvir and remdesivir, these two molecules gives the best prediction. And this is the model. Now this article published in CCHPS journal. Now, we can use this model, this one variable y is equal to minus 0.936 x minus 7.631. So this model we can take as HTS, high throughput screening model, to predict the other molecules. Next. Now you see these are the compounds which are given for the treatment of COVID-19 patient. I discussed with Dr. Saxena, I discussed with Dr. also. Dexamethasone, doxycycline, azithromycin, clarithromycin, ganciclovir, tenofovir, azbudin, cefixime, cefodoxime, ciprofloxacin, parfloxacin, dapagliflozin, ofloxacin, moxifloxacin, levofloxacin, bedaquiline, EDAJ, 876, lefamulin. You see, these are the compounds which are given for the treatment of COVID-19. This dexamethasone, doxycycline, azithromycin, clarithromycin, ganciclovir, tenofovir, azbudin, cefixime, cefodoxime. And our docking studies, they have been predicted. But our docking studies cannot predict this azithromycin, clarithromycin. So maybe other mechanism of action, because we have used this SARS-CoV-2 vectorial design. And what we did actually, for the chest infection, pneumonia, and for the tuberculosis, these compounds are given. These are the given as a drug molecule. Therefore, pneumonia is common for SARS-CoV-2 infection also. Therefore, we have taken this molecule in our study to dock against SARS-CoV-2 protease, and we have seen that these are successfully docked, and out of these compounds, bedaquiline, lefamulin, they are giving the best dock score, and their experimental activity, their predicted activities was 0.783 and 0.937. So these two compounds can be predicted as highly potent molecules, and this article published in CCADD journal. So this is the beauty of our model, and this model has been developed in our JIPA library. Next. So apart from mask, social distancing, sanitizer, and we should take a healthy food, particularly vitamin A, vitamin C containing rich food like carrot, green peas, tomato, sweets of pumpkin, bitter gourd, methi, spinach, broccoli, green papaya, rabada, cinnamon, soap, black pepper, kamusi, turmeric, natural spice, green tea, almond, garlic, milk, fruits, amla, pears, vitamin C containing fruits, and non-veg fish. Next. Finally, some ideas for collaboration. This 6Y6C, this is the gene, human gene that inhibits the CoV entry, coronavirus entry. So genomic scientists they can explore this gene. Now, two anti-coulomb sensing drugs that could allow with the purpose anti-COVID-19 chemotherapeutics. These are some ideas for collaboration. Next. Now, I would like to thank my academic Guruji, Professor Saxena, and academic Guruma, Professor Vinudha Saxena, for teaching me, because I have been taught a lot in Islamic communication, and I thank to my student Mohi and Namita for doing a lot, and this Mohi, because without student I am blind, they have done this work, particularly this fellow Mohit, he has done this work, HTS micro screening work, and my collaborator, Dr. Asmita Sumaswamy, or it is from Nirmala College of Pharmacy. We are doing a great collaboration. Next. And these are some articles that published in high impact journals, and these are based on COVID-19 design. Finally, I salute my mother India, and thank you all.
Thank you so much.
Thank you so much for excellent talk, Dr. Nandi. Your talk was very informative and indicated the use of different natural products to boost the immunity. Now the talk is open for questions. If any question is there, you can ask. Any question? If there is no question, then we can invite the next speaker. Now the next speaker is Dr. Chandra Saurabh Azad. He was born in Uttar Pradesh and he graduated from Hindu College, Muradabad, and he earned gold medal for his master's degree, and he passed from Mahatma Jyotiba Phule University, Ruhelkhand. He completed his PhD at the Council of Scientific and Industrial Research CSIR CDRI in Lucknow. Under the guidance of Dr. Anil Kumar Saxena, he joined Professor Mark Olson's research group as a postdoctoral fellow at the Tianjin University, China. After completing his first postdoctoral fellowship, at Guru Gobind Singh Indraprastha University of New Delhi. In September 2021, he joined Professor Fraser Stoddard, Nobel laureate in 2016 Chemistry Department Research Group, and focusing on the synthesis and fluorescence characteristics of supramolecular self-assemblies and molecular machines. Now I invite Dr. Azad for his deliberation.
My screen is visible to everyone. Thank you, ma'am. Such a nice introduction. It's good to see you after a long time. I'm audible to everyone. Hello. Hello. Am I audible? Am I audible?
Now you are audible. Now he is
Okay.
audible.
Now my screen is in the sharing mode.
Yes, 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 you are audible.
Okay. So good afternoon, everyone. Today I'm going to talk about the synthesis of highly fluorescent thiazole-based self-templated surfactant as a sensor of tryptophan and melatonin. Currently, I am working in Northwestern University, and I'm working with Sir Fraser Stoddart, who is the recipient of 2016 Nobel Prize in Chemistry. So before starting with my talk, I want to talk about some memories associated with Dr. Saxena when I was joined the PhD under his supervision in CDRI. And you can see me like standing right behind there in a violet circle. And I spent so many good memories with Dr. Saxena. This is the picture that we are celebrating the birthday in the lab. And this is the final day when my PhD got signed by the Dr. Saxena. So let the talk begin. So my work depends on the sensing of amino acids and especially the tryptophan. And tryptophan is very common amino acids, and we know that it is an essential amino acid, and it's known as a very significant role in human and animal neuroregulations, organ development, metabolism. L-tryptophan is one of the 8 essential amino acids for human normal body activity, including brain function, neural regulatory mechanism. Many reports find that serotonin and melatonin are metabolites of the L-tryptophan. When L-tryptophan is improperly metabolized, toxic metabolites are produced in the brain to cause schizophrenia, drowsiness, nausea, hallucination, headache, loss of appetite, and maybe the Parkinson disease. Unfortunately, the amount of L-tryptophan in vegetables and fruit is very inadequate. A human and the animals cannot synthesize the L-tryptophan themselves. It must be obtained from food and pharmaceuticals formulas. The WHO recommends the dose of intake of L-tryptophan as a 4 milligram per body weight kg per body weight per day. However, excessive L-tryptophan may produce toxic waste in the brain, causing delusion and hallucination. Therefore, detection of L-tryptophan for preventing brain disease is very significant. And now it's the L-tryptophan, and we can talk about a little bit how it metabolizes and how it regulates our brain function. Tryptophan just metabolize the 5-hydroxytryptophan with the enzyme, the 5-hydroxy tryptophan 5-hydroxylase, and it's further metabolized to 5-hydroxytryptamine, that is known as the serotonin. It's a very important neurotransmitter that further metabolized to N-acetyl 5-hydroxytryptamine and finally to melatonin. And we know that serotonin is a very important neurotransmitter that regulates our mood. It can make us happy or it can make us depressed, and is a key neurotransmitter that's responsible for depression. And melatonin is really essential for our sleep cycles, and it's inadequate in the body that we cannot fall asleep and our body sleep cycle get disturbed. So for sensing of the tryptophan, I choose the surfactants. So why the surfactants? As we know that our human body is made up of 60% of the water and our brain is 73% of the water. So most of the measurement we need to do should be in the aqueous media. So my focus on the surfactant. As we know, the surfactant is like have a hydrophobic head and a hydrophobic tail and hydrophobic head. So it can be cationic like cetylpyridinium chloride. It can be anionic like sodium stearate, or it can be non-ionic like tritonex, or it can be lauryl amidopropyl betaine. It's a amphoteric. That surfactant can be classified on the basis of the charge they carry, and they can self-assemble in a different systems like micelles, liposome, and bilayer sheets. So my work is really inspired on the paper that published in OBC in 2013 from Mark Olson. So they synthesized this surfactant, and it is made up of bipyridine having a 11 carbon chain. And they found out when they add with this DNP tag, the template, the CMC, the critical micellar concentration of the surfactant decreased significantly. So I thought, like, if it's possible to modulate the CMC, critical micellar concentration, by adding the template, so maybe I can change a little bit of the structure so I can SAMP template the molecule. And we can see it in the this slide. So what I did, actually, I introduced one heterocycle between two bipyridine, and this heterocycles give a sufficient electron-rich environment to interact with the pyridine, that is the electron deficient because it's a positive charge over here, and they can act as a self-templated manner. So they can aggregate themselves more efficiently than the previous report. And the synthesis of this molecule is a bit challenging because we need to do some quantitative additions, and the purification of a charged molecule in normal column chromatography was not so easy. So we got the molecules. After this, we characterized this molecule by the X-ray crystallography, and luckily I got the crystal structure. It was not easy because we know that if we have a long chain in the molecule, so it can flank and it's not easy to set up in a crystal structure. But luckily we got the structure, and what I thought so far, it's really proven by the crystal structure, all the thiazole ring found to interact with the electron deficient pyridine molecule. And now I need to show this molecule should work to sensing of the tryptophan or melatonin. So in this slide, I'm explaining we did some conductivity measurement and we found out there is a sudden cross point here. We found the CMC of the molecule, synthesized molecule is 0.235 millimolar. And this is the 2.73 millimolar published in 2013 OBC paper. That our CMC is significantly decreased from the previous paper. That is significantly or importantly prove that our molecules is self-templated or the thiazole between the bipyridine helping the methylation. Then we add the tryptophan and melatonin, and then melatonin tryptophan. After the addition of melatonin tryptophan, there is a significant decrease in CMC was observed. This above 3 charts is like above 3 graphs is for the bromide and lower ones for the chloride. And we also repeat the experiment by measuring some variable surface tension plot by de novo rings, and we got the very concurrent result that we found in the conductivity measurement. We also measure the hydrodynamic distributions of the missiles, and we found when we add the tryptophan and melatonin in the surfactant, there is a significantly increase in the size of the missile. And the bromide size is significantly bigger than the chloride one. And we know that the bromide is a much bigger counterand than the chloride. After that, we did some photophysical properties. We found a very good, excellent quantum yield. It's approaching to the 0.9 in acetonitrile. In water, it gets quenched because of the dissolved oxygen in the water. And we got a significant, like good Stokes shift, 3663, with an excellent half-life. And then we can see here, after addition of a tryptophan and melatonin, in 2 vials, in the left-hand side, it is like after addition of, this is the example of only the tryptophan. If we see, we can see that there is a little bit change in color in a visible light. But when we irradiated the sample with 365 nanometer, we can see that there is a significant quenching of the fluorescence. So actually, we designed a surfactant that can sense the tryptophan and melatonin in an off manner. And to find out the mode of the quenching, we did some calculations. We determined the Stern-Walmer constant, and we found out at the higher temperature, 65 degrees centigrade, there is a significant decrease in the Stern-Walmer constant. That simply indicating, and the relationship is quite linear, that indicating that the quenching is not dynamic. It's a static quenching. If it is a dynamic one, so the plot should not be the linear, and it get deviated by increasing the temperature. It should get deviated. We also repeat the experiment with the chloride, and then we also also test the other counterand chloride. This time we increase the bromide one, and we repeat the same experiment to find out the mode of the quenching. And in the proton NMR, we found out there is a significantly upfield of the bipyridine proton was observed by addition of tryptophan and melatonin. That simply indicating the tryptophan and melatonin is in between the missile and interacting with the electron deficient bipyridine moieties. This is for the chloride one. And to check whether the system is capable enough for detecting the other aromatic amino acid, we repeat the experiment with phenylalanine and tyrosine, but we couldn't find any significant quenching in the efficiency. And right now, this assembly is under like evaluation as a thermal shift assay, because we know that when we increase the temperature, the proteins tends to defold, like they try to open up. When it's try to open up, there is some amino acids, and especially we're talking about the tryptophan. The tryptophan can expose to the outer surface rather than inner one. And by adding the surfactant, if the fluorescence get quenched, so we can measure the environment of the tryptophan in the protein. So right now we are analyzing the surfactants. It's a thermal shift assay for analyzing the structure of the protein. And except this, we also did the cyclic voltammetry. We also did the surface dynamic contact angles. We also did diffusion order spectroscopy. There is a lots of data we did to prove our theory, because the timing and loss of graph, I'm not not showing these things, but this is the recent work that I finished so far. And this is like a Fraser Stoddard, and here's this one is my ex-boss Mark Olson, and it's me. We met in China. And thank you all for listening. Any questions or comment?
Oh, thank you so much,
Thank you,
Dr.
ma'am.
Azad, for the excellent talk. It is open for discussion. If you have any question. Thank you, sir. If you have any question. Any question, please? It is open for discussion. If no question is there, then we can go ahead with our next speaker, Dr. Jishan Fatima. Dr. Jishan Fatima is working as an associate professor in Amity University, Lucknow. She completed her PhD from CSIR CDRI in 2007, and she published more than 20 research papers and review articles. And she has 2 international patents to her credit and filed 2 national patents in her credit. And she has been guiding many PhD scholars and master scholars. She obtained the Best Oral Presentation Award for paper entitled Docking Studies of Anti-Alzheimer Compounds in National Symposium on Integrating of Natural Healthcare Systems and Modern Science, Potential Merits and Future Roadmap, held on 2nd to 3rd November 2019, organized at the Bhim Rao Ambedkar University, Lucknow, and also achieved the second prize for the paper presentation entitled Database Management in ICMR and CSIR sponsored National Seminar on Biostatics and Research Methodologies, with an overview on clinical research, held on 12 to 13 February 2011 and PSIT campus, organized by Institute of Pharmacy, Ramveer Singh Institute of Technology, Kanpur. Now I invite Dr. Fatima for his talk.
Thank you very much, ma'am. A very good afternoon to you.
Thank
you. Can you hear me? Can you hear me? Am I audible?
As you can start, you can give your presentation.
Am I audible to you?
Yes, you are audible.
Okay, thank you. A very good afternoon to all the dignitaries present and the persons who are listening to this seminar. I'm really thankful to the organizer for giving me an opportunity to present my piece of work, the novel carbamates as a potential acetylcholine esterase inhibitors for Alzheimer's disease. Since very long, under the guidance of Dr. A.K. Saxena, who was my PhD guide, I've been working for Alzheimer's, and Alzheimer's is the biggest problem. The reason why the Alzheimer's is a problematic issue, because first of all, it is occurring in the older age, and since this particular disease is a progressive neurodegenerative disease, and in this disease, what happens actually, the person who himself is having a disease the life becomes so pathetic, because he forgets even the normal actual course of work, like brushing the teeth, going to the bed, and the person also forgets that what is the name of his near and dear ones. So in that case, it is a burden on the caregiver, the family members, and the patient itself. As per the estimated As per the estimate, it says that by the age of 65 in 2050, more than 16% of the people will be suffering from Alzheimer's. And every year it has been estimated that 10 in every 100,000 individuals, they develop dementia before the age of 65. Before the age of 65. The situation in India is better as compared to the world. And the reason that is cited by why this situation is better in India is that we being an Indian mostly have curcumin in our diet. And one of the mechanism which which is proposed for Alzheimer's control is an oxidative pathway. So if we have an antioxidant in our diet, maybe because of that, the Indian populations are not much more the Indian population, we're finding the cases lower than that of the Western countries. Now, if you see the development of Alzheimer's, basically find there are different strategies that there are different pathways, pathologies that have been mentioned, pathways that have been mentioned for the development of the Alzheimer's disease. And if you look at the brain of the healthy patient and the Alzheimer's patient, you'll find that there are neurofibrillary triangles and the amyloid plaques that are being developed. There is also, if you compare the brain of a healthy Alzheimer's patient with the brain of the healthy brain with an Alzheimer's patient, so you find that a lot more differences is there in the brain size. And the reason why these Alzheimer's is developing, we have different hypotheses. I won't be going into the detail of these hypotheses, just to mention to you that we have amyloid cascade hypothesis, which is a major hypothesis and which emphasizes upon that there is a formation of amyloid plaque. And this amyloid plaque is responsible for the development of Alzheimer's. Likewise, we have these are the amyloid plaque that is causing the Alzheimer's. Then the tau hypothesis is responsible for the development of tau protein. There's a hyperphosphorylation of the tau protein. Glutaminergic hypothesis, where it says that there is a deterioration of proper synaptic function through NMDA. Likewise, we have the mitochondrial hypothesis, neuroinflammatory hypothesis, oxidative hypothesis. And the last but not the least is the cholinergic hypothesis. So according to the cholinergic hypothesis, it says that the acetylcholine at the synaptic site, where you can see in the picture that this is the synapse where there is a production of acetylcholine, and the enzyme that is present is acetylcholine esterase. Once the acetylcholine is there and it is delivered as function, it is degraded by acetylcholine esterase enzyme. But what happens in an elderly patient? In elderly patient, exactly, this particular enzyme, acetylcholine esterase, when it is destroying the acetylcholine, so much of the acetylcholine is not available. So it does lead to the cognitive decline and the person is not able to remember. So there is a problem in there is a loss of cholinergic function. And when there is a loss of cholinergic function, the cholinergic function, the person cannot remember things. Now, exactly what is alarming is that this disease was particularly discovered in the year 1906 by Alois Alzheimer's. But currently, we just have only 3 FDA-approved drugs. The 3 FDA-approved drugs that are donazepam, rivastigmine, and galantamine that are being used for Alzheimer's. Recently, Mementi was also included. These 3 drugs that are available for Alzheimer's are your ACHE inhibitors. And they're only providing a symptomatic relief. By symptomatic relief, we mean that therapeutic relief is not coming. It's the symptom-based relief that is being provided to the patient. Recently, Aduhelm is a monoclonal antibodies that has been developed and it has been approved by FDA. But unfortunately, this particular Aduhelm that is being taken up, it is not devoid of side effects. And the side effects for this particular disease, for this particular drug includes, there's a swelling in the brain. Then there you have the tiny brain bleeds. There is headache, diarrhea, confusion. And last but not the least that I'll say is the high cost There's a very high cost that is associated with this particular drug. Thus, we cannot make this particular drug available to the common man. Now, to find the more to develop the more knowledge and the better understanding, and to develop more therapeutically useful molecules, I have taken up basically carbamates. The reason for developing these particular carbamates is, first we have to understand how it is binding. So in my previous work, which I had done at CDRI, I had performed the QACR studies on different sets of 77 molecules which I had taken into being, and from that knowledge, I had accumulated that if the molecule is to have an anti-Alzheimer's activity, the molecule should have the less molar refractivity, and the molecule should possesses the high hydrophobicity, because the hydrophobicity will be required for the molecule to get into the CNS. So this was the equation that was developed. This was already a published work, which I'm just showing it to you because I'm correlating it with my current work. On the basis of this development, we also came across that if the molecule is having an acridine nucleus, then it will be more active. And this acridine nucleus, we found it in the tacrine. That was the first drug that was approved for Alzheimer's. But unfortunately, it was removed because it was giving the hepatotoxicity. Now, from those understanding, we had already synthesized the carbamates molecule that was already patented in the CDRI. Now, moving ahead with that particular work only, though I had changed the nucleus and we have bring about the biosteric changes in the molecule. So I designed certain molecules based upon the drug rivastigmine. The structure is there in front of you. Now, based upon this rivastigmine, the molecules were designed and they were subjected to docking analysis. Okay, because what I wanted to see is that if we first dock the molecule to the active site, at least we will be able to know what is the docking score and how the molecule is docking. So we were lucky enough. This is a rivastigmine, and as you can see, the docking score that we obtained for this particular molecule, rivastigmine, in the active site was minus 4.985. Similarly, on this basis, we first docked the designed molecule. So we had designed the molecules, and then we docked them to the active site of the receptor. So after docking them to the active site of the receptor, we found that these molecules were having the better scores as compared to your rivastigmine, that is a drug which is available in the market. Now, the question arises that, okay, if these molecules, so these are the binding, you can see over here, the binding pattern of these molecules, the binding scores were generated, and these are the interactions you can find in the molecule. See, we found that EK13, this is the structure of the molecule. So EK13 was having more interaction. As you can see over here, we have the pi-pi, we have the hydrogen bonding interaction, then we have the pi-cation interactions that were available as compared to rivastigmine, where the interactions were low. So due to this reason, we found out, okay, that because of these interactions, the docking score of rivastigmine we found was minus 4.985 as compared to EK18, which was minus 8.56. Now this forced us to synthesize these molecules, and theoretically, we can always say that our molecules are better than the drug that is there that is already existing. But practically, we have to prove that the molecule which we have in hand is which we are designing is better than the drug which is already there. But unfortunately, we performed the acetylcholinesterase inhibitory activity for such molecules. It was just an in vitro study that we had done. We had replicated the experiments in 3, and then we found out that these are the results of my activity, where we found out, okay, that EK13, here you can see this is EK13, it was having the activity. It was having the activity, but unfortunately, the activity was not more than the drug that is actually there, which is rivastigmine. So theoretically, it is very good that we can predict, but until and unless we have the practical aspect of it, we cannot say whether the molecule will be active or not active. From this experiment, what we have learned is, here you can see that the IC50 value of this particular molecule was found to be 27.15 micromolar as compared to rivastigmine, which was 3.61 micromolar. Okay, but if I see the binding score of rivastigmine was less as compared to this particular molecule. So the binding score was better. Now, as far as my understanding of this particular thing is compared, what I can say is that we have got a scaffold, and definitely in future we are trying to optimize this molecule. We're trying to develop more SCR regarding this molecule, so just in case we can come across a new potent and more effective molecules. With these words, I would like to wind up my talk. I really thanks the organizer for giving me an opportunity to present this small piece of work over here. And I'm really grateful to my guide, to my mentor, Dr. A.K. Saxena. Once again, I wish him a happy belated birthday, sir. I cannot connect you yesterday as you were there in the symposium, so I just could deliver you a message. Thank you so much. Thank you so much, Medulama. Thank you so much. And the organizers.
Thank you so much, Dishan Fatima, for excellent talk. It is open for discussion. If any question is there. Thank you. Thank you. Thank you. Now it's open for discussion now. Okay. I invite first Dr. Ashish Nandi to accept the memento for his deliberation for the excellent talk. आए तो ये दूं आपको
I think he accepts. I think his photo is not there. So thank you very much, Shishi.
for Dr. Ashish Nandi.
Dr. Ashish Nandi. Thank you very much.
Next is
Now, yeah, Shishi Nandi. Yeah. Now, Shishi Nandi, please come. He's not visible again. Okay.
No.
So, but he is here in front of us. So, thank you very much. I think it was a very nice presentation. And thank you very much for this.
I have accepted the memento from the core of my heart.
Thank you. Thank you. Thank you.
And now we invite Azad.
Dr. Azad.
Dr. Azad. Yeah, maybe that is some problem in visibility, but the memento is here.
No, they will display. No.
Yeah, Azad, I am very happy that now you are working there and where you wanted, and your work was really very, very fascinating. And I think you're using your skills of organic chemistry synthesis So I wish you all very good luck and thank you again very much for participating in this. Thank you. Accept this. And now we
Right,
invite
Dr. Jishan
Ishan
Fatima.
Fatima. Yeah, she is visible here. And thank you so much, Ishan, for your message and your participation in this. And please accept this memento to you.
Thank you so much, sir. Thank you.
Thank you. Now we have to present memento to the chairman.
Chairperson.
The memento.
करेंगे ना.
Yeah, this is memento is here.
Yeah.
And thank you very much. Okay.
Thank you so much for the token and giving me this opportunity to preside this session. Thank you so much. I accept the token. Oh, thank you.
Hello. Now this is the lunch break and followed by poster session. The lunch break would be up to 2:30. No, sorry. Yes, up to 14:30. So lunch break is up to 2:30. All the delegates are requested to enjoy the lunch followed by the poster session. The poster presenters, must be present in front of their corresponding posters where you have pasted at the specified location, because two evaluators, Professor Kopil Kumar and Assistant Professor Ankita Sharma, two chairperson, two evaluators should be there. So they will visit to evaluate your poster. All the poster presenters, they must be present in their particular location in front of their posters. After completion of the lunch and poster session, the last scientific session would be started at 14:30. Honorable Chairperson is Professor A.K. Saxena and invited speaker Dr. Marian Rajko and Professor Kunal Rai. So be particular about the time, although we are late by half an hour, but please manage and cooperate. Thank you very much.
Thank you very much for introducing, and I'm very happy to chair this session, particularly in which the speakers are very well known to me, very good friends of mine. And first I introduce Dr. Marian Racco. He is a senior researcher at the Union Institute of National Institute of Chemistry in Ljubljana, Slovenia since 1994. His research is focused on QSAR and chemometrics in the analysis of proteomics and genomic data, and to modeling of interactions between receptors and molecules. I think this is what the drug design is important for. He had obtained his PhD degree in 1990 from University of Erlangen, just again from Germany, and particularly his subject was quantum chemistry. Thereafter, he did his postdoc from University of Nanu. And in 1994, he joined the National Institute of Chemistry in Ljubljana, and where he's continuing one way. And he has got a lot of papers, I think, and I have interacted with him. I have visited Slovenia, Ljubljana. I think I met Shishin Nandi there. Then since then, we have a very good friendship. He is a very permanent member of CMTPI organizing, International Organizing Committee. So nothing more to say, and certainly I'm so thankful to him that he has chosen this. He's one of our Jibbar family. He had been here also in 2020. So with these words, I think I'll request Marian Racco to deliver his talk.
Hello, do you hear me? Can everybody hear me? Dear Professor Saxena, thank you very much for this cut introduction. I would like to thank to give me the opportunity for this talk, and I would like to congratulate the organizers for a very, very excellent organization. Do you hear me?
Share the screen.
Okay. Can I now?
Yeah, you can start. You can share the screen. You are audible.
Okay.
I have to close my screen.
I don't know how is this. My screen sharing. Can you see this, my screen now or not?
Yes. If you can make that PowerPoint is fully screen, that would be nice. PowerPoint, you can come
okay.
down. Yeah.
Here?
Yes. I think it should be okay with this.
Okay,
yeah.
It's good.
Yeah. Fantastic. Good.
I think okay. Oh, I'm sorry. I'm coming from National Institute of Chemistry. from the theory department and actually laboratory of chemometrics. These are our team, and we have also quite a good collaboration, and most of the results which I will show here come from our collaboration with Professor Subhash Basak and Professor also Nandi. He should be here, but I don't know why okay, Professor Ashish Nandi from Kolkata. So what is here is the outline of my talk. I would like to present some chemometrical approach, some chemometrical things, how to treat the data and how to get information from the data. And at the end, I will show some examples how we treat the chemical databases. So this is what we know, QSR modeling, or how we treat nucleotide databases. This was done with a collaboration with Professor Basak and Professor Nandi. And how can we treat the proteomics data? This was done with Professor Basak and Professor Witzman from Indiana University in USA. Here are some books. I will share later this information. If somebody finds interesting, can have a look into this book. But now, today we live in the world of big data. What actually means big data? When we have a huge number of data, for example, by high throughput screening of large chemical databases. So we have data for over 10,000 molecules. Then, of course, genomics, we have up to billion sequences for genome. And the proteomics, of course, we have information for over 1,000 proteins in what we get in every measurement, in each measurement. And of course, the proteins are large molecules. They have 10,000 of atoms. And so to handle all of these data needs very specific computational tools. On the other hand, we have what we can say a small data. This is the classical toxicological data. We have databases with up to hundreds or up to 1,000 compounds. And we have actually quite a limited data about the in vivo data or clinical and epidemical data from clinical and epidemical study. And our task is, of course, to integrate the big and the small data to use the information getting from these big data methods and try to find some explanation or in this area of practical toxicology. Of course, practical task is how to compress this data. I will show, I will present two methods, PCA, Mahalanobis distance, or even compressing of data into a single index and so on. But let me go now to the new or concept in toxicology. This is maybe a decade old, that if we understand a toxic effect of a molecule, we have to solve this puzzle. It is called adverse outcome pathway. And this is a concept, actually. AOP is a concept when final toxic effect is a consequence of linked events. And these events, of course, occur on the molecular level, goes over organelle, cellular tissue, organ, organism, and to the population. So, and if we understand now entirely the toxic effect, we must understand all of these steps. And of course, in silico methods, computational methods, chemometrical methods play a crucial role in solving of this puzzle. But if I go back, of course, we start with our databases or with our data. The first question is how we represent the data. We must represent the data in a suitable form, which is, of course, understandable for computers. And it was maybe shown 50 years ago when we started with this question. Of course, such a representation should be uniform, unique, and reversible, but no representation fulfills all those requirements. Uniform means that the dimension of our representation is the same for all our objects. Unique means that if two objects are different, of course, the representations are different, and reversible means we can construct our objects back from the representation. But this is quite a serious task, and actually very few representation fulfill the reversibility. What are now practical examples? For example, we're in the QSR modeling, on the computational modeling, we represent our molecules with structural descriptors. This is the representation of a molecular structure. I will show later a small example when 300 molecules are represented with more than 4,000 descriptors. So it means it's quite a huge amount of data. Or we can represent other example, we represent the virus genomic sequences with different representations, like a neighboring matrices. Then Professor Randic and Professor Basak worked a lot about this graphical representation of genomic data, or Professor Nandi also worked in this area with he introduced like an index Nandi descriptor or so on. And of course, we can represent the proteomic data with some similarity index. And of course, these are 3 examples, but any problem requires actually its own thinking about the representation. Now, if we solve this problem of representation and we calculate representation vectors for our objects, then of course we put all of our objects in a matrix, and this is data matrix, which is starting point of any chemometrical or computational analysis. We can do a lot of now algebraic operations with this matrix, like calculating the average values, or what is very important to calculate the variance and covariance metrics. This somehow measure the dependence of our variables. For example, the covariance matrix is a basis for numerous chemometrical methods, like a principal component analysis, Mahalanobis distance calculation, and so on. Let me start now to the second point. I would like to present 3 chemometrical methods for analysis or our data metrics or our data. But first of such interesting point are distances. Euclidean Manhattan, I won't talk about this because it's a straightforward. But what is very interesting is Mahalanobis distance. I digged a little bit to this history, and here is the intuitive picture, what we are actually, what is Mahalanobis distance. If we have a set of data, let me see here in this ellipse, in this circle, and then we put to this set another point, like this X, and then is a question, is this point belong to this data set or not? If we just calculate the distance, you see, we could say yes, but somehow we see that this is out of this ellipse. And Mahalanobis distance gives here an answer. Actually, how come to this approach? Actually, it starts with 1856 when German mathematician Riemann introduced non-Euclidean geometry, and actually later on Albert Einstein applied these principles in non-Euclidean geometry in general theory or relativity. The space is, according to this theory, not Euclidean, but curved, and the light does not travel along straight lines, rather along curves. And this is because of gravitation of terrestrial bodies. And 63, applied Indian statisticians Prashanta Chandra Mahalanobis, the same principles of non-Euclidean geometry in statistics. And he also, in this original paper, wrote about the Einstein general gravitation theory. So it's quite interesting paper, but maybe not very easy to read. And the basic is, of course, the covariance matrix, or more specific, the inverse of the inverse matrix, and we can calculate distances exactly the same as in special theory of Einstein, the distances between points. And let me see now a very simple example. If we now consider those blue spots and we calculate the Mahalanobis metric for those points, and then we calculate the distances of this red circle, so we get that points perpendicular to this are on the has a much larger Mahalanobis distances as the points which are along this along these points, and we could say that those points are out of our data set, because they are on the very large distance, but they in the metric in the Euclidean metrics, they are on the same distance. Now, the second method is the principal component analysis. I maybe won't go here in the details. It is just the transformation from old coordinates to new coordinates in such a way that we preserve the entire informational content of our data system. And of course, here also a basis is covariance matrix. We have to solve some algebraic problems with covariance matrix, get the eigenvectors and eigenvalues, and those eigenvectors are our new principal components. And to each principal component, we get also information how much of the total variance this new principal component carries. This is with eigenvectors. And so what is in the end of our transformation from new variables, we can actually reduce our system to a handful of new variables and remove all others. And those new variables carry entire information of our system. Maybe I will show this later on the example. And third point is the counter-propagation artificial neural networks or Cohone networks. This is also a mathematical tool to project a multidimensional spaces into two-dimensional maps in such a way that somehow similar objects are located here in this network close to each other. So in the end, we get two-dimensional maps, and each in each square here carry one or more our objects, and here is some property colored, so we can see how those objects are organized in two-dimensional map. Now, let me go now to our first example. We treated solvent databases with 235 compounds, and each compound were represented with over 4,000 Dragon descriptors, and we did some study connected to the developmental toxicity of those compounds. We would like to implement a clustering scheme for those compounds, considering their developmental or neurotoxic properties. And first, what we did is the PCA of our 4,000 dimensional space with 4,000 descriptors, and we got a very actually interesting results there. Actually, the first one principal component carries over 85% of the total variance. So if we take 5 or 10 these compounds, these descriptors, new principal components, we get entire information of our 4,000 descriptors which were calculated with Dragon. So it's quite a drastic reduction of dimension. And so here is to see these are two principal components, and so are our solvents now presented in a two dimensional space, and entire variance is practically entire variance of our data is included in these two principal components. Actually, we took now the 20 of first principal components and make a Cohone network and get such a colored map. These areas, red, are mostly populated with toxic compounds, and this green area is a non-toxic, and these are blue dots are the components which are with known toxicological data. These are actually toxic compounds. The results are not very actually very good. The compounds are quite scattered across the map, and we can only indicate some small clusters within those data set. Another example was a set of conazoles. Conazoles, these are chemicals which are used in agriculture, human veterinary medicine, and also in human medicine. And they are quite strongly regulated by all authorities. And we also performed a similar study, calculated descriptors, made the PCA. We see that with a handful of principal components, we can describe entire information of the system. So these are our principal component plots, and this is our, at the end, our Kohone network plot with these conazoles. Let me show our second example was in a cooperation with Professor Basak and Nandi. We cluster Zika virus sequences originating from different geographical regions, and we use the different computational and sequence descriptors. So what is our strategy? Collect the virus sequences, calculate descriptors, carry out PCA Mahalanobis distance analysis, and also using self-organizing map technology. So these are our data, over 310 samples from different countries, Asia, South America, Africa. And what we did with those data, we calculated different sequence descriptors, like a Nandi's descriptors, and then count of triplets, count of neighbors, and trying to represent these entire viruses of this kind. And then we performed all of those analyses. Here is an example of Mahalanobis distance. For example, there are 37 African samples, and they have a quite larger Mahalanobis distance in comparison to Asia and South America. And in both cases, in any kind of descriptors we take, the samples of Africa are far apart from Asia and South America samples. But here is also, we can indicate some particular outliers in all of these cases, and this is quite precisely described in this reference. Here is also a PCA picture of those data sets. So we see that the Africa samples are separated, but they are Asia and South America quite on the same place, and there are some outliers. And also the same analysis we did with a self-organizing map model. And the last example I will show is the proteomics data. This was done with a collaboration with Professor Batsak and Professor Witzman from Indiana University. And of course, the proteomic studies are quite a complex study. We have to separate the proteins. We measure the protein status of cells, which are sometimes treated and sometimes not treated. And then, of course, we have to compare those data. Here we have quite a complex set. There were three types of cells. And they were treated with two kinds of nanomaterials, multi-wave carbon nanotubes and titanium dioxide nanobelts. And they were treated under different experimental conditions, like different time of exposure or different concentration. So we have quite a lot of independent variables, and we made some analysis of data, of those data, using the similarity index, which directly compare the abundance of control samples and the treated samples. And we analyze those data on different ways, and actually with all methods, it turned out that the samples are with are well separated, which were treated with after 24 hours. So the time of exposure, we have two times of exposure, 3 hours from 24 hours, but this time of exposure is most important feature in the separation or in the clustering of those data. This study was actually extended with silver nanoparticles, and which were also treated in 3 hours and 24 hours, but those data were not completely comparable with our previous data. So we did some analysis which proteins could be could play the most important role in exposure to silver nanoparticles. We collected some data from literature, what was known about the silver nanoparticles, and here are some proteins which could be assigned to maybe to silver nanoparticle exposure. Okay, but in general, in comparison, we can also perform very general comparison between silver nanoparticles and multi-wave carbon nanotubes. Here is the average of similarity index. We can see that the similarity index is lower for silver nanoparticles in comparison to other particles. And this is the red columns here are the percentage of the highly disturbed proteins. And we can see that in the silver nanoparticles, there are much more proteins highly affected in comparison to other type of nanoparticles. These are some additional chemometrical methods, and also in comparison to previous study, no evident separation between exposure to 3 or 24 hours. And we can also know specific groups of proteins can be identified in this level of study. So this would be everything from here. Thank you for your attention, and I would thank again the organizers for this invitation.
Thank you very much, Maya and Rako, for a very excellent presentation, that too very much keeping in time. And I think let us give a clap for him.
Okay, thank you.
You have very nicely judged with that data handling, which is very important nowadays, because the problem is people are facing, as let's say, that the number of data set is less and parameters are far more. So the principal component analysis application is really very useful. if there are any questions, it would be nice, and Professor Racco will answer those, but I don't see any questions so far. This chat box is concerned for question answer, but from the audience or even who is wants to ask the question. So if there are no more questions, I thank you, Professor Racco, for your presentation, and very important actually what you have presented, particularly handling with proteomics data is far more difficult, but at least I am myself has used it so far as small molecules are concerned and the parameters. So I think variable selection, this is a very important aspect of physical component analysis. So thank you very much. Thank you very much for participating at this occasion. Thank you very much.
Thank you.
The next speaker for this session is Professor Kunal Roy, who has been the ex-head of the Department of Pharmaceutical Technology, Jadavpur University, Jadavpur. You know, particularly in this, so far this area of drug design is concerned, I think Kunal Roy is pioneering in this field, and he doesn't need in one way introduction, but still he's has a lot of things to his credit. Recipient of Commonwealth Academic Staff Fellowship he got in 2007, then Marie Curie International Incoming Fellowship in 2013. His field of QSR, molecular modeling application in drug design, property modeling, and particularly he has been very much popular, or he's working on toxicity data, and he has got very high index, H index 45, and the citations which are more than 10,000. He's a co-editor and editor-in-chief. Co-editor-in-chief in Molecular Diversity and editor-in-chief International Journal of Quantitative Structure Activity Relationships. His papers have been highly cited, and one of the most important, which I found last year, when Stanford list of top 2% scientists of the world was there, he was holding I think the first rank in India, but I think he has got a, maybe, I don't know, he's saying what is his rank in this year, but his world rank is 87 in the subfield of medicine, biology, molecular chemistry. So first of all, congratulations, Kunal, for all this. I'm very happy that you are coming up a rising star of this India in this field. With these words, I will request you to kindly give your lecture.
Thank you, Dr. Saxena. Can you hear me?
Yes, yes.
Okay.
Presentation is also very much. Yeah.
Okay. Thank you, and very good afternoon to all. Today, my topic is a new similarity-based read-across algorithm for the prediction of small data sets, case studies with nanotoxicity data. So, in fact, I usually speak on QSR, but today I will not speak on QSR. Today I will speak on read-across, which we can use for small data sets is using a similarity-based approach. So QSR is basically a statistical approach which can be applied when we have sufficient number of data points available. But in case we do not have sufficient number of data points, then getting statistically reliable models and to validate that model using another set of data becomes difficult. So in these cases, we can use similarity-based approach of read-across, and this is also acceptable from regulatory perspective by various organizations like ECHA, OECD, US EPA, and all. Okay, and here as case study, we are discussing about nanoparticles. As we all know that nanoparticles are very interesting class of materials, and these are being used in various fields due to their interesting properties like high surface area to volume ratio, high reactivity, specific physiochemical properties, and etc. And we find their application in pharmaceutical industries, solar cells, ceramic industries, textile industries, food processing industry, and so on. And these nanomaterials can be accumulated inside cells, and they can cause toxicity. They can cause membrane damage. They can release toxic ions, impairing enzyme function, and they can generate reactive oxygen species, which can in turn damage protein and nucleic acid, giving rise to various kind of events which ultimately can kill the cells. And we have very limited toxicity data available for nanomaterials. Usually the toxicity data points for nanoparticles will be very limited, of the range of 18 to 20, and it becomes very difficult to generate statistically validated QSR models with such small data sets. And on the other hand, we need to generate data. We need to bridge the data gaps because we do not have sufficient toxicity data of various possible nanoparticles on various endpoints, not only for humans, but also from ecotoxicological point of view. We know that there are several endpoints for aquatic toxicity, for terrestrial toxicity, and we have hundreds of endpoints, but we have limited toxicity data for these nanoparticles. So we have to assess their toxicity potential, both for humans and also for the ecosystem. And we need to bridge the data gaps using some model-derived predictions. Among nanoparticles, we have two major groups. One is carbon nanoparticles, and the second group is metal oxide nanoparticles. Metal oxide nanoparticles have diverse applications such as cosmetics, textiles, paints, water treatment agents, solar batteries, and automobile catalytic converters. But in spite of their widespread use, it has been seen they show significant toxicity in humans, also causing respiratory, cardiovascular, inflammatory diseases. So we need to generate more data before their safe application for various uses that we can use in our daily life or day-to-day life. For example, say as a sunscreen, when we use metal oxide nanoparticles in cosmetics, so we are directly exposed to these nanoparticles, and we need to know what is the toxicity potential. And at the same time, evaluation of toxicity of these nanoparticles may require use of some animals which will have ethical aspect. For example, say in European Union, it is banned to do animal experimentation for development of new cosmetic ingredients. So we cannot use animal data for development of safe cosmetic ingredients. So we have to rely on the model-derived predictions. So this slide shows in a graphical way the representation of toxic effects of metal oxide on E. coli cytotoxicity, showing that metal oxide nanoparticles are involved in disruption of electron transport chain. They are involved in membrane damage. They release free electrons, attacking proteins and nucleic acids. They block signal transduction, etc. Now, metal oxide nanoparticles can venture human system through breathing, gastrointestinal tract, skin, and then they can circulate via blood and lymphatic system. And because of generation of reactive oxygen species, they cause mitochondrial damage. And considering the complexity of nanoparticles and their application to mankind, we need to have the data about their toxicity. And as I've already mentioned, we have access to a limited number of toxicity data of metal oxide nanoparticles. So we need to generate data from alternative approaches, including computational methods, which involve QSR and other computational methods. Now, these computational approaches are time and cost effective. They support 3R principles: replacement, reduction, and refinement of animal experimentation. And they can be used for virtual compounds. Even having the compounds physically, we may screen some virtual library. And based on that, we can prioritize our experiments. We can avoid involvement of cost for doing experiment with less promising compounds. We'll invest only for those compounds which are more promising and which have potential to show less toxicity. And these methods are also supported by various organizations like European Center for Validation of Alternative Methods, International Organization of Medical Sciences, EU REACH regulation, US EPA, and OECD. Now, I've already mentioned that QSR is a statistical based approach, which is suitable for moderate size or large size database. But if the data size is very small, then it becomes difficult for us to apply QSR, because to develop a statistically validated model and statistically robust model, we should have sufficient number of data points. But in case we do not have sufficient number of data points, what strategy will follow? We must have some data generated using modeling approach, but in case we are unable to apply QSR, then alternatively, we can use category-based approach, which is called read-across. Read-across is accepted by REACH and US EPA. It can be used for data gap filling. In case of read-across, we should have two groups of compounds. One group is called source compounds, and another group is called query or target compounds. And then based on the similarity, we can predict the response or activity of the target compounds from the source compounds. So it can be of two types. One is analog approach and the other is category approach. In case we have limited number of source compounds, then we'll use analog compounds. And in case we have sufficient number of compounds, we will use category approach. So it may be say one-to-one type, one-to-many type, many-to-one type, or many-to-many type. In case of read-across, we can apply both interpolation and extrapolation. As you know, interpolation means the target compounds is in the property space which is within the source compound, but in case of extrapolation, it is beyond that. Now the question is, what is similarity? We represent a chemical compound structure using various structural representation in graphical way, but the graph does not allow us to do numerical manipulation. So we have to convert the information that is present into in the chemical structure into numerical form, and we can generate different descriptors. And these descriptors may be of different categories like 0D, 1D, 2D, 3D, and so on. So 0D may be say some constitutional descriptors, 1D, so maybe fingerprint, 2D, the graph theoretical descriptors, 3D, which involves the 3D distance and bond angle, torsion angle, etc. In case of 4D, we also include the conformational aspects. Also, we can go further in 5D, 6D, and 7D. In any case, when we have the chemical structure information presented in numerical form, then we can apply the feature selection strategies to understand which descriptors are important for our particular response endpoint. And then we apply appropriate regression or classification-based strategies to develop our model, which are then tested for their robustness and acceptability using various statistical techniques. And also they're applied to a separate data set to know their applicability for new data set in a procedure that is called validation. So say these are the structures of some of the simple compounds like ethylbenzene, nitrobenzene, chlorobenzene, etc. But these are pictures to us. We cannot do any kind of numerical manipulations. So you have to convert the information present in this structure into numbers. For example, say how many carbon atoms are present in each structure, how many nitrogen atoms are present. So these are very basic level of information. These are 0D information. But we may have some higher level of information also where we use say 2D representation of graph and can derive different kind of matrices like adjacency and distance-based matrices. And we can derive several graph theoretical descriptors, or we can consider their 3D geometry and derive different 3D descriptors, or we can do some quantum chemical treatment. We can we can get their different property values after consideration of the structure at the quantum chemical level. So there are different level of calculation of descriptors, and we can get information about molecular structure in different levels of theory. But in a nutshell, that descriptors are only numbers. They are numbers which depict information about molecular structure or their property. And we have to pre-process this descriptor matrix to get rid of redundant descriptor columns. We have to avoid the intercorrelated descriptors, noisy variables, and so on. And after reduction of the size of the descriptor matrix, we will apply appropriate feature selection technique to know which features are important for our particular endpoint. And then we apply appropriate regression or classification-based strategies. In case we have our response data in the continuous scale, we'll use regression-based strategies. But if we have our response in graded values, then we'll apply classification strategies. And finally, we will get some model. In most of the cases, it will be in the form of some equations. But if we are applying some machine learning technique, we will not get any equation, but we will get some calculated or fitted values. And in case of the query compounds, we will get predicted values. And then we can compare the predicted values against the experimental ones. In case of test set, which we do not use during model generation, they are unseen to the model. So model has not been derived from the test set information. So test set is new to the model, but test set can be used to predict the response values for the test set. And in that way, we can judge how good or bad the model is during prediction for the new compounds. And that we can judge again using some metrics. There are different kind of metrics for external validation, like U-score external F1, F2, and so on. But we again come back to similarity, because we are discussing about read across, and read across is a similarity-based approach. Now it may be of two types. Say, for example, we have one compound for which activity is known here, and another compound, the activity is known here. Actually, the change of slide is taking some time, although yes, now it has changed. So say this is one compound and this is another compound for which we know the response values. And this is a compound for which we know the structure, but its response value is unknown to us. So if you want to know this in an interpolation method, we can know this. You can see we have derived this the slope of the line from these points, and then we can get the predicted value of the response point, knowing the slope of the line and the descriptor that has been used to derive the similarity. So here we are using only one descriptor. Again, here in this case, our unknown compound or compound with unknown response is here, but our two other compounds for which we know their response, they are here. So this is extrapolation technique. We are using these two compounds to know the slope, and then we are using the closer compound to know the predicted value of the response using the slope that has been calculated by two of the data points. But here we are using only one descriptor, or one feature rather. But when we have more number of features, then what we do? So that has been already explained by the previous speaker, Professor Mariam Kratko, that we can use a procedure called principal component analysis, which is a data reduction technique where we can reduce the dimensionality of our descriptor matrix to a lower dimension. And the first principal components will have a higher variance, explained variance. So we can take those two principal components. As you can see, we have plotted PC1 versus PC2, and in this principal component space, the data points are plotted, and you can see some clusters. So this cluster shows us that which compound is similar to which compound. So based on the similarity, we can then select the compounds based on which we will predict the endpoint value. And then there is another method that is called self-organizing map, which is an unsupervised method of training. So here we can use neighborhood kernel, and it is not unlike the ANN, which is a supervised method. We are not using back propagation here, but the data points are maybe that is projected in the surface where we can find the closer compounds. We can identify the closer compounds. So in read-across, actually, we identify the closer compounds, and based on the endpoint value, we can predict the endpoint value for our target compound. So this is the basic theory. Now, what about QSR? QSR is a statistical method where we correlate the biological activity of chemicals using descriptors, which are representative of molecular structure and or property using statistical tool. So in the very basic kind of QSR, we get multiple linear regression type of model. So this is the model that we can see here. You will see in the left-hand side, there's a Y value, that is the endpoint or responding model or dependent variable. In the right-hand side, you have X1, X2, X3. These are the independent variables or descriptors by which we will model the endpoint. And A1, A2, A3, these are called regression coefficient, and A0 is the intercept value. And when you know this equation, you will be able to predict the endpoint value of any new compound by just putting the descriptor values X1, X2, X3, because these are obtained from chemical structure or some of their properties. So you will be able to calculate or predict. Then you can plot the observed versus predicted endpoint values. And if you have good quality of prediction, you will see the low degree of scatter. So the points will be close to the 45-degree angle line. So that is the basic notion of any QSR. But to derive this model, to derive this equation, you should have sufficient number of data points. So usually for multiple linear regression, there should be 5:1 ratio maintained at the very lower end. That means for each descriptor, there should be at least 5 number of data points, but that is a very basic requirement. You should actually have more number of data points. So if we have, say, 50 data points, you cannot use the descriptors more than, say, 5, 6, or 7. But in that case, also you have to check for interpolation among the descriptors. So you can then go for a more robust technique like partial least squares, which can convert the original set of descriptors into latent variables, and in that way you can reduce the number of descriptors that are being used in the final model generation, because number of latent variables will be always less than the number of the original number of variables. So you can, in that way, reduce the noise from the system, and you can get the more robust model. But in case, if we have very low number of the data points, say you have only 15, 20 data, but the endpoint is very important for us, say toxicity of nanomaterials or some catalysis endpoint. So in those cases, it is very important to know those endpoint values for new compounds. It is very important to predict. And if you go for a synthesis of those materials and go for experimental testing, that takes time and involves resources also. So we have to prioritize our understanding of toxicity of those compounds. In that case, it is very essential to know that the probable toxicity values for those data points. So we have to apply some kind of modeling technique. And as I've told that we have limited number of data points, becomes difficult for us to apply QSR, because in case of QSR, usually what we do, we divide the data into training and test for validation purpose. So I'll come to that, but before that, any regression-based model that we have obtained is usually judged for their statistical quality in terms of various metrics like determination coefficient, explained variance, variance ratio, and standard of estimate, and so on. I will not go into details about this because we have limited time. And for validation of models, we have to apply internal validation in leave one out or leave many out approaches, where this cross-validated R-square or Q-square values are taken into account, and the value of which should be more than 0.6. And for external validation, usually we divide the data set into 2 parts, training and test. And test set is kept aside, which is never used for model development. So this is used only for validation purpose from the metric value. And there are other methods of validation also, like bootstrapping and wide randomization. So we start with the data set of chemicals with definite endpoint value. And for biological QSR, we usually convert to the logarithmic conversion, the response values. And we draw the chemical structure, compute descriptors, and from the QSR table, And we do our data pre-treatment by removal of interpolated descriptors and so on. Then we divide the data into training and test. Training set is used for model development using the usual procedure of feature selection and then classification or regression. And then the developed model is applied on the test data to know the quality of prediction using various validation metrics. Now the question is this division of data set into training and test. If we have a limited number of data points, it becomes very difficult for us to divide the data into training and test. So what we do in such cases, we can use a read across, because read across is not a statistical model building technique. We can use this for small data set, and we do not require many number of data for the prediction purpose. So here calculation is comparably easier than QSR, and this is the alternative tool for hazard assessment, and it's aimed at filling data gaps. And as I mentioned, for nanotoxicity, we usually have a limited number of data available, so we can apply read across for toxicity prediction of nanotoxicity. So objective of this particular work that I am presenting here is to develop an easier and efficient method for quantitative read across predictions. Quantitative toxicity predictions using various small data sets, here specifically metal oxide nanoparticles. And we'll compare the results with some previous methods. And we also aim to develop an application, web-based application, which can be used by others for their prediction purpose. So here we have used 3 different data sets, all are nanotoxicity data sets. The first data set is for metal oxide nanoparticles against human keratinocyte cell line. Second one is metal oxide nanoparticle data against Escherichia coli, and third one is metal oxide nanotoxicity against Escherichia coli in dark condition. And here we have used the limited number of descriptors as reported in the previous reported papers, because we need some features to know what is the basis of similarity. So for example, in the first case, we have used electronegativity of the cluster and enthalpy of formation of metal oxide. In the second case, we have used enthalpy of formation and charge of the metal cation. In the third case, we have used enthalpy of formation and absolute electronegativity of the metal oxide. Now, as I mentioned, that this is a similarity-based method, and to show to the audience that the method works, so we need at least 2 sets of data. One set will be used for source, or they are used as source, or you can term them training compounds. Although the term training is not appropriate here, source compounds are more appropriate. And second set is target sets, for which you need the prediction. And similarity here, we have calculated using 3 methods: Gaussian kernel, Laplacian kernel, and the Euclidean distance-based similarity. And we have computed toxicity using weighted average predictions using the formula that is shown here. You can see the formula using this weighted average prediction. We have got the weighted average prediction. And what we have done, we have given weightage to the closed training compounds based on the similarity to the test compounds. And then we have sorted the training compounds in descending order, and we have selected up to 10 most similar training compounds. I will come into details, but first let me show the different kernels that we have used. The first kernel we have used, Gaussian kernel, which is based on this formula. The formula, I think, has not come to your screen till now. It is coming because it's taking some time to change the slides over Zoom. The next slide will come. Yes, it has come. And you can see that this is the formula. And this is basically the Euclidean distance between the 2 vectors. So the x and y are the 2 vectors with these elements there. And this is the usual Euclidean distance, so L2 norm. And sigma is the hyperparameter here, which is to be optimized. I have used different values of this hyperparameter: 0.25, 0.5, 0.75, 1, 1.5, and 2. The second kernel function is Laplacian kernel function, which is of this formula. The formula will come soon on your screen. And here we have used Manhattan distance, which is actually L1 norm. So x and y are 2 vectors and of length n each. And we calculate the L1 norm, and this is the formula, as you can see now. And this is your Laplacian kernel function, and gamma is a variable number or hyperparameter here, which is to be optimized. We have used different values like 0.25, 0.5, 0.75, 1, 1.5, and 2. And third method is Euclidean-based similarity approach. What we have done here, we have done prior scaling of the descriptive values for the both target compounds and source compounds using the formula, the original descriptive value minus mean of the source compounds divided by standard deviation of the source compound. Using this formula, we have scaled them, and then we have calculated Euclidean distance for individual test compound or individual target compound and source compound. And then we have converted this calculated ED into the scale of 0 to 1. We have rescaled them, and then we have calculated 1 minus Euclidean distance, that is the measure of similarity. So we have used this similarity along with other two kernel function derived similarity, that is Gaussian and Laplacian kernel function derived similarity. And once we get prediction for the target compounds, then we have like our QSR derived prediction in case of target set or test set, we have observed values known for us and prediction value known for us. So then we can apply our usual external validation metric like Q square F1, Q square F2 as we use in QSR. So this part is statistical, but the previous part, that is the derivation of the predicted value of the target compound, that part is not statistical. So this evaluation part of Q square F1, Q square F2 and RMSE, this part is using usual statistical formula for calculation of different validation metrics. And also we have additionally done classification based analysis, because sometimes it may so happen that we get prediction, but the quantitative prediction may not be that much good, but at least the prediction should be such that the compound is not misclassified. That means a toxic compound should not be classified as non-toxic, and non-toxic compound should not be classified as toxic. So that much clarity in classification should be there. So for that, we have also done classification based metric analysis by considering the mean value of the source compounds at the threshold, and we have compared the predictions against that threshold and found out the number of true positive, true negative, false positive, false negative, etc. And then we have gone for calculation of sensitivity, specificity, precision, accuracy, F measure, G means, Cohen's kappa, Matthews correlation coefficient, as is done in any usual classification based QSR problems. So, and we have also developed a Java-based application for prediction of the this nanotoxicity data using read across, which is available from this website. The web address has not still come on your screen. It is coming. You will see that there is a link given from which you can access it, but the access is restricted. One has to fill up some form and do the license agreement form signed, and then the permission is granted to download the files. And here the input is given from using training and test data in XLS format, and user has to provide the value of hyperparameter like sigma, gamma, etc. If you do not know about the values, then you can give value 1 as default, and then number of close similar compounds, the distance threshold and similarity threshold. And the program generates 2 output files. One is the predicted response, and another is the experimental response, sorted experimental response with respect to the distance and similarity. These are some of the snapshot of running of the program. These are the program output files that one can see while running the program. Now, these are toxicity prediction using the Euclidean distance-based similarity estimation. As you can see, for 3 different data sets, taking the number of source compound 9, 8, and 8, only 9 and 8 and 8, it is very difficult to develop a QSR using 9 or 8 compounds. Well, that is not statistically meaningful. All reviewers would reject the paper because you have so limited number of compounds. So it is unbelievable that you have got a compound that will have good stability, robustness, and predictability, and it can be used for a large number of compounds. So that is not believable. So reviewer will immediately reject your paper. So in this case, the external validation matrix, you can see the for dataset 1 and 3, these are passing the values of threshold that is more than 0.6, but in case of dataset 2, it is lower. So we have to optimize the different constant values. Now, I have already shown that we are using different hyperparameters like sigma in case of Gaussian and gamma in case of Laplacian and cases. You see that in case of data set 1, the best values are obtained when the sigma value is 0.75, lowest RMSEP. For data set 2, the best value is coming from sigma value of 0.5, and in case of data set 3, again 0.75. Again, for gamma optimization for Laplacian kernel, we see when the gamma value is 1, it is giving the best value, that means lowest RMSEP in case of data set 1. In case of data set 2, when gamma value is 2, and in case of data set 3, when the gamma value is 1.5. And then we have also optimized the number of closed training components, because all available source components are not needed, because 9 and 8 components even may not be needed. So we have optimized that. We have used 2, 5, 7, 9, and we have seen in case of data set 1, it is giving 5 number of closed training components is sufficient. In case of data set 2, we see that depending on different metric values, the number of closed compound may vary, but and also depending on the approach, the Euclidean or Gaussian kernel. In case of Euclidean, it is only 2, and in case of Gaussian kernel, it is 5, and both Gaussian and Laplacian kernel. And in the third data set, in all cases, only with 4 compounds is giving the best result. So not even the all available source compounds are required. We can optimize the number of closed training compounds. Then we have gone for threshold optimization for distance and similarity. In such cases, we have not used the number of compounds that should be used. Instead, we have used the threshold value for distance and similarity. And we can see for different data set, we have obtained different level of threshold of distance and similarity. For example, for data set 1, the distance threshold of 0.4 and similarity threshold of 0.05 is optimum. In case of dataset 2, distance threshold of 0.5 and similarity threshold of 0. In case of dataset 3, distance threshold of 0.4 and similarity threshold of 0 is optimum. So these are some radar plot to show the results. I'll go to the comparison table. So this is the classification based matrix using the best obtained setting. For Euclidean distance, it is 0.4, and for Gaussian and Laplacian kernel, the similarity is 0. In case of dataset 2, and in case of dataset 3, Euclidean distance similarity 0.4. And we can see the sensitivity, specificity, accuracy, precision, all are 100% in case of dataset 2 and 3. And in case of dataset 1, in 2 cases it is 100%, in other cases near 100%. So as you can see, the classification ability of the model is very good. Now we compare, it's not model, but prediction. So now we compare our results with previous published results. And we have used here our best setting for dataset 1. It is Euclidean distance threshold 0.4. It is showing Q-score F2 value 0.96. Whereas previously published quantitative read across using principal component method, it is only 0.74. And using nano QSR, it is 0.83. As you can see, it is superseding. So we are getting better result in case of dataset 1. In case of dataset 2 also, in case of Gaussian kernel, we are getting Q-score F2.91, but previously published method quantitative read across is 0.8 and nano QSR 0.83. In case of dataset 3, Euclidean distance is showing the best result 0.95 as Q-score F2. In case of previously published quantitative read across using principal component analysis, it is 0.91, and in case of nano QSR, it is minus 0.20. So here actually you can see the read across is performing better than QSR. These are some of the graphical representation to show the result. I will come to the conclusion because I think I have consumed my time. So we have developed here a new read across algorithm based on various similarity estimation techniques. We have used Euclidean distance, Gaussian kernel, and Laplacian kernel. Optimization of sigma and gamma values have been done for Gaussian and Laplacian kernel functions. And we have seen that we can optimize the number of closed training compounds to 2 to 5. And we can find out the threshold, optimum threshold. And we have seen that in most of the cases, the threshold value, distance value is 0.4 to 0.5, and suitable similarity threshold is 0 to 0.5. So if anybody is starting a new work, if you do not know the required threshold, you can start with this threshold for your work, then you can optimize. And this program is a Java-based program. This is available in this web page. You can download subject to fulfilling some requirement. That is, you have to fill a form and you have to sign an agreement. And this similarity-based read-across algorithm are easy to use, efficient, and expert independent alternative method for toxicity prediction of metal oxide nanotubes. So this is the, as you can see from the snapshot from the website that is available. And this is also published in Environmental Science NAD from Royal Society of Chemistry. So this work is supported by ACRB. And those who are new to the field, if they want to know more about read-across and QSR, this is the basic book on QSR, understanding the basics of QSR for application in pharmaceutical sciences and risk assessment. This is a brief form. In Springer brief, you can have an idea about QSR in more brief form. You can quickly go through this. And it's some application of QSA in different fields like drug design, predictive toxicology, and risk assessment. And these are more advanced application in different areas of pharmaceutical, chemical, food, agriculture, and environmental science. These are application of QSA in diverse fields like computational modeling against Alzheimer's disease, multi-target drug design, in silico drug design for drug repurposing, and ecotoxicological modeling. And these two are more recent. In silico modeling of drugs against coronaviruses, published in 2021, and also chemometrics and cheminformatics in aquatic toxicology, recently published. Thank you very much. If you have any question, I'll be happy to answer.
Thank you very much, Professor Kunal Roy, for a nice presentation and lecture, and dealing with this problem, how if you have a less number of molecules using similarity index, and that to how to do that. It's a very important technology, rather I should say. So this session has been very informative for data handling as well as using this. So if there are any questions, I am not seeing any question from in the chat box. There are only good comments. Very helpful session, very informative session. There is no question here. I think from the audience who are here offline.
Any questions, students? No questions. Any questions?
Yes. There are no questions from the audience, and so it means that people have understood. But I think the students must have noted, what he has written several books, and they are asking many times from me that which are the good books or for this. I think you can see this. No doubt in internet they are all available. So you can have this data, but I think Kunal Roy has done very good work, and particularly these things are used in REACH. And I know we were together, and I was initially, so I suggested those people in government of India that we should take Kunal Roy. I think this must be progressing. REACH is very important aspect for the prediction of toxicity of the chemicals. I think with these comments, I thank both the speakers of this session, Mari Reko and Professor Kunal Roy, on participating in this symposium and coming. Thank you very much. Now
Thank you very much. Thank you.
Now I would like to invite Dr. A.K. Saxena sir to honor our speakers of this session and present momentous speakers.
So please, first moment to you. Yeah, Professor Marin Reko. It should be on the screen. Yeah, this is for you, Marin Reko, and I think this will be sent on mail. And thank you very much. Next one for Kunal Roy. Yeah. So Professor Kunal Roy, this is for you.
Thank you very much.
And from here. And thank you very much.
Thank you very much, sir. Now I would like to invite director giver to honor Dr. A.T. Saxena, sir. Please.
where again this is coming up, and that was all concerned the COVID, and the first lecture was of, and the second lecture was of Professor Bhartam from NIPER. Only I think he again gave presented kind of a practical approach and the design aspects which he used. And of course, I already did about my lecture. His lecture was very comprehensive lecture, kind of overview for lecture, and there both the methodologies and also the real application of them. I think this was the lecture. I would not say that it was deeply explaining, but giving the concepts. The most important part in this was that I showed the practical that boosted in practice. I said, I heard a lot of good students, my colleagues have participated in. I think it was almost a lot of last 5 years, and some of the work said, thereafter, Professor Indra Ghosh, you had been she also gave a very nice talk and very comprehensive and here and particularly for COVID, which can be used as so this was followed by next session in which Professor Dr. Ashish Nandi he again spoke of the group forms by cause and group. They again elaborated that part, an analysis of prediction methods and the better prediction of viruses, how does it take place, and the validation of these. This was followed by, of course, our we made very overview of all the natural products and drugs of university and finding out that this was short time. Then my students, Dr. Chandra Saurabh is a very outstanding IST full practice in that this university, and they are paying work on this also establish a whole medicine and China and all, and these books now very good. How we use this knowledge. They do medicine, it is application. Chemistry is a thing. Chemistry is a very what is this where you use chemistry everywhere? How is it? So that was also very good. This was followed by Shan Patma's lecture. He also now connected some way because he's the guide of our last session was very good opportunity which I just share this session in which the first speaker was Martin Reco from Slovenia, and I both lectured how to handle it. Most important, sometimes you have some less of a lot of data, a lot of parameters, how do you use them? How do you use them? Then, of course, there are a lot of statistical problems. If you have very less data, you still want to infer the right information. And that was very nicely shown by Mr. Kunal Roy on his similarity analysis. So I think that the whole symposium design has been very development for others and it's very informative because you know how the government design and the practical part and how to handle the data, get the molecules, different methods, and what is could have been also introduced what is 0D, 1D, 2D. I think those who are online aspects, you must be our experts, but I would say that those students who are here, and those that join the education, that B Farm or F Farm, will come from whole institution, and it's there for all, for the whole of India, for the whole of the globe, because the same is whole is global. And but I think for you people who are very they can take that skill. Okay. In terms of school, I would say that I don't put the context things to that extent, but it has given you something that you have something with you. And innovation, you are inclusive children. Then there are the references, and you can have what is you again, you can consult also with your, and if some of your students, even one or two or three or four of you want to come up in your life, wants to become scientist, wants to know something, and if you are interested, so this is the opening. You can find out references, read yourself. Then if you don't understand, then you can come to the health yard and approach scientists who have giving classes, like every one of them is very good. So I think this is has been a good platform for youngsters, graduate students, and in fact, faculty also in this countries of science. I think this is the summarize the whole.
Thank you very much, sir, for summarizing whole symposium in few minutes. Thank you very much, sir. This symposium now.
First of all, I congratulate the organizing team, especially Director of the Institute, Organizing Secretary, Dr. Sushil Nandi, and this team for organizing such a wonderful event, which has covered all the aspects of drug design, starting from classical approaches to modern approaches and the recent approaches. And the most important approach which has been used for designing the recent pandemic of COVID-19 was very interesting. And with these words, I request all the students to take interest and to analyze carefully what has been spoken in this symposium, what are your interests in which you want to go up and up. So with these words, I congratulate again, and it's a very fruitful and important seminar for students also, for faculty also. They can benefit themselves by discussing all the aspects with speakers, because they all are very open to all the researchers, etc. So thank you so much.
Dr. Professor Dr. sir, to share his experience of this symposium. Please, sir.
Thank you very much. First of all, I would like to say that this is a very nice opportunity for Global Institute of Pharmaceutical Education Research to celebrate the 70th birthday of Honorable Chairman Sir, along with such a intellectual and important symposium on drug design and development. So this event was really a very informative intellectual program. Attendees and researchers, they were going to get an idea to make their research, to explore their research, and to further they can increase it. And in this symposium, the main point is that more than very renowned scientists from more than 14 countries have participated, and all the lecturers. I must say that they are completely getting an idea about how the drug molecules are designed and how this molecular modeling can be done to computer drug design. So overall, I want to say that this overall program symposium on drug design is very informative, and all my students, if they are getting something from it, it will be very helpful for them, and they can have an idea that what the field of pharmacy is and what they can do in future, and how they can and where they can want to stand themselves. So in pharmacy, the career growth is sky is the limit. So you have an idea about those things.
So I congratulate all of you, all my family, all my people family, efforts of all my people family members and staff who have really done a remarkable job to make this program a success. Thank you very much.
Now I would request Organizing Secretary of the Symposium, Dr. Sisi Nandi sir, to say few words regarding this symposium. Please, sir. Now I would request to Dr. Kapil sir, sir, please say few words regarding this symposium.
Thank you. First of all, I would like to say that it was in hybrid mode. Hybrid means online as well as candidates were there that were present physically, and many hundreds were present in the online mode, in the Zoom and in the YouTube. And this event can be at any time, can be seen any time in the YouTube. We will share the links. I think it is already shared with you. In this event, many institutions locally appeared in this event, and all plus as 14 countries people, professors, researchers, academics were there. And it is the first time in our history that we are having audience of the 14 countries. And I think right now they are also listening to us. The lecture given by Professor Dr. That was very interesting. And this day, 70 days will be record for a long time for the celebration of this birthday celebration of Dr. And we will remember it forever. That's one thing that was seen in this event, and that was the representation of the poster in the online mode. Really, I have never seen in any institution, national or international wise, where the posters are presented online. There is a link in the event section. People PDS poster presented. All posters that we have received are in the online. First, whenever the successor told us that to arrange such type of event, such type of organization, that poster will be available online, and any professor and person can give their comment. And it is the vision of Dr. Saxena that these things were first handled by us. And you will surprise to know that our expert has taken 4 days to create a single page. 4 days was with that single page of 24/7 extract. What is 40 extracts are there? And in last, it is a memorable day, and it was really good for all students, the audience that are presented online and offline. Thank you.
Now, last but not the least, Organizing Secretary of this event, Dr. Sisi Nandi sir. Sir, please say few words.
Now it's the moment for memorizing and summarizing all the lectures and posters. It is really difficult to exist in the current scenario globally because pandemic situation is going on for the last 2 years. But still, we are elite and we have been struggling for our existence. Or scientist Darwin said, struggle for the existence. We have been struggling for the existence because our mother nature gifted us medicines, foods for maintaining our lives and existence. So I say so many times to my students, to my colleagues, there is no substitution for mother. There is no substitution for mother nature. Mother nature equivalent. We must have to take care of our mother nature, who gives us life, who gives us food, who gives us medicine. So under this current scenario, we we must have to take care of our environment and Mother Nature. Overall, we invited speakers, poster presenters from multidisciplinary areas of pharmaceutical, medical sciences, environmental protection, ecotoxicity prevention. They have delivered the talk from which we gain a lot of knowledge on the drug design and drug discovery research, because they have discussed the latest art of drug design and discovery research. Professor Kunal Rai is my parent teacher from Jadavpur University. He delivered the last talk, and he has shown one link where his group developed one software. That link I have copied. As you are the young scholars, you can search in the Google by putting that link, and you can access that software, because Sir has announced that you can access the software for drug design and toxicity modeling research. For that, we need to have some documentation, but Sir has said document will be, we shall do some agreement, it will be okay. If you would like to access the software, it will be free. This is for your good information. So as a young student, at least you can do some research work through accessing this kind of software. This is very good. For that, I shall definitely help you how to operate this software. And now, when all the laboratories were closed under the pandemic situation, we are under a lot of tension. We have done a lot of fight. But what scientists they did, they devoted themselves. Within the 4 work, they devoted themselves to protect the lives. It's true. So when, as a scholar, young scholar, you have been kept at home. Don't waste your time searching the mobile. You search the correct thing through mobile. Read some work, potential work. Our students, our scholars, they have presented very nice work. Kopil sir, Ankita ma'am evaluated. Kopil sir already told about the poster presentations, that it is really difficult to predict which one is the best. Really difficult. So like that, all other scholars, you motivate yourself to put into the constructive way, how to spend your time in a constructive way. In Uttarakhand, this JIPA is the institution who organized conference at an international level. Even government institution also, they are thinking many times how to organize, what to organize. But we are under the umbrella of Dr. Saxena, and Sir enriched ourselves to teach from each and every point of view. We are fortunate to encounter that point of view. So we regularly first at the first year of its establishment in the year of 2010, from 14 to 16 countries scientists came here. It's very, very impressive, very, very impressive. Then again 2012 conference was there. In 2013 conference, 2014, CFTPI organized in 2017. This is the first time in India under the chairmanship of Dr. Saxena and under the chairmanship of European Professor DDDR. This is the first time in India CFTPI conference organized here. We are very proud that we are the part of JIPA. We are the members of JIPA, and you are the heartbeat of JIPA, students. Our strength comes from you. So we have heart, but you are our heartbeat. That is why you have to learn. In this conference, seven speakers from outside, 7 speakers from abroad. And receivers from India. So during the pandemic situation, in this current scenario, when online conferences are there, organizations, they are organizing conference, 1 or 2 lectures per day. But in a session, we have arranged 3 lectures per session. So it's very impressive. We have been organizing. Again, we will organize for you. This is for you only. Definitely we are learning, but you are the forefront. You are the forefront of learning. So priority should be given to you. When lecturer deliver the lecture, they ask first priority should be the young scholars and young students. So try to understand the importance of this. Definitely, from this conference, one thing I have learned. Diploma, I have forgot their name. 3 students came to me. They have discussed with me application for curcumin against cancer. And from that discussion, I got one idea. I called them. I told after the conference we shall sit together and we shall write a review what. Who are the students? Please stand up. These are the 2 very brilliant students. You will come. We shall write a review article because we have given one tremendous idea. So you are the young. Your brain is very young. So by the discussion, teacher is also learning. That is very important. This is the outcome of our seminar, that our young students, they have learned, they are enormous generator of new ideas. Thank you very much.
Thank you very much, sir, for your words of wisdom.
Hello. Now, from the audience, young scholars, young researchers, now this session is devoted for you, devoted to you. If you have any query, questions, please raise your hand. If you would like to say something, what in Hindi, you speak in Hindi, speak in English, whatever you want. What experience you have achieved and what you would like to achieve. Want to achieve, what is your opinion? So you please share. Raise your hand, please. Who would like to share 1 2 sentences? Raise your hand. Hindi, you can speak in Hindi. Faculty also.
Any student?
Student, faculty, staffs.
Yes, Javed, please.
First of all, I want to congratulate you for your 71st birthday. I have a question. I have a question. What advice you want to give to the new students who want to follow this research? Thank you.
Very nice question, and I think this must be a question. First of all of you should do education, and I'm very happy that yes, some of you very good because you should all always online program a lot of सबसे इम्पोर्टेंट है कि आपका हेसिटेशन माफ होना अभी बस सोचिए कौन कि आप कुछ बोलेंगे तो लोग क्या सोचेंगे या नहीं इसको खत्म कर जिस दिन आपने अपने इसको खत्म कर दिया तो आप अपनी लाइफ में आगे सकते तो वन थिंग क्या सोचा आपको जो अंदर से आ रहा है पूछिए अगर आप नहीं पूछेंगे जैसे क्लास में लेक्चर भी दे रहा है कभी भी आप पूछना चाहिए मेरी समझ नहीं आया so that will remove hesitation to get हुआ अब क्या है तो यहाँ से क्या मुझे इसको नहीं बहुत ज्यादा इंग्लिश मीडियम नहीं था मैं स्कूल तो मैं बोल नहीं पाता स्कूल का कैसे उसको पता था हेजिटेशन है ना आप बैंक का शीशे के सामने खड़े हो
डीएनए में उसका कैसा लगता है ये रिकॉर्ड होगा तो उसके टेप ऑन करता है सुनता है कि मेरा कैसा उससे बात क्या आप बहुत स्पीकर बन जाते हैं तो आपको अप्रिशिएट करते हैं ये एक बात उसी बात आपका रिसर्च में सबसे इम्पोर्टेंट चीज है जब भी आप कोई चीज देखिए उसको दें तो आप उसमें क्यों हो रहा है तो क्यों हो रहा है तो आप मुझे देखिए कैसे हो रहा है क्या हो रहा है अब डीप में जाएंगे साइंटिस्ट बना न्यूटन के बारे में जब वो सब देखते हैं एक से तो उसका सोचते कि नीचे क्यों गया ऊपर तो आप क्वेश्चन करेंगे अपने से तो आप उसको आंसर देंगे तो वहीं पे इनोवेशन आप एकदम इनोवेटिव माइंड रिसर्चर्स का इनोवेटिव माइंड हम हर चीज में इनोवेशन देखिए चाय बनाते घर में वो भी इनोवेशन है कितनी चाय पत्ती डालनी है कितनी अदरक कितना टेस्टी बनाना है क्या फ्लेवर आता है तो अच्छा होता है आपको चाय बड़ी अच्छी बनाई केमिस्ट्री हो गई वही तो केमिस्ट्री इसमें आपको इसमें जॉब को अपनी सक्सेस पानी है तो आप जॉब को जॉब की तरह ना समझ के उसको अपनी हॉबी बनाइए उसमें इंटरेस्ट है तो आपको फिर अकेले नहीं करना इसमें मजा आता है तो आपने जिस दिन खेल बना दिया उसमें इंटरेस्ट लेने लगे आप भी सक्सेस हो ये अब इससे आपका कैसे करेंगे तो अंदर आपकी याद है तो आप लोग पढ़ेंगे वही पढ़ेंगे बहुत लोग सुन भी रहे हैं लाइफ है many students who used to come for research with me because research के लिए आते थे ट्रेनिंग के लिए आते थे पोस्ट ग्रेजुएशन के लिए कल आपने कहीं मुझे मैसेज पढ़े मैसेज तो मैं यही कहता हूँ मेरे पास तो एक क्या है रिसर्च करवा सकता हूँ मलेरिया में करा सकता हूँ और अल्जाइमर में करा सकता हूँ ट्यूबरकुलोसिस में करा सकता हूँ बहुत कुछ करा दूँ मैं कैंसर करा सकता हूँ स्टूडेंट्स पता जो मेरे पास आए और आप बेस्ट स्टूडेंट आते हैं एमएससी का हुआ या एमएससी एमटेक एमकॉम उनके पास ठीक है एक लड़का है बीटेक का था पिछली बार आया था उसने अपना वो लिखा तो हाँ स्टूडेंट्स को हमारे पास आते हैं तो बहुत फीलिंग हो क्योंकि गवर्नमेंट एजेंसी सेलेक्ट करती है बैंक से आगे क्वालीफाई करती है और अच्छे होते हैं नो डाउट लेकिन जो भी होता था उसका तो तुम्हारा इंटरेस्ट है उसका कैंसर मैं प्लस होता था मैं इसको कैंसर पे ही टॉपिक दूंगा तो इसको मजा आता मैं कभी को मंगेगा मैं चले जाओ अब जाएगा तो जाएगी वो नहीं तो वो लाइब्रेरी में जाएगा कौन सी किताब लेगा उसको लगेगा कैसा नहीं है तो मेरे में काम कर रहा है सब ने बोला नहीं पढ़ने लगेगा क्या कैंसर एक घंटा खराब लगता है शाम को उत्तर को बताना तो मैं ऐसी चीज करता हूँ उसको भी अच्छी आप मिलते हैं तो सिर्फ आप वही काम करोगे जीवन में जिसमें आपको अच्छा लगे तो आपको मजा आए जब मजा आएगा तो आप करेंगे आई दिल नाम करेंगे मजा आता है मैं वो करता हूँ तो मुझे आगे भी करता हूँ तो ये आपको जब डिसिप्लिन आपके अंदर जाएगा तो आप बढ़िया से बढ़िया ऊंची और एक बात और बता दूं जब आप किसी चीज में एक्सेल करेंगे तो मनी आपके ऊपर से बरसता आप जॉब के पीछे जॉब आपके पीछे आप रुपए के पीछे रुपये आप
Any committee members? Yes, please.
क्वेश्चन है आयुर्वेदा और फार्मेसी को लेके कैसे इंडस्ट्री डेवलप कर सकते हैं क्या कर सकते हैं आई थिंक आपने जो सुशीला जी का लेक्चर था सुना उसमें स्टेप को थोड़ा कवर किया है दो बातें हैं जो ये रिसर्च है तो अगर रिसर्च में आएंगे तो सारी जो हमारी मॉडर्न मेथड ऑफ मेडिसिन है जिसको आप एलोपैथिक बोलते हैं एलोपैथिक ड्रग्स है आयुर्वेद विधि से हम यूज कर सकते हैं बट मैं जो है आपको मोस्टली
यानी ऐसी तरह से आपको पता है कि आयुर्वेदा इसी की में आयुर्वेदा काम करता है और आयुर्वेदा बहुत स्ट्रांग हमें कोई किसी का डॉक्टर उसको पूछे तो मैं कहूंगा कि वो बता कि जहां पे प्रूवन है 100% है बैक्टीरियल इन्फेक्शन तो मैं कहूंगा आपको एंटीबायोटिक पे जाना चाहिए आयुर्वेदा उसको इतना बढ़िया है इसको वंस अ वीक लॉन्ग टर्म प्रैक्टिस है सब में आयुर्वेदा के अंदर तो डिसीज को जैसे किसी के किडनी स्टोन हो किडनी स्टोन आई वुड से आपके आयुर्वेद द्वारा ये काम देखोगा होम्योपैथिक का होम्योपैथिक सिस्टम है तो ये देखना है कि किस स्टेज में कौन सी चीज ज्यादा इफेक्टिव है ज्यादा और बैक्टीरियल चीज को बताया था कल्चर सेंसिटिविटी करो बैक्टीरिया पड़ा यही आपको 24 घंटे में ठीक होता है उससे एकदम ऊपर नहीं उस तरह दूसरी मैं कहता हूं मैं आयुर्वेदा वालों को कहता हूं आपके अपनी साइंस को यू शुड ट्राई टू डू दिस वे दैट prove that is the only difference science is on experimental if you show and it is there इस तरह से आप आगे बढ़ सकते हैं बट आपको इस पोजिशन वाले में अपना व्यू आप क्या बोलते हैं कोई भी स्टूडेंट आपको कैसा लगा आपको अंदर से क्या आया अगर कोई इसमें कहोगे सकता कैसा ऑर्गेनाइजेशन था अच्छा लगा कि खराब लगा वैसे यू कैन आल्सो हैव फीडबैक फॉर्म्स
Okay, thank you very much, sir.
one thing only I
Now I will request Dr. A.K. Saxena sir to declare results of e-poster and offline poster. Please, sir.
I think so the first prize I think is a combined for both online and offline posters. So the first prize goes to Ms. Ishita Day from University of Kalyani. The second prize goes to Priyanka Saw, University of Kalyani. The second prize in the there has been a tie or something in which second prize is for 4. I think there will be competitive one. The first was Priyanka Saw, University of Kalyani. Second is Priyanka Day from Jadavpur University. The other one, Ekta Khale from IIM University, Gwalior. The fourth same category is Priyanka Chaje, BCS, DDDR College of Pharmacy, Maharashtra. One similarity, I think, that there are 3 Priyankas. Priyanka So, Priyanka Day, and Priyanka. And I think this is something. So I congratulate all participants for the second prize. And the third prize goes to Mr. Munir. The certificates will be sent by online because most of them are online and so
any comments? And would like to congratulate and say that it has been very hard effort of team of who have organized this symposium. I have been sharing symposiums, organized more than 100 symposiums, many symposiums where and I think and I was this part has been done by the team of because he has been associated with someone who are bad, one was okay. But I mean, this is a tension is coming up. And as I am known all over the world, I'm not saying in India, the best organizers of conferences. I always feel that people will come up in the same rank as what time CDR existing in Texas. Yeah, so this is a culture I think all of you should learn. Organizing of a conference, art also. Every aspect of the book down. So I think they are learning and they have done very nice. One conference which was organized in Goa, it was also very outstanding. It was on a fully international level offline conference. Many of the fellows went there, they attended it and they organized it. So I think this is coming up. Whatever I would say, journal, by mistakes you learn. You do something that you have to think that this is not. This goes on into it. And the whole thing is in your life also. So I would be very happy that I think Shila is wishing that we have organized CPPMS 2020. So usually this every 3 years we shall be doing because 2020 and 2023 and go forward. So I think even if those who are on online will be prepared to, if it is on all offline, the online offline hybrid mode, I don't know, but CTPMS will be there in 2023. These words I once again thank you all the organizers, the Chipper family, and all for this outstanding performance.
Many of the participants of our CTPMS and DDDR, they have requested to the organizers that in the next conference it will be nearer to the airport. Yes. Many of the participants of CTPMS and this, they have requested that in the next international conference to be hosted by JIPA, that should be nearer to the airport.
I think this is a very good suggestion idea because many of my friends and persons and others would like to come from abroad and it takes time from coming from very onwards. So I think we shall look into it and maybe, hopefully, might be possible that we can have an airport within 50 kilometers from here. So we have one person, but maybe it's not that very operative, maybe other one may come. Otherwise, we will do that. We will take care of it and we shall do. And that is, thank you very much.
Thank you very much, wishes and blessings. Thank you once again. Now I would like to invite Dr. Sushinathan sir for to please vote of thanks. Thank you very much, sir.
So now this is the moment for sharing the emotions and feelings which are coming from the heart by getting knowledge, experience, advices, suggestions from this conference. On behalf of the organizers, I would like to one minute. On behalf of the organizers, I would like to thank to all invited speakers, chairpersons, poster presenters, online, offline, young scholars, students who participated and supported us, and they have discussed multidisciplinary areas of pharmaceutical, medical sciences, environmental protections, etc., etc. All the speakers, they have been briefed by Dr. Saxena. So, particularly by name, I don't want to extend anymore because we have very time limit. Now, evening is coming very soon. After that, this tea session is also there. So, I convey my regards to all the honorable speakers. I convey my regards to honorable chairpersons, especially Dr. B.B. Amos sir. He is so devoted and he maintained a very good time with us from the beginning to end. Professor Hajare sir, Professor Vidula ma'am, and I would like to thank to Honorable Chairman of the Institute and Patron of the Symposium, Dr. A.K. Saxena sir, Professor Vidula ma'am, co-patron, Dr. May Aruni Saxena, co-patron. I would like to thank to our very honorable and dynamic director, Dr. Deepak Teotia. So, under the sir's guidance, director guidance, we have a very competent team. We are able to do, we can do anything. This strong determination we have now. Thank you, sir. And we have very efficient and very potent and very good friend, co-chairman Dr. Kopi Kumar. Kopi sir maintain the web uploading of poster session, and this is to say that this is the novel idea which is coming and nobody has done till now, that posters they have been uploaded to the website so that globally it can be seen and globally interaction for getting the knowledge. Some posters resolution quality is poor. We have sent email to the poster presenters that please send us for the high resolution data, and that will be replaced by the old one. So poster session is in online, which is not that today conference saying that poster session closed till online. No, it will be many more days also for learning, because I do not have, the organizers did not have any time to so much see the posters. So we can also get knowledge through these uploaded posters. Now, no words is coming to thank the co-organizing secretaries, Rajan and Sarfaraj, very young, very dedicated. One newcomer, new madam, who very recently joined, but was very active and very potent, Opuma Joshi ma'am, I would like to share one sentence. After completion of the work yesterday, after completion of the lecture and poster session, while returning back, I and Kobil sir was going by car. Sarfaraj, this man or and sir, they are going by a motorbike, and it was in the evening. So we went with a great happiness and energy, and we have in our mind that we shall be there and to wish our chairman sir, and we would like to have our chairman sir more and more time, more and more time. Okay, thank you, and no words to thank to management. Cultural, he arranged along with the students, very decorated, we can see here, well decoration is here, from beginning of the gate, institute, from beginning of the gate to this seminar hall. Thank you students and thank you management. And you know, we get energy from food, we get energy from the food. So no words, by giving thank to food committee, including Sandha, Amit, Monir, Bhavna, Vaishali, Mohit, Kumar. They paid themselves, they have given a lot of time. And this registration, during the registration committee, Adia, Ankita, Swati, they have given all the time. Thank you very much. And regarding seminar hall arrangement, our Varun sir, Dimti, they have devoted themselves. Nice, you have done very nice work, sir. And our very small member, Tonu ma'am, she has done excellent job. Because whatever document we need, whatever document maintenance she has saved in the computer and she has been time to time. Thank you, Tonu. And you know, regarding transport, we cannot move a single inch without transport under this situation. So transport committee, Amita Ikram, thanks a lot. Our chief guest came from the Pondava airport, and Ikram escort the chief guest. Thank you, Ikram and Amit, very young and important members. And Deepak, Rahul and Aman, they have operated a nice computer operation under the guidance of us, and of course, sir has guided. And no words to say how peons devoted themselves. Excellent. Our peons, Sunil, Bhupen, Kasiram, they have devoted themselves to maintain the seminar hall and everything. Very smoothly it goes. And I convey my regards to caterer who prepared the food, and also decorator who built the tent above the roof, and it was smoothly done. Thank you very much. And a big thanks to news fellow who has who has circulated our news in 24 hours TV, 24 Ghanta, 24 into 7 days, 24 into 7 channel. Our news is there, white is there. You will be having. I shall submit it in your groups. Okay. Yes, 24 Ghanta and Suragina channel. So thank you very much, sir, for your active support and cooperation towards circulation of the message. Finally, I would like to salute our doctors, nurses, medical staffs, pharmaceutical and medical scientists, other medical staffs, other administrative staffs, police department. I salute all of you for your devotion to protect our life and situation. Thank you very much. And now the program officially ends. I invite you all for the high tea. Please, thank you very much.